2022, Nuevos Horizontes.
Es un honor poder contar con todos ustedes. Este simposio constituye el evento final, broche de oro de nuestro curso de posgrado de Inmunoterapia 2022, desde el gen hacia el paciente, que se llevó a cabo en la Facultad de Ciencias Exactas y Naturales de la Universidad de Buenos Aires durante las últimas 2 semanas y al que han asistido 130 jóvenes estudiantes, científicos, médicos, biólogos, bioquímicos, farmacéuticos y veterinarios de todo el país y del continente. En este simposio homenajearemos y celebraremos las carreras y vidas de gigantes que han construido, cada uno desde su lugar, los pilares de la inmunoterapia en cáncer y en enfermedades autoinmunes. El doctor James Allison, la doctora Padmani Sharma, el doctor Lawrence Steinman, la doctora Lori Glimcher, el doctor Ignacio Melero y el doctor Sebastián Amigorene. Nuestro sistema inmunológico tiene la capacidad de reconocer y controlar amenazas externas e internas, microbios como virus, parásitos, hongos, bacterias y también células malignas o tumores. A su vez, despliega un abanico enorme de mecanismos homeostáticos, células regulatorias y señales inhibitorias que contribuyen a resolver la respuesta inflamatoria y prevenir respuestas autoinmunes. La inmunoterapia capitaliza estos mecanismos, los potencia y logra incrementar la respuesta inmune en cáncer y suprimirla o atenuarla en enfermedades autoinmunes o inflamatorias crónicas. En este contexto hemos sido testigos de una revolución enorme en inmunoterapia en cáncer, con las terapias de anticuerpos que bloquean puntos de chequeo inhibitorios como CTLA4 y PD1, anticuerpos agonistas de moléculas coestimulatorias y las terapias adoptivas y estrategias de vacunación con neoantígenos y terapias adoptivas con células T quiméricas o CAR T-cells. A su vez, la inmunoterapia en enfermedades autoinmunes e inflamatorias ha logrado diseñar estrategias más racionales, selectivas y seguras. Estas terapias, que se implementaron a partir de la investigación de mecanismos básicos del sistema inmunológico, han logrado salvar miles y miles de vidas a lo largo de todo el planeta, aumentando dramáticamente la sobrevida libre de enfermedad en un gran número de pacientes. El éxito de estas terapias en la práctica clínica nos recuerda, y le recuerda a nuestros gobernantes, la necesidad imperiosa de apoyar la ciencia básica para lograr implementar avances verdaderos, robustos, sólidos, que se traduzcan en nuevas terapias efectivas en la práctica clínica. En este contexto, y especialmente en nuestra región, nos queda un largo trecho por recorrer para lograr que estas terapias sean accesibles a todas y a todos, en absolutamente todos los rincones del país, en todos los hospitales, en todas las ciudades y todos los pueblos, logrando mucho más inclusión. Todas y todos tienen el derecho de vivir más dignos, más y mejor. Gracias a todos los que hicieron esto posible, al Ministerio de Ciencia, Tecnología e Innovación, en especial al ministro Daniel Filmus y al secretario Juan Pablo Paz, por apoyarnos en esta aventura y ofrecernos esta casa para hacer nuestro simposio. Gracias a la Facultad de Ciencias Exactas y Naturales, donde soy profesor, especialmente al decano Willy Durán, al exdecano Juan Carlos Reboreda, porque esto comenzó la organización hace 4 años ya, antes de la pandemia, iba a ser Inmunoterapia 2020 y ahora terminó siendo 2022. Y a la vicedecana Valeria Levy por ofrecernos toda su ayuda y apoyo. Gracias al CONICET, acá representado por la querida Ana Franchi. Gracias al IBIME, representado por su directora, Victoria Lux. Gracias a las fundaciones sin fines de lucro que confiaron en nosotros y nos apoyaron para realizar este evento. La Fundación Bungeborn, la Fundación Sales, la Fundación Williams, la Fundación Barón, gracias a los laboratorios que nos apoyaron, al laboratorio Bristol Myers Squibb, BMS, y al laboratorio Bagó Bioprofarma, acá representado por Sebastián Bagó y Leonardo Fernández. Gracias a las sociedades que nos acompañaron, la Sociedad Argentina de Inmunología y la Sociedad Argentina de Oncología Clínica. Gracias a mi querido codirector de curso que aceptó esta aventura, el doctor Jorge Geffner, cuando se lo comenté, café de por medio, allá por el año 2018. Gracias a todo mi equipo que participó en el curso y que me está ayudando muchísimo, desde la recepción hasta absolutamente todo, los quiero muchísimo. En especial, gracias inmensas a la doctora Ada Blidner y Victoria Zumblad. Sin ellas hubiera sido imposible organizar este simposio. Ellas son investigadoras de nuestro laboratorio que han hecho un trabajo excepcional y se lo pusieron ahí en sus hombros. Sin el compromiso enorme de ustedes hubiera sido imposible organizar todo. Gracias especiales también a Amalia Boto por su enorme ayuda en este último tiempo. Los invitamos entonces a esta aventura, a explorar la historia de los pioneros de la inmunoterapia y a celebrar sus logros, que son los logros de toda la humanidad. Gracias inmensas, doctor James Allison, doctor Padmani Sharma, doctor Sebastián Amigorena, doctor Ignacio Melero y doctor Lawrence Steinman por honrarnos con su visita. Now I will shift a little bit to English so you can understand, but at least the names you understood. Great. So good morning, welcome to the Symposium Immunotherapy 2022, New Horizons in the Treatment of Cancer and Autoimmune Diseases, in our house, the House of Science and Technology in Argentina. This symposium is the closing day, the final event of the Immunotherapy 22 postgraduate course, which took place in the School of Exact and Natural Sciences at the University of Buenos Aires during the last 2 weeks and attracted more than 130 students from all the provinces of Argentina and several countries from Latin America that are here in this row. Today, we will honor and celebrate the scientific careers and lives of pioneers who have built the pillars of immunotherapy in cancer and in autoimmune diseases. Dr. Jim Allison, Dr. Lawrence Steinman, Dr. Lori Glimcher, Dr. Padmani Sharma, Dr. Ignacio Melero, and Dr. Sebastián Amigorena. Our immune system has the ability to recognize and control external and internal threats, microbes like viruses, parasites, fungi, and bacteria, but also malignant cells or tumors. At the same time, this tightly regulated system displayed a myriad of homeostatic mechanisms, regulatory cells, and inhibitory signals that contribute to suppress or attenuate inflammatory responses and prevent development of autoimmune diseases. Immunotherapeutic strategies capitalize on these mechanisms and potentiate them, enhancing immune responses in cancer and suppressing or attenuating them in autoimmune diseases and chronic inflammation. In this regard, in the past decades, we have witnessed a revolution in cancer immunotherapy, mainly with the development of immune checkpoint inhibitors like CTLA-4 and PD-1, blockers, agonistic antibodies against co-stimulatory molecules, vaccinations with neoantigens, and adopted transfer of T cells and chimeric antigen receptors or CAR T cells. On the other hand, immunotherapy of autoimmune diseases is paving the way by offering more rational, selective, and safer immunotherapeutic modalities. These treatments, which emerge from the investigation of key basic mechanisms, have saved thousands of lives all over the world. The success of these therapies in the real world reminds us and our governments the compelling need of supporting basic science to implement new effective therapies in the clinical practice. In this context, and particularly relevant to our region, to our countries, there is an urgent need to make these therapies available to everyone, in all the country, in all the cities, in all the towns, and in all the public hospitals of our country. Everybody has the right to live a longer, healthy, and worthy life. Thanks to all of you who work so hard to make this symposium possible. To our Ministry of Science and Technology, our Minister Daniel Filmus, and the Secretary of Science Juan Pablo Paz for supporting this adventure. to the School of Exact and Natural Sciences, especially to the Dean, Dr. Willy Duran, to the ex-Dean Juan Carlos Reboleda, and the Vice-Dean Valeria Levy, to CONICET and IBIME, to Ana Franchi, the institution where we work, the non-profit foundations for science, Bunge Born Foundations, Sales Foundations, William Foundations, and Baron Foundation. To BMS, Bristol Myers Squibb, here with Mara, and to Bagó Bioprofarma Laboratories, to the Argentinian Society of Immunology and the Argentinian Society of Clinical Oncology, to Dr. Jorge Geffner, the director of our course and the symposium, who accepted this adventure, to all my lab team, particularly many thanks to Ada Blidner and Victoria Zumblad, two research associates from my lab who worked so hard to make it possible. Without your commitment and passion, it would have been impossible to organize this symposium and course. Thanks also to Amalia Boto for your help. Now, we invite you to join this adventure, to dive into the history of these giants, the pioneers of immunotherapy. Let's celebrate your achievements, which are the achievements of all mankind. Thanks so much for visiting us in Buenos Aires, Dr. Jim Allison, Dr. Padmani Sharma, Dr. Sebastiana Migorena, Dr. Ignacio Melero. It's a great honor to have you here. Thanks so much.
Toma la palabra el decano de la Facultad de Ciencias Exactas y Naturales de la Universidad de Buenos Aires.
Gracias. Hola a todos y todas. Enorme placer poder estar acá en este hermoso evento. Y el primer felicitado por todo esto tiene que ser mi querido amigo Gabriel Rabinovich. Esto es espectacular. Digo, termina en una conferencia, pero viene de una escuela de 2 semanas y esto no hubiera sido posible sin el trabajo que Gabriel hizo para que esto existiera. Así que nada, fundamentalmente desde la facultad, agradecerte, Gabriel, decirte que no te vamos a dejar escapar de la Argentina, que está bárbaro que tengas buenos vínculos con todo el mundo, pero que la facultad te va a encadenar si hace falta para que sigas haciendo este tipo de cosas que marcan la diferencia. Yo le decía recién al querido amigo Daniel que este tipo de actividades marcan la diferencia. Primero, la importancia de la vinculación entre la investigación básica y aplicada. A veces se trata de mostrar que hay algún tipo de contradicción entre ambas y para nada. Y este tipo de actividades muestran que llegar desde la investigación bien básica, que es esencial para tener buena investigación aplicada, y llegar hasta el paciente, es lo que tenemos que hacer, es hacia donde camina la ciencia, y por supuesto que la facultad va a estar apoyando eso en todo momento. Así que nada, reitero ese agradecimiento a Gabriel y ojalá que pueda seguir organizando este tipo de actividades y va a tener todo el apoyo de la facultad para hacerlo. Agradecerle al otro amigo que está por ahí, Jorge Geffner, que no es de nuestra facultad, pero que es casi como si lo fuera, que también venimos trabajando en conjunto, hicimos varias cosas en la pandemia durante 2 años ahí, desde mi lado más cuantitativo, desde el lado de él más médico, pero trabajamos juntos con el gobierno nacional, con el gobierno de la provincia de Buenos Aires, muy cerca para tratar de pelearle a la pandemia, en tiempos donde no nos podíamos ver. Me parece que está buenísimo esto, que nos podamos ver acá y podamos juntarnos. Otra cosa que me pareció significativa de este curso de 2 semanas fue la presencia de gente de todo el país. Yo pasé varias veces por el aula del Cero Más Infinito, ese hermoso edificio que tenemos en Ciudad Universitaria, y el aula, una de las aulas grandes de ahí, que tiene como 110, 120 asientos, hermosa aula de seminario, tenía gente sentada en las escaleras, en cualquier lugar, y por lo que me decían, gente de toda la Argentina. Y me parece que también eso marca la diferencia, ¿no? Quizás a veces la Argentina es muy grande y no siempre los recursos han estado particionados equitativamente en todo el país. Entonces, la posibilidad de tener estos tipos de eventos bien federal me parece que es un gran logro y también es para felicitar a los organizadores en ese sentido. Y después agradecerle a otros amigos que están por acá, a Daniel, que está acá al lado, el ministro de Ciencia, a Juan Pablo, que veo ahí, el secretario del Ministerio de Ciencia, a Ana, Fernando, digamos, al CONICET y a la agencia, que siguen apoyando la ciencia en tiempos que son difíciles, pero están siempre cerca y apoyando este tipo de actividades. Así que nada, me parece que está bueno resaltar eso. Y no mucho más, le dejo la palabra a Daniel. Por último, in English, congratulations to the prestigious professors that are receiving today this important award of our university and our School of Science, and thanks a lot for being here. Gracias a todos y todas.
El ministro de Ciencia, Tecnología e Innovación de la República Argentina dará unas palabras de bienvenida.
Muy buenos días a todos y todas. Bienvenidos, bienvenidas. Una alegría enorme que estén aquí con nosotros, especialmente a quienes van a ser distinguidos por la Universidad de Buenos Aires, que es nuestra querida universidad para la que la atravesamos y una de las universidades más prestigiosas de América Latina y del mundo. Es un honor, un orgullo que este evento se realice en la casa, como decía Gabriel, de la ciencia, que es el Ministerio y este Polo Científico-Tecnológico. Estamos acá para agradecer, para felicitar y estamos acá para comprometernos. Estamos para agradecer y felicitar, en primer lugar, el trabajo enorme que hacen los científicos, en este caso en inmunoterapia. Nos parece imprescindible, lo plantearon ambos que precedieron el uso de la palabra, la vinculación entre la ciencia básica y la ciencia aplicada para nosotros es fundamental, es decisivo que esto llegue después a los pacientes, que llegue en el sentido de lo que planteaba Gabriel Recel también, en forma igualitaria a todas y todos. Tiene sentido si esto puede realmente resolver problemas de salud y evitar millones y millones de muertes en el mundo. Por eso les agradecemos profundamente el trabajo que realizan y la posibilidad de democratizarlo y trabajar con países y investigadores como los de la Argentina, que realmente en condiciones difíciles desarrollan una tarea enorme. Quiero también felicitar y agradecerle a Gabriel, a Jorge y decirle que esté tranquilo, Willy, porque si se portan bien, dentro de poco van a estar ahí en Exacta, muy cerca de Exacta, porque estamos planificando construir el edificio de varios centros de investigación, un gran edificio de investigación con 5 centros de investigación, va a estar el centro de Ana también, va a estar allí, en la ciudad universitaria, así que van a ser vecinos, vas a poder reclamarle todo lo que quieras desde muy cerca. Y es importantísimo que ello ocurra. Cuando nos contó la iniciativa Gabriel, nos pareció que no podía estar el Ministerio ausente. Como decía Willy, lo valoramos a Gabriel, valoramos el trabajo que hizo su equipo y valoramos más todavía que lo haga en la Argentina. Para nosotros es fundamental, es decisivo que nosotros podamos también tener de la Argentina un desarrollo científico-tecnológico que nos coloque a la par del que se hace en los países centrales. Nos parece excelente que el curso haya tenido, y están aquí creo, los participantes del interior del país, de las distintas provincias que no comúnmente tienen estas oportunidades, y trabajar para que realmente distribuyamos el conocimiento de esa manera y que demos posibilidades de investigación absolutamente a todos y todas, nos parece central. Decía que agradezco, felicito y nos comprometo. De casualidad me enteré que el doctor Amengorena, uno de los que va a ser reconocido por la Universidad de Buenos Aires, es argentino. Y también de casualidad me comentó el doctor Amengorena, recién cuando lo saludé, que es hijo de científicos que se tuvieron que ir en la noche de los bastones largos. Entonces creo que también esta posibilidad que tenemos hoy de entregar el doctorado tiene un sentido reparador. Y esa es la parte del compromiso. Nosotros necesitamos comprometernos que en la Argentina no haya más políticas pendulares respecto de la ciencia. Necesitamos que en la Argentina, y copiando en cierto sentido o tratando de imitar lo que pasa en los países centrales, la ciencia y la tecnología sean políticas de Estado. No podemos permitir más idas y vueltas porque el golpe que significa eso para el desarrollo de los jóvenes, de quienes tienen que elegir la vocación tempranamente hacia la ciencia, es muy difícil si uno no tiene un futuro y empieza o a no elegir carreras científicas o a pensar de que la única forma de desarrollarlo es fuera del país. Entonces, felicitamos, agradecemos y nos comprometemos en trabajar para que esto sea así. Hemos tenido buenas noticias últimamente, Gabriel, lo sabes bien. La ley de financiamiento de la ciencia fue por unanimidad. La ley de desarrollo de la bio y nanotecnología, que compartimos aquí con Bagó la posibilidad de presentarla junto con el presidente de la Nación. La ley que está por salir de crédito fiscal para las empresas, la ley de economía de conocimiento, todas las leyes salen por unanimidad. Eso quizás nos garantiza que dejemos de lado la idea de que la ciencia tiene que estar atada a los calendarios electorales y que depende de quién gane una elección si se la va a seguir apoyando o no. Así nadie va a seguir la ciencia y así tendremos, a pesar del talento que tiene la Argentina, seguiremos teniendo formas distintas de interrumpir lo virtuoso de ese avance. Así que, reitero, los felicitamos, les agradecemos que estén en la Argentina, les agradecemos, sabemos que es un esfuerzo, depende cómo uno mira el mundo, podemos estar en el fondo, nosotros ahí abajo en el sur, pero sepan que nuestra decisión de apoyar la ciencia y la valoración del esfuerzo que se hace, no lo hemos sentido quizás nunca tanto como en la época de la pandemia, que hemos visto las dos cuestiones. Cómo hoy podemos estar todos acá, creo que es una de las primeras veces que estamos todos sin barbijo, que podemos estar todos tranquilos justamente por el aporte de la ciencia, pero también vimos que la desigualdad respecto de la ciencia hizo que algunos países que concentran y monopolizan el conocimiento decidieran sobre la vida y la muerte de toda la humanidad, que pudieran definir quiénes eran los primeros que se salvaban y quienes no tenían esa oportunidad. Y en ese sentido tenemos que dar la pelea como das vos, Gabriel, desde acá, como da todo tu equipo, como dan todos quienes están en tu facultad, nuestros investigadores del CONICET y de las universidades. Así que felicitaciones, gracias y no les quepa duda que nos comprometemos a trabajar fuerte en este camino.
So, good morning, everybody. It's a pleasure to be here, and I would like to congratulate Gabriel Rabinovich and all of the outstanding personalities here. Today concludes a full 2-week seminar, which would not have been possible without Gabriel. So just don't even think about the possibility that we will let you get away. Because it is you who make things that actually make a difference. There is an absolute link, as the two previous speakers mentioned, between basic and applied science. And we really must make sure that from basic science, it gets to the patient as a form of applied science. And we will provide support to your school always. This is a full commitment. I would like to acknowledge as well Jorge Hebner, who's there around. He's not from our school, but we met during the pandemic. We worked from different areas. But I think this is the first time that we all get here together, and it's good to see you all without face masks. And this is also thanks to science that this has been possible. Another source of joy for me is to see that we have a very general attendance from all ways of our country. And it is beautiful to see that, because that makes the difference as well. Because sometimes resources are not distributed in an equitable manner. So this is another achievement worth mentioning. As I said at the beginning, I would like to thank, among others, Daniel, the minister, the secretary, Ana, Fernando from CONICET, and well, as we are commemorating or celebrating some of you here, We want to welcome those who are being awarded the recognitions. It is an honor to have you here and also a pride to see you all here in this scientific and technical site. I would like to commend as well the work of scientists in the field of immunotherapy. And as already mentioned, so that basic science gets to applied science to everybody so that we can save millions of lives. So thank you and welcome to the researchers from all of Argentina. Thanks to Jorge and Willy as well. And let me tell you that we will be building 5 research centers so that you can all get a hold of each other and pass on whatever complaints you may have. So we will work because this is important to happen. And without the initiative of Gabriel and the minister, this event would not have been possible. And Argentina needs development both in science and technology. And it is excellent to have the participation of so many of people from different countries here, because sometimes this has not been possible. And I would like to thank once again those who are receiving the awards today. A while ago, I had the possibility of greeting Dr. Amigorena, and I learned that he is Argentinian and that he actually was forced to exile himself during what is called in Spanish, La Noche de los Bastones Largos. So this event also serves a repairing role, and that is where the commitment I also wanted to stress comes into play. So we want to make sure that there are no longer back and forth policies. We need to make science a state policy regardless of the political color in power, because this impacts on young people and their decision for their lives. Because if it goes back and forth, they will probably not be choosing a career in science, or they would rather go abroad and make a professional career over there. So, congratulations. Thank you for the effort of coming down here, depending on how you look at it. You could say that we are the southernmost country. And you know that we have made the decision to acknowledge you and to support you and science. We saw, especially during the pandemic, that there were many inequalities, and some few countries were actually the ones who had the power to decide who could live and who would die. So we want to continue developing our research so that we can make it more equitable for everybody.
Perdón, propongo un aplauso para la traductora. Excelente.
A continuación leeremos un mensaje del rector de la Universidad de Buenos Aires, profesor Ricardo Gelpi. Por este medio, quiero enviar mis saludos a todos los participantes del Simposio Internacional de Inmunoterapia 2022, que hoy los congrega, y en el que se hará entrega del título honoris causa de la UBA a profesionales de la talla internacional que hoy nos visitan. Es un honor para la Universidad de Buenos Aires y en particular para mí, como representante de nuestra comunidad universitaria, reconocer mediante estos títulos honoríficos, a los más grandes referentes del campo de la inmunoterapia. Sus carreras, su prestigio, sus trayectorias y sus méritos sobresalientes los hacen indiscutiblemente merecedores de la más alta distinción que otorga nuestra universidad. Felicito a nuestros 6 doctores honoris causa, Dr. James P. Allison, Dr. Lori Glimcher, Dr. Sebastián Amigorena, Dr. Padmani Sharma, Dr. Ignacio Melero y Dr. Lawrence Steinman. Ustedes, con su presencia, enaltecen nuestra universidad, afianzan nuestro campo del saber y proporcionan esperanza a las y los futuros investigadores. Por otro lado, como investigador, profesional de la salud y docente, creo imprescindibles los intercambios entre científicos de nuestro país y del mundo, que se producen en el ámbito de encuentros como el que hoy nos convoca. Son enriquecedores en todo sentido y en este caso particular tienen una propuesta abierta a un público más amplio. Tanto colegas como jóvenes estudiantes y público en general pueden acceder a escuchar y preguntar directamente a los más respetados investigadores e investigadoras en inmunoterapia. Por eso agradezco a los directores de este simposio, los doctores Gabriel A. Rabinovich y Jorge R. Geffner, grandes científicos que nos representan en todo el mundo y que hoy son los anfitriones de este encuentro. En ellos también felicito a cada uno de nuestros investigadores e investigadoras, nuestro mayor orgullo y quienes ponen a nuestra universidad y al país en lo más alto del desarrollo de las ciencias. A todas y todos los presentes les deseo que puedan disfrutar de ese simposio y esperamos que siempre tengan deseos de regresar a nuestra casa. Muchas gracias y buena jornada. Ricardo Helpi, rector de la Universidad de Buenos Aires. A continuación, damos comienzo al acto en el que se hará entrega de los diplomas que acredita a los profesores Lori Glimcher, Sebastián Amigorena, Padmani Sharma, Ignacio Melero, Lawrence Steinman y James Allison como doctores honoris causa de la Universidad de Buenos Aires mediante resoluciones del Consejo Superior número 1081/22, 1082/22, 1083/22, 1084/22, 1085/22 y 1133/22. Presiden este acto el ministro de Ciencia, Tecnología e Innovación de la República Argentina, profesor Daniel Filmus. El decano de la Facultad de Ciencias Exactas y Naturales de la UBA, doctor Guillermo Durán. El profesor doctor Gabriel Rabinovich, investigador superior de CONICET y director del Laboratorio de Glicomedicina de IBIME CONICET. Nos acompañan también profesores, alumnos e invitados a quienes agradecemos su presencia. Afterwards, Professor Gabriel Rabinovich will present the laudatio to introduce Professor Lori Glimcher.
So, Dr. Lori Glimcher is a magna cum laude graduate of Radcliffe College and holds an MD degree cum laude from Harvard Medical School. She held the position of Professor of Medicine at Harvard Medical School from 1991 until 2012. In 2012, Dr. Glimcher became Dean and Professor of Medicine at Weill Cornell Medicine and Provost for Medical Affairs at Cornell University. In 2016, Dr. Glimcher was named President and Chief Executive Officer at the Dana-Farber Cancer Institute. She is currently the Director of the Dana-Farber Harvard Cancer Center and the Richard and Susan Smith Professor of Medicine at Harvard Medical School. Dr. Glimcher's pioneering research identified key molecular pathways that contribute to autoimmune, infectious, and malignant diseases. Of note, she identified several critical transcription factors that control CD4-positive T-cell differentiation. Among her major discoveries, Dr. Glimcher discovered the CMAF transcription factor that regulates IL-4 expression during Th2 differentiation, that Tibet's controlled commitment of interferon gamma producing Th1 cells, and that the XPP1 transcription factor is required for the development of antibody-producing plasma cells. More recently, Dr. Glimcher identified the IL-1 alpha XBP1 pathway as a central immune escape mechanism in ovarian cancer and thus a novel target for cancer immunotherapy. Dr. Glimcher is member of the U.S. National Academy of Science, the American Philosophical Society, and the U.S. National Academy of Medicine, and is a former president of the American Association of Immunologists. She is also the recipient of many awards and honors, including the American Association of Immunologists Lifetime Achievement Award, the William Coley Award for Distinguished Research in Basic and in Tumor Immunology, the Stephen Beering Award from the Indiana University School of Medicine in recognition of outstanding research contributions to the advancement of biomedical or clinical sciences, and the L'Oréal UNESCO Award for Women in Science. She is also the recipient of the 2012 Leloir Award from the Minister of Science and Technology in Argentina, and the 2017 Award for Distinguished Research in the Biomedical Sciences from the Association of American Medical Colleges. In addition to her research efforts, Dr. Glimcher has been a staunch advocate of improved access to health care, progressive health policies, and medical education, while simultaneously serving as a pioneering mentor and role model for cancer research trainees and all students in science. For her fundamental discoveries and transformative achievements, Dr. Lori Glimcher has been distinguished and is being distinguished today as Doctor Honoris Causa, the highest recognition awarded by the University of Buenos Aires.
Dr. Glimcher was not able to attend, so we will listen to her thankful message.
I am immensely honored and privileged to receive an honorary degree from the University of Buenos Aires. And I'm also very humbled to be receiving this degree with Nobel laureate and my dear friend, Jim Allison. This first-class, world-renowned university has been a fount of exciting discoveries that have improved the lives of patients with many different diseases. The University of Buenos Aires is the premier institution of higher learning in the country and one of the most prestigious universities in North and South America and the world. I was very impressed to discover that it has educated 17 Argentine presidents, produced 4 of the country's Nobel Prize laureates, and is responsible for approximately 40% of the country's research output. And I especially want to offer my gratitude to the following individuals. And that's the Minister of Science, Technology, and Innovation of Argentina, Professor Daniel Filmus, the Dean of the University of Buenos Aires, Professor Dr. Ricardo Gelpi, the Dean of the School of Exact and Natural Sciences of the University of Buenos Aires, Professor Dr. Guillermo Duran, and finally, my dear friend, Professor Dr. Gabriel Rabinovich, who's head of the Laboratory of Glycomedicine at IBYME CONICET, and a senior researcher at CONICET. So thank you again for this immense honor. I am so grateful to all of you. I am immense.
Seguidamente, el profesor Dr. Gabriel Rabinovich formulará la laudatio académica al profesor Sebastián Amigorena.
Dr. Sebastián Amigorena was born in Buenos Aires, Argentina, and earned his PhD degree in Paris in 1990. After 3 years of postdoctoral training at Yale University, he returned to Paris to an independent position at the Curie Institute. Dr. Amigorena has provided numerous seminal observations that have contributed to our understanding of the immunobiology of antigen presentation in both physiologic and pathologic settings and have profound implications for cancer immunotherapy and vaccination. His main scientific interests are at the frontiers of immunology and cell biology. Dr. Amigorena is a worldwide leader in studies focused on antigen presentation and cross-presentation by dendritic cells. His work elucidated critical endocytic and phagocytic pathways and described several unique molecular components associated with the specialization of both pathways. He also made significant contributions to our understanding of the dynamics of cytotoxic T cells and their contributions to the rejection of solid tumors. More recently, he made several pioneering observations on the role of epigenetic reprogramming and its contribution to T cell memory formation. Moreover, he has identified two distinct populations of cell precursors that generate T lymphocytes that infiltrate tumors in non-small cell lung cancer. Dr. Amigorena has received numerous awards, including the prestigious Senior European Research Council ERC Award, and is currently a member of several journals, editor boards, including Science. Currently, Dr. Amigorena holds the title of Outstanding Scientific Director at CNRS and leads the Immunology Department of Immunity and Cancer, as well as the newly created Cancer Immunotherapy Center at the Institute Curie, a unique center devoted to translational research in France. Dr. Amigorena has been an active collaborator with scientists worldwide, most notably with colleagues in Argentina. From his fundamental and transformative discoveries, Dr. Sebastián Amigorena is being distinguished today as Doctor Honoris Causa, the highest recognition awarded by the University of Buenos Aires.
Hace entrega del título y la medalla que acredita al profesor Sebastián Amigorena como doctor honoris causa de la Universidad de Buenos Aires, el ministro de Ciencia, Tecnología e Innovación de Argentina, profesor Daniel Filmus. Next, Professor Gabriel Rabinovich will present the laudation to introduce Professor Padmani Sharma.
Dr. Padmani Sharma was born in Guyana and was awarded both MD and PhD degrees in immunology from the Pennsylvania State University. She completed a residency in internal medicine at Cornell University Medical Center, New York Hospital, followed by additional clinical training at the Memorial Sloan Kettering Center in New York. Currently, Dr. Sharman is professor of genitourinary medical oncology and immunology in the Division of Cancer Medicine at the MD Anderson Cancer Center, Houston, Texas, where she specializes in renal, prostate, and bladder cancers. She's also the scientific director of the Immunotherapy Platform and co-director of the Parker Institute for Cancer Immunotherapy at the MD Anderson Cancer Center in Houston. Dr. Sharma's research has focused on the mechanisms and pathways that control tumor rejection. Her major findings include the elucidation of the inducible co-stimulator ICOS pathway and its role in promoting resistance to immune checkpoint blockade. In 2006, Dr. Sharma designed and conducted the first neoadjuvant study featuring the anti-CTLA-4 monoclonal antibody ipilimumab. The result of the study established the safety profile of this drug and prompted Dr. Sharma to conduct other clinical trials, including the one that featured the use of anti-PD-1, nivolumab, to treat patients diagnosed with metastatic bladder cancer. Dr. Sharma was also the principal investigator of clinical trials in which nivolumab alone, as well as in combination with ipilimumab, were used to treat patients with metastatic renal cancer. These studies facilitated the approval of this drug by the US Food and Drug Administration. Her studies also highlighted new mechanisms of resistance to immune checkpoints and paved the way for the design of novel anti-cancer immunotherapeutic strategies. For her pioneering work, Dr. Sharma has been awarded several prizes, including the Emil Frey, I am pronouncing well, Emil Frey III Award for Excellence in Translational Research in 2016, the William Coley Award for Distinguished Research in Basic and Tumor Immunology in 2018, the Women in Science with Excellence Award in 2020, and the Randall Prize in Cancer Research in 2021, among others. She has shown remarkable foresight and persistence in her effort to ensure that cancer immunotherapy will be available to all individuals worldwide in single or combination drug strategies. For all her fundamental and transformative discoveries, Dr. Padmani Sharma is now being distinguished as Doctor Honoris Causa, the highest recognition award given by the University of Buenos Aires.
Now, the Dean of the School of Exact and Natural Sciences of the University of Buenos Aires, Professor Dr. Guillermo Duran, delivers the Diploma and Medal, which recognizes Professor Padmani Sharma as Honoris Causa Doctor of the University of Buenos Aires. Seguidamente, el profesor Dr. Gabriel Rabinovich formulará la laudatio académica al profesor Ignacio Melero.
Dr. Ignacio Melero was born in Madrid. He speaks Spanish. He earned an MD degree from the university. Well, Sebastián also speaks Spanish. Different Spanish. Dr. Ignacio Melero was born in Madrid. He earned an MD degree from the University of Navarra School of Medicine, followed by residency training in clinical immunology at Hospital Universitario de la Princesa, Universidad Autónoma de Madrid. As part of his PhD studies, he pioneered the characterization of a class of natural killer cell inhibitory receptor known as KIRs. In 1994, he moved to Seattle, where he studied mechanisms used by T cells to ignore tumor antigens and the role of T cell co-stimulation in mouse cancer models. His instrumental studies on CD137-mediated co-stimulation in anti-tumor responses resulted in the design of novel immunotherapeutic agents, several of which are currently in phase 2 clinical trials. In 1998, he returned to the University of Navarra, where he is currently a full professor of immunology at the Clínica Universidad de Navarra and the CIMA Investigation Center. His current research focuses on cancer immunology and immunotherapy with a translational approach that proceeds from experimental models all the road to clinical trials. His program has pioneered several new monoclonal antibodies, gene therapy, adopted T-cell strategies, including chimeric antigen receptors. He has conducted more than 40 clinical trials as principal investigator. His work has been recognized with several prizes and awards, including the 2004 Bayal Prize for Exceptional Studies in Medicine, the 2006 Conde de Cartagena Prize of the Royal Academy of Medicine, the Lara Award in both 2011 and 2019, and the Francisco Cobos Award in 2020. Dr. Melero is currently the senior editor of several journals, including Cancer Discovery, Clinical Cancer Research, and Journal of Immunotherapy of Cancer, and is among the most highly cited scientists in biomedicine. Dr. Melero is currently a tenured full professor at the School of Medicine at the University of Navarra and director of expert degree in immuno-oncology, also of the University of Navarra. He has been appointed as an academy member of the Royal National Academy of Medicine of Spain. He has been extremely generous with his time and efforts and collaborates with scientists all around the world. For his fundamental and transformative discoveries, Dr. Ignacio Melero is being distinguished today as Doctor Honoris Causa, the highest recognition award by the University of Buenos Aires.
A continuación, hace entrega del título y la medalla que acredita al profesor Ignacio Melero como doctor honoris causa de la Universidad de Buenos Aires, el decano de la Facultad de Ciencias Exactas y Naturales, profesor doctor Guillermo Durán. Next, Professor Gabriel Rabinovich will present the laudation to introduce Professor Lawrence Steinman.
Dr. Lawrence Steinman, Larry Steinman, was born in Culver City, California. After receiving his MD degree from Harvard University, Dr. Steinman completed his residency in surgery, pediatrics, and neurology at Stanford University. He then received both doctoral and postdoctoral training at the Weizmann Institute of Science in Rehovot, Israel, and the U.S. National Institute of Health in Bethesda, Maryland. He is currently a full professor in the Department of Neurology at Stanford University. Dr. Steinman's research program focuses on fundamental cellular and molecular mechanisms implicated in neuroinflammation and neurodegenerative diseases. The results of his groundbreaking work have generated unprecedented insights and opportunities for the design of new therapies used to treat multiple sclerosis and other autoimmune and inflammatory conditions. Dr. Steinman has contributed several seminal discoveries, including the first identification of pathogenic T-cell clones capable of generating the neuroinflammation and demyelination characteristic of multiple sclerosis. Moreover, together with his team, Dr. Steinman has developed new therapies capable of preventing immune cells from having access to the central nervous system by blocking alpha-4 integrin. This discovery ultimately led to the development of natalizumab, which can be used to treat relapsing multiple sclerosis. Moreover, he has made pioneering discoveries of CNS inflammation and has contributed to our understanding of osteopontin and homeostatic mechanisms involving alpha-crystalline. More recently, his outstanding observations revealed a link between multiple sclerosis and Epstein-Barr virus, which highlighted additional novel immunotherapeutic strategies that might be used to treat the disabling disease. In addition to his scientific achievement, Dr. Steinman participated in several clinical trials and is a founder of several biotech companies, including Neurocrine Biosciences, Byhill Therapeutics, Atreca, Transparency Life Sciences, and Tolerion, that were instrumental in promoting the clinical translation of his discoveries. Dr. Steinman received several prizes and awards, including the S. Ware Mitchell Award from the American Academy of Neurology, the Friedrich Sayce Award in Immunology from the University of Berlin, the John Deisel Prize for Outstanding Contributions in Multiple Sclerosis of the National Multiple Sclerosis Society, Outstanding Inventor Award from Stanford University, and the Charcot Prize for Lifetime Achievement in Multiple Sclerosis Research from the International Federation of Multiple Sclerosis Societies. He's also a member of the U.S. National Academy of Science and the U.S. National Academy of Medicine and the American Association for Advancement of Sciences. Larry Steinman has collaborated actively with several scientists from all around the globe. For his fundamental and transformative discoveries, Dr. Lawrence Steinman is now being distinguished as Doctor Honoris Causa, the highest recognition awarded by the University of Buenos Aires.
And now, Professor Gabriel Rabinovich delivers the diploma medal, which recognizes Professor Lawrence Steinman as honoris causa doctor of the University of Buenos Aires. Next, Professor Gabriel Rabinovich will present the laudatio to introduce Professor James Allison.
Can you hear me? Dr. James Allison was born in Alice, a small town in Texas, in the United States of America. Dr. Allison received his PhD degree from the University of Texas in Austin in 1973 and engaged in research at many institutes of higher learning, including the Scripps Clinic Research Foundation in La Jolla, California, the University of Texas System Cancer Center, the University of California, Berkeley, Memorial Sloan Kettering Cancer Center in New York, Weill Cornell Medical Center in New York, and the Howard Hughes Medical Institute in Chevy Chase, Maryland. In 2012, he took on the position of full professor at the University of Texas MD Anderson Cancer Center in Houston. Among the highlights of his distinguished career, Dr. Allison identified regulatory pathways that control T-cell responses. This groundbreaking research has pioneered a revolution in cancer treatment, as his findings have paved the way to our unleashing the immune system so that it can attack tumors. As part of his work, Dr. Allison has established an entirely new principle for cancer therapy. As but one example, his findings revealed that CTLA-4, which is a protein present on the surface of activated T cells, functions primarily as a brake on the immune system. Dr. Allison was the first to realize the therapeutic potential of releasing these brakes, thereby providing immune cells with the capacity to attack tumors. His work has led to the development of ipilimumab, the first in class of drugs known as checkpoint inhibitors. As a group, these drugs have yielded unprecedented responses in patients with advanced melanoma. In October 2018, Dr. Allison and Dr. Tasuku Onjo shared the Nobel Prize in Medicine in recognition for their discoveries that led to a novel type of cancer therapy through the inhibition of negative immune regulation. Among numerous prizes and awards, Dr. Allison became a member of the United States National Academy of Science and received the 2015 Lasker-Bakey Clinical Medical Research Award. On a more personal note, Dr. Allison has always been extremely generous with his time and reagents and has collaborated with many scientists around the globe to expand the field of cancer immunotherapy. Currently, Dr. Allison is a professor and chair of the Department of Immunology at MD Anderson Cancer Center. He's also the executive editor of the Platform of Immunotherapy and director of the Parker Institute for Cancer Research. He's also Deputy Director of the David Koch Center for Applied Investigation in Genitourinary Cancers. He also created and performs on harmonica each year at the American Association of Clinical Oncology and the Society of Immunotherapy of Cancer with his blue band that he created known as the Checkpoints. For his fundamental and transformative discoveries, Dr. James Allison is now being distinguished as Doctor Honoris Causa, the highest recognition awarded by the University of Buenos Aires.
Now, the Minister of Science, Technology and Innovation of Argentina, Professor Daniel Filmus, delivers the diploma and medal, which recognizes Professor James Allison as Honoris Causa Doctor of the University of Buenos Aires. So that we can make a group picture. De esta manera, damos por finalizado el acto. Agradecemos a todos su presencia, agradecemos la presencia del señor ministro y los invitamos a escuchar las conferencias de los homenajeados. Ahora. Cuando se retiren las autoridades, invitaremos al profesor James Allison a dar el discurso de apertura.
Este lo usé, sí.
¿Lo usaste? Pero este que estaba antes, ¿lo usaste? Listo. Este se lo trajo recién a Alice.
Ah, buenísimo. ¿Cómo lo empezamos a presentar nosotros?
Sí, ahora es todo tuyo.
Ah, bueno, porque no. Gracias a vos. Gracias. Gracias.
Felicitaciones de vuelta y vamos hablando.
Gracias, sí.
Yes.
Ahí está. Yes. So we will now start with the first talk. Now we will start with all the symposium. We have the 6 talks, and we will start with the first talk of Dr. Jim Allison. Go back to the first one. You want to the previous one? See the title.
We invite Professor James Allison to pronounce the opening lecture entitled Immune Checkpoint Blockade in Cancer Therapy, Historical Perspective, New Opportunities, and Prospect for Cures.
I will be here in case you need. And this is a water for you.
Okay. Thank you very much, Gabby, and I want to thank all of you, the distinguished scientists and leaders in science in Argentina who were responsible for us being here, and to the students of the course that is just finishing up. I'm so happy to have a chance to talk to you guys. Just before I talk, I want to just wish all of you success in your careers. It's hard work, but if you keep doing it, you'll have not only get some stuff done, but enjoy it and have fun along the way. I certainly have. So what I'd like to do today is give you, first of all, a bit of a background on the science behind checkpoint blockade, some notes about where we are, just brief description of where we are in the field of checkpoint blockade in cancer. And then because there's a lot of confusion about checkpoint biology and about, most people think there's 2 checkpoints, CTLA-4 and PD-1. They're quite different, actually, in their mechanisms of action and what they do. And I want to spend some time talking about that, the mechanisms, how they differ, and why they work together so well. And then finally talk about some things that, new stuff that we're doing that I've added to the talk, largely because of Dr. Rabinovich and his work to acknowledge the importance of myeloid cells, and also to Dr. Amagreno, where some of us are going sort of next in what we're doing in this field. So most of the work I'm going to show you, the work goes back a long ways, particularly to a really terrific graduate student named Max Trummel, who, with me, worked out the understanding of the biology of CTLA-4 and the early work on showing its activity, its applicability to treating cancer. The work I'm going to show you today is largely the work of a postdoc, actually 3 postdocs, Spencer Way, Stephen Mock, and Naveen Sharma. And so, I'll just first review where how complex we finally realized how T-cell activation is by the mid-90s. This just shows that until then, a lot of people in immunology just thought the holy grail was recognition of antigen MHC complexes on the surface of cells and recognition by the antigen receptor. But it became obvious pretty soon after we had worked out the structure of the T-cell receptor and made some antibodies to it, that was not enough. There were additional signals that were required. A number of labs showed that, and that they could only be provided by dendritic cells that you know about here, and that Dr. Amagrena has told us so much about. Dendritic cells present not only the antigen receptor signal, but also co-stimulatory signals by expressing molecules called B7s, B7-1 and 2, that bind with a molecule that we showed was CD28 on the surface of T cells, and contemporaneous recognition of those 2 signals turns on this pathway, I'm not going to go through the details, but it induces anti-apoptotic factors, components of the cell cycle machinery that allows cells to divide, growth substances such as IL-2, and so the T cells take off. You know, we've each got about 10 to the 10th T cells probably of different specificities, and so you've only got a very small number of any one of them, and so you've got to build an army from those cells before you have enough to go out and deal with a virus infection or, as we will be discussing, a tumor. And so you need hundreds of thousands to millions of those cells, and you've got to generate them from a very small number of cells within a few days to a week. So you go from maybe a dozen cells to hundreds of thousands to a million. And so these are the two signals that initiate that. And these cells go and as I said, generate the army by initiating this program. But what we learned, along with Jeff Bluestone in the early 90s, was that also initiates an off program by activating the gene that encodes a molecule called CTLA-4 that's very homologous to CD28. It binds the same ligands, actually, but binds them with an avidity about 4 to 5 logs higher than CD28 because of some special features of their structure. And that accumulates with time and gradually stops the proliferative process, we think, by just out-competing CD28 for its ligands. That's a little bit controversial. Some people think that it actively signals, but I don't think there's any results that don't really convince me that that's essential to the process. In any event, it turns off all this stuff. It doesn't kill T cells and actually stops them. You know, it was thought when this work was being done that T cells expand for a while and then they either get tired and die, which, you know, is kind of a nice way to describe it, but not very satisfying to a scientist. Getting tired is a little bit hard to define, almost as hard as exhaustion, which I'll be talking about later. But anyway, it was also thought that maybe it was activation-induced cell death, and that's part of it. But it turns out that CTLA-4's job is to stop that process, and we know that because TAC-MAC and Arlene Sharp and Cynthia Chambers, the late Cynthia Chambers, while in my lab, showed that if you knock out CTLA-4 and T cells, they can't stop dividing, and it's a lethal defect, and mice die of just rampant lymphoproliferative disease by the time they're about 2.5 to 3 weeks old because their T cells react to environmental antigens and just can't stop dividing. So it's a critical downregulator of immune responses. You have to have it to stop that. And so its relationship to cancer was not necessarily obvious, but one of the things that we realized early in our studies after the B7-1 and B7-2 molecules were discovered was the lack of ability of the immune system to destroy cancer cells was not due to lack of antigens, as many people had proposed, because we could put B7 molecules in almost any tumor in mice and they would not grow. They'd be immediately rejected. So they would elicit a very strong and then protective T cell response. So there was no problem with the antigens on cancer cells whatsoever. The problem is that they did not express the B7 molecules. And so, wait a minute. I've gone all the way back, excuse me. My thumb is too big here. This is not moving it. Okay. So anyway, tumor cells are therefore, after they start dividing, are invisible to the immune system. Again, not because they don't have antigens, but because they don't have that second signal. So the only way that the immune system can detect them is when the tumor gets big enough, let's say, and runs out of oxygen or growth or whatever, dies, causes an inflammatory response. Dendritic cells come in and deal with that by phagocytizing dying bits of the tumor cell, then the DNA in their cytoplasm is detected by DNA sensors, much in the way they would detect viruses or bacterial products, but it's not a very particularly strong signal as we know now, and probably doesn't result in full activation. In any event, that though gets antigens expressed on the surface of the dendritic cells in the context of B7, and all that stuff that I talked to you about happens, including the CTLA-4 program. And so what had occurred to me when we began to put all this biology together was that might be the problem, because the tumor gets a head start, and if it gets so big that you can't generate enough T cells to take it out, it wins, the tumor wins. And so had the idea that if you just disable the brakes with an antibody to CTLA-4 and just let the T cells keep going as long as you need them in a way that's reversible, because if you stop giving the antibodies, everything comes back to normal. That might be useful in treating cancer. Anyway, I'm not going to show you. Oh, and that was compelling for two reasons. One is you notice that the drug is active on the T cell. It has nothing to do with the cancer cell. So this is potentially a universal treatment for cancer, because the kind of cancer shouldn't matter to the immune system. You know, the immune system doesn't know the difference between a prostate cell or a melanoma cell. It just knows that there are mutations in there. There are things that are different than normal and ought to be attacked just as if there was a virus in them. Now that sounds great, except it's not quite true. For several reasons that we could talk about later. It doesn't work universally, although we hope to make it that way. But anyway, there are limitations. And the second thing, though, is because this process starts with cell death, you could add CTLA-4 blockade to conventional therapy such as chemotherapy, radiation, hormone deprivation, all the things that you're aware of, and give those agents some of the benefits of immunotherapy, one of them being polyvalency, because the immune system can detect potentially a large number of the mutation products in the cancer cell, not just the ones that the genetically targeted inhibitors like BRAF, BRAF inhibitors and MEK inhibitors and all that. It can target the changes associated with the whole carcinogenesis process, not just one molecule. And the second one, and thereby giving those things, not just the polyvalency, but also memory, because once you get the T cells, of course, a lot of them stick around as slowly renewing stem-like cells, and when the tumor comes back, can be reawakened to deal with the tumor again. And also the thymus is there, and the immune system's a living thing, and can, once you get it started, adjust to changes in the immune system, unlike chemotherapy or radiation, where once you give them, the drug is gone, and you're left with whatever effect you had when the drug was there, but there's nothing lasting about them, which is, of course, been the goal of immunotherapy forever. So this is just one of an example of many experiments that we did for several years, trying to convince the clinical establishment that this was something worth trying in the clinic. But this is just a transplantable tumor growing on the backs of mice. You can see it grows. We have to euthanize the mice after a few weeks because it gets so large. If you inject antibodies to CD28 and you block that co-stimulatory step, the tumor grows faster, telling us that there was a nascent, inherent immune response to the tumor, but it just couldn't get the job done. But we injected antibodies to CTLA-4, the tumors would grow for a while and then just precipitously be destroyed. And the mice were permanently immune. We could challenge them for the rest of their lives again and again with 100 to 1,000 times as many tumor cells, and the mouse would still reject them. And so this proved that the idea of checkpoint blockade, CTLA-4 blockade, had some relevance. And after a while, we managed to, as was mentioned by Gabby, convince a company called Medarex initially to make a fully human antibody to CTLA-4, and then we developed it with the help of Bristol Myers Squibb, and got it all the way through clinical trials. I'm not showing you the clinical trials, but the trial actually, because of this progression before regression, the end part of the trial wasn't the typical progression-free survival, but was overall survival. So it took over 5 years for that trial. And so in 2010, it was approved by the FDA for treatment of late-stage melanoma. And just to put it in context, at that time, the median life expectancy after diagnosis for late-stage melanoma was 7 months, no matter what. There was no therapy that had ever been approved by the FDA for treating it, and there had been no drug of any kind that had ever prolonged that. before then. But the results of the trial of IPI showed that there was about a 4-month increase in median survival. But the cool thing, there was this tail on the end of the curve at about 3 years that stayed there for 5. But anyway, was published in 2015. This was about 5,000 patients by then, for whom there was 10 years follow-up from all of the early trials. So this is just a meta-analysis. It's not randomized or anything. But that data shows, again, there's an inflection point at about 2 to 3 years where it flattens out, and a little over 22% of the patients were alive 10 years after a single round of therapy with anti-CTLA-4. So they were cured of their disease. And I can use that word, I think, with some confidence and without hesitation or being accused of being, too inflammatory or whatever by anyone, because it's after 10 years. I think people with this sort of disease can forget about looking over their shoulders and wondering if they're going to wake up the next morning having recurred. And actually, 10 years was, of course, just the minimum. There's one patient from the phase 1 trial that's now 21 years out. And there's also several, including a woman that I know quite well who was treated when she was 22. Now she's 37 and has some lovely children that are in their teenage years. So this is a curative disease, a curative treatment for a fraction of patients with melanoma. But why not everybody? Why is it just 20%? Well, I could give you some mechanistic explanations for that. Maybe priming isn't going on while the drug's on board or something. Of course, the more interesting possibility to scientists is maybe there's another checkpoint, because CTLA-4 was the first cell-intrinsic checkpoint that had been discovered. And so Tasuku Hanjo, several years earlier actually, had described a molecule called PD-1 that was on activated T cells that he thought played a role in negative selection in the thymus. But actually at about 2000 or so, the year 2000, when our phase 1 data were coming out, he teamed up with Gordon Freeman and Arlene Sharp at the Dana-Farber, and they showed that there were 2 counter receptors for PD-1, PD-L1 and L2, and that it indeed was a checkpoint, but very different than CTLA-4. For one is, its ligand, at least one of them, PD-L1, is regularly expressed on tumor cells. Like I said, PD-1 is expressed on T cells after activation, but what happens is if the tumor cell, the T cell has specificity for a tumor cell, makes gamma interferon, the tumor cell can respond to that by upregulating PD-L1, which then inhibits TCR signals, it looks like, in the T cell, and defends itself against the attack in that manner. And so very different than CTLA-4, which works at priming. PD-1 works on terminally differentiated effector cells. So right there is a critical difference between the two. And so there's an antibody made to it, like CTLA-4, it worked against a bunch of different cancers. They were mechanistically different, so we put them together in my laboratory and showed that they're additive in mouse experiments. But, Jeff Walchuk and others at Sloan Kettering and elsewhere did a series of phase 3 trials. This was a very large one, showing the combination of the 2 agents. And what you can see here is the 5-year follow-up now. In this particular analysis is 52%. There's other ways of, I don't completely understand, but there's other ways of calculating it, and other analyses have shown it's 58%. But let's just say that we've now turned this disease, which I said had fewer than 3% survivals at 5 years, to now well over 50% survival. So we could cure 50% now, a little bit over half of patients with this deadly disease. But again, why not all of them? So we'll get to that. I'll talk about that, and Dr. Sharma will as well in her talk. But it's not just a disease of melanoma. As you can see here, these are the earliest approvals of CTLA-4, PD-1 antibody, Pembro, Merck Associates, and then the combination of anti-CTLA-4 plus Nivo, which is Bristol Myers Squibb's PD-1 antibody, where that combination was approved in 2015. And since then, this actually is only a partial list. There's probably twice this many approvals now of CTLA-4 or PD-1 in combinations in many different types of cancer that you see here. Of note, I want to just point out that this combination of anti-CTLA-4 plus PD-1 blockade is now advancing in many kinds of cancer because of its effectiveness. And it's also moving earlier and earlier, and now is being used in a neoadjuvant setting. It can be quite effective there. So it's really changing the face of cancer therapy. I'd like to also point out that there's approval of PD-1 antibodies for treatment of cancers. You see cancers of any origin, any cellular origin, that have defects in DNA damage repair and are likely, therefore, to have high loads of neoantigens, including a trial presented at ESMO this year, where there's 18 out of 18 patients who responded to PD-1 therapy, I guess, that all the patients were selected by having a lot of mutations, and that spared them the need to go to surgery for anal cancer. So there's a lot of cancers here. I'll point out that Dr. Sharma has led the trials in bladder cancer, for example. Anyway, in bladder, kidney diseases like that, the response rate's 30 to 40% or so to these agents. So not quite where we're at in melanoma, but there are long-term durable survivals in many kinds of cancer. But as I said, not quite as even up to 60%, certainly not 100%. And also there are other kinds of cancer, such as glioblastoma and pancreatic cancer, that so far we've had little, if any, success with checkpoint blockades. We're beginning to realize why that is, I think, and hopefully we'll make some progress there. So now let's consider these checkpoints. And their similarities and differences. Well, first of all, as I told you, CTLA-4 is hardwired. It's involved in every response. It stops the T cell expansion phase, and the absence of it is lethal. But anyway, it's a hardwired checkpoint, if you will. Whereas PD-1's induced response, the tumor cells put up the ligand to PD-1 to protect themselves against immune attack. CTLA-4 works during priming. PD-1 seems to works against the CD28 process during priming, and PD-1 seems to target the TCR pathway, and it works on fully differentiated cells in the course of their carrying out their normal function. Because it works during priming, CTLA-4 blockade has been shown both in animal models and in clinical trials to bring new clones into the response. We've shown experimentally that it can lower the threshold of TCR signal strength that you need to activate a clone, which may play a role in preventing activation of peripheral T cells by result in autoimmunity. But in any event, it can bring new clones in. PD-1 just expands the ones that are already there. It does not bring new clones into a response. They differ their speed. CTLA-4 blockade is often slow. PD-1 is pretty rapid. As I'll show you shortly, CTLA-4 blockade primarily affects CD4 cells, although can expand CD8s as well, whereas monotherapy with PD-1 affects only CD8 T cells, not CD4 cells at all, from what we can tell based on studies in mice and in patients. As Dr. Sharma will show you later, CTLA-4 blockade in humans can drive T cells into cold tumors, such as she'll show prostate and also renal cancer. But PD-1 doesn't seem to be able to do that. The T cells have to be in there before we'll expand them. Adverse events following responses to CTLA-4 blockade are very frequent. They're less frequent with PD-1. They're the same sorts of responses, colitis, pneumonitis, hepapocystis, a lot of itises, a lot of inflammatory diseases. The common ones are not really autoimmune in that they can be cured with systemic steroids, for example, and the patients can be weaned off of them. But there are cases of rheumatoid arthritis, of type 1 diabetes, and more recently a T-cell mediated cardiomyopathy where T cells are actually attacking muscle fibers, an antigen in muscle fibers of cardiac muscle that can be fatal. So there are serious autoimmune diseases, but these are 1% or so of the patients. And for the cardiomyopathy, I don't have time to talk about it. We developed an animal model and have come up with a way of treating that effectively, at least in mice. Anyway, so as I said, the disease recurrence after response is rare in CTLA-4. It's significant with PD-1, 25 to 35% in melanoma. and lung cancer, for example, 2 years or so after treatment. And as I'll show you today towards the end of the talk, is we've got recent unpublished data showing this is because of fundamentally different effect of those two antibodies, of the blockade of those two signals on the generation of immunological memory. So there is an inherent biological reason for the differences in durability of the response, perhaps. So when Spencer Wade came to the lab, we were beginning to play around with confocal microscopy, which everybody knows now. I'm not going to go through the details, but it's just high parameter analysis that's allowed by using heavy metal ions attached to antibodies. And so you could do 43 parameters analysis by flow. This just shows what a tumor looks like when you group the cells according to similarity, using antibodies to all the major kinds of immune cells in a tumor. The work I'm going to show you used a panel of antibodies to T-cell differentiation antigens. We excluded other cells, B cells, myeloid cells, etc. And we looked just at T cells and had markers for all the major surface antigens, transcription factors, etc. That allow us to tell the different kinds of T cells apart. Also markers for proliferation, for apoptosis, for things like that, so we could tell if they were dividing or not. And we kept track of individual mice so we could relate changes to the outcome of the therapy and figure out which cells were reported responsible for it. So this is the overall experimental outlay. We just give mice tumors, and then at day 13, a time point chosen because that's about when it starts being rejected in mice that are successfully going to be treated, harvest the TILs and analyze them. I'm showing here the data with a tumor called MC38. It's a carcinogen-induced colorectal carcinoma, very immunogenic, lots of mutations. But we've done the same gotten the same results with a very poorly immunogenic B16 melanoma that has very low mutations, very poorly immunogenic. We have to use a vaccine in addition to the checkpoint blockade, but the lessons are the same. And what we learned, with the one exception I'll point out, exactly the same thing we see in patients. Of course, we haven't been able to get successive specimens from patients by getting untreated and treated specimens. We've been able to infer what the things are. So this very complicated slide just shows the groups that we get of T cells when we look at it. And here blue is the isotype controls, red CTLA-4, the green TILs from PD-1 treated mice. And then these, the arrows indicate their effect. The arrows indicate the association of the cells that we're talking about with tumor size. So if the arrow goes up, that means that kind of cell is associated with bigger tumors. And if it goes down, that means smaller tumors. Anyway, something you can see right here is there are two types of FOXP3 positive CD4 cells in this analysis. One that has KLRG1 as a mature cell, the other one KLRG1 negative. In any event, it's really clear with these KLRG1 positive ones at least that there is a great decrease with PD-1, which seems to be by dilution because of expansion of other cells, and even more with CTLA-4. And this is due to depletion of Treg cells by the antibody. That's a property that the antibody that we made to mouse CTLA-4 has. It binds to positive signaling FCR receptors, it's FC, binds to them, and then allows, because of the high level of expression of ICOS on Tregs, it allows macrophages to kill them by virtue of the expression of about 100 times more CTLA-4 than effector cells. So it takes them out, just in the tumor though, not in the peripheral tissue. And I have to describe it that way and just point out that ipilimumab, the antibody to human CTLA-4, does not do that so far as we can tell. Dr. Sharma looked in bladder and kidney patients for evidence of depletion and did not see it. And Tony Rebus did an examination of anti-CTLA-4 of ipilimumab plus tremelimumab, another anti-CTLA-4 antibody. It was purposely designed not to deplete, and neither one of those deplete in melanoma. So this is an artifact well, not an artifact, but a property of the mouse antibody. People are trying to modify the human antibodies to give them that property, and there's an early clinical development. But so far, the work with ipilimumab is just working on the cell intrinsic pathway of CTLA-4, which you see here. And that was defined many years ago by Arlene Sharp's lab and my own, showing that TCR transgenic T cells bred onto either with the wild type or bred onto the CTLA-4 knockout background. If you mix those, put them in a mouse and immunize it, the CTLA-4 ones, the ones lacking CTLA-4 would divide many more times than the wild type. So that is cell intrinsic, and that's what we're seeing in humans now. In any event, if you look at the CD4s, you can see this population of cells goes up greatly. And this is this is a T-beta expressing PD-1 low TH1-like cell that makes gamma interferon TNF-alpha. And you'll hear about this more from Dr. Sharma. As Gabby mentioned, she discovered this in her pioneering 2006 trials in bladder cancer. And you'll hear a lot more about them. I'm going to talk about them just in the context of this therapy. But anyway, you can see they go up. We call them TH1-like because they express a molecule called ICOS, which normal helper T cells do not. And so I'll come back to that a little bit later. That's greatly expanded by CTLA-4, not at all by PD-1. There's another population of what seem to be just immature, undifferentiated T cells there, unactivated T cell, CD4 T cells. It also is not affected. So CD4 and PD-1 does nothing to either one of those. And that population is associated with smaller tumors. If you look at the CD8 population, it's more complex. We found 5 different populations based on different markers, but they come down to 2 important ones. This one, which is PD-1 high, TIM3 high, ISO LAG3 high, has T-BET. These are exhausted, what people now call exhausted T cells that have this triplet, PD-1, LAG3, TIM3, all 3 inhibitory molecules on their surface at very high levels. So these cells are really incapable of proliferating in vivo. So I'll come back to that in a minute. Well, anyway, I'll finish those. Those cells are increased by PD-1, but not by CTLA-4. And that's significant. These are the cells that everybody concentrates on when they say, talk about therapy, say these are the cells that are taking out the tumors. And they're allowed to do so because the PD-1 antibody is blocking PD-1. But what I have to point out is that the cells still have that phenotype. They still have PD-1, LAG3, and TIM3 on their surface. And so when the antibody goes away, and we know they're proliferating here because of markers, not just because the numbers went up, because we have markers showing that they're actively making DNA and actively proliferating. But when the antibody goes away, they're going to stop. And this has been shown by John Wherry and a number of other people have shown that there is a epigenetic event in them that locks them in that phenotype. And so they are irreversibly, it's thought, locked with this ability to not respond unless you block that. And recently, it's been shown that if you add LAG3 to the PD-1, it works with it. I'm not going to go into that. The mechanism is just recently published and hasn't been confirmed yet, but anyway, it does seem to work in clinical trials. Anyway, that's expanded by PD-1, but not by CTLA-4. There's another population of cells which have just PD-1. They're activated, so every activated T cell has PD-1 on it, CD4 or CD8, but it has very low levels. It also has T-BET. And these have other genes that, other pathways activated. That these are just their standard effector CD8 killer cells, granzyme, et cetera, Those are expanded by both PD-1 and CTLA-4. Again, both of those are associated with smaller tumors, and these I won't spend any time on. NKT cells, gamma delta cells, we don't see any changes in. In any event, hit the screen off. Okay. Now see what I'm doing. So anyway, this just summarizes that. CTLA-4, you get these ICOS T1, CD4 ICOS positive TH1-like, and then effector CD8s. And with PD-1, you get these exhausted cells plus the effector cells. So that's the mechanism. They share one target and the others are different. So it might be that the reason they work better is because it might just be the sum of those pathways, or maybe it does something different. Right, an interesting question, where it'd always go afterwards. And so we repeated this analysis just using a fourth group, which is both antibodies together. And so this just shows you what happens with those exhausted cells. Again, increased by PD-1, not by CTLA-4. When you add CTLA-4 to the PD-1, it actually, they actually go down. As you can see here, they go down considerably to below control values. So those cells are actually decreased when you add CTLA-4. Kind of a puzzle, right? We'll come to that in a minute. But not only, this is the Spearman's coefficient showing the association between numbers of cells of that phenotype versus tumor size. And either in control mice or mice treated with PD-1, the more of those cells, the smaller the tumor. But when you add CTLA-4 to PD-1, not only the number got down, but they don't make any difference anymore to the anti-tumor response. They become irrelevant to the anti-tumor response. Okay? But if you go on, if you look at the effector cells, the terminally differentiated non-exhausted cells, they go up even higher when you add CTLA-4 to PD-1, which you might expect, because you're taking off PD-1, the blocking them, plus allowing more co-stimulation to get them. And I'm not going to show you here, but the data, but the CD4s actually go up much, much higher when you give both antibodies too. In any event, what happens is when you give them is the combination collapses from one shared mechanism with two different ones to just the CD4 and just the CD8 effector cells. The exhausted cells don't matter anymore. And so this has implications for therapy where patients now are given an induction phase of CTLA-4 and PD-1 and then a maintenance phase of PD-1 antibodies for as long as 2 years in some cases, whereas the CTLA-4 is not continued. It might be that that's not essential. We don't know. It's going to take a lot of work to change that. But anyway, the scientific question that arises, of course, is what happened to those exhausted cells when you put the combination together. So there's at least two possibilities. One of them is, do we just turn them back into effector cells? Well, again, there's thought to be an epigenetic change in them, so that's possible. And we're looking for fingerprints of that epigenetic state now, but it's unlikely, I think, but we're trying to rule it out. The more interesting possibility is that CD8 T cells may not become exhausted if they have ready access to CD28. If you've taken CTLA-4 out of the picture, so they can very easily, by encountering dendritic cells in the tumor, for example, or maybe they go back to the draining lymph node, or maybe they see it in tertiary lymphoid structures in the tumors. But in any event, if they could have ready access to CD28 co-stimulation, they may be able, it may allow them to avoid exhaustion. Interesting hypothesis that we're now looking into. Okay, so just for a moment, I want to talk about these ICOS cells that Pam discovered. ICOS is a member of the CD28 B7 PD-1 family. It has its own ligand. If you ask immunologists what it does, I'll say it's either on follicular T helper cells and helps make type 2 cytokines and helps T-B cell collaboration and immunoglobulin class switching, or it's on a subclass of FOXP3 positive CD4s that inhibit T cells. Well, neither one of those would be good in a tumor. It would actually be bad. So why are these cells in there? What are they doing? And why are they so good for the response? Well, again, Dr. Schormer first found those in the 2006 neoadjuvant trial. She showed that the cells were there. They go up in not only in the tumor but in blood in patients that got IPI. If she sorted all of the T cells in tumors that express the antigen NY-ESO-1, where she had the antigen can make peptides, she showed that all of the tumor-specific cells that detected NY-ESO-1 peptides had ICOS on their cell surface. There were no ICOS negative tumor-reactive CD4s. And then while Jed Wolchok's phase 3 trial was going on, Jed and his team looked at patients there and found that the patients who had extended survival expressed increases in their blood of ICOS-expressing cells, but those who didn't tended to show no benefit whatsoever. So it's a marker of IPI activity. So I'll show you in a moment. She also showed that it's required for optimal efficacy. It went on to show that it has because it has a PI3Ki binding motif in its cytoplasmic tail, it signals through the AKT pathway and can result in increasing T-beta activity and can be targeted. So very quickly, to show you, this is the experiment that she did, showing this is a model where one of our tumors, B16 melanoma, with a vaccine. If you give that within in this case with just the antibody with a vanilla vaccine, what you see here with nothing in it, just irradiated cells, you get about 25% or so activity. But if you give it with N-acetyl-A4, here you can. Sorry, that's with just the vaccine. And if you give it with in a mouse, excuse me, I'm going to back up. I'm confusing this a little bit. Anyway, if you give N-acetyl-A4 in a wild-type mouse, you get this kind of level of activity, about 90% response. But if you carry it out in ICOS negative or ICOS ligand negative transgenic mice, knockout mice, you lose over half of the efficacy. So I confused the slide with the next one, which shows you can then increase that. This experiment that we did with Pam, where we made a vaccine that has either vanilla vaccine or one that has ICOS ligand on its surface, and give that to mice. And here are all the controls. If you give the tumor plus the vanilla vaccine, you only get about 10% or so efficacy. But if you put ICOS ligand on that same cell, it goes up to better than 80%, almost up to 90%. So that increase there is due to giving signals through the ICOS molecule, through the AKT pathway. And this is something that we've been trying to get into in the clinic for some time. And if you do that same experiment, do this one in ICOS knockout mice, you lose that. So it is signaling through the ICOS molecule. So again, another potential checkpoint, but in this case, I don't know if you call it a checkpoint or not, but a positive molecule that could be targeted with an agonistic antibody to increase it. So this then led us to go back. These cells, the observation that they went up led us to ask where they came from. Because what we found when we applied this CyTOF type of analysis, high parameter analysis, cells that were CD4, it had T-beta, it had ICOS, we could not find in patients, in the PBMC of patients, we could not find those cells unless they'd been given IPI. In mice, we could not find them unless they'd been given IPI, or we noticed that there was a lot of those in mice that were CTLA-4 deficient, if we looked at them when they were young enough, before they died. So we conducted the same sort of analysis before, where we looked both for clusters by looking at cells that were similar and analyzing the same data a different way to look at archetypes to find those cells which are the most different, to look at wild type, heterozygotes, and CTLA-4 knockouts. And what you can see, the next slide, hopefully, is this. Well, it's not rotating. Let's see. Yeah, it's not rotating. But anyway, what you can see clearly is that there's this, here's the blue and the green are hets and wild type. And as these reds are cells that are not found in the mouse, in the lymph nodes, in any of those organs. Particularly interesting is this cluster down here, which is like Australia, is cut off from the other continents. No connections, it looks like. Probably drink different beer there and ride kangaroos instead of horses, I don't know. But in any event, very different world of T cells down there. And if you look at what they are, this just shows again cells that you cannot find. The blue and the green are clusters that you cannot find in lymph nodes of mice. And again, are in PBMC of humans, okay? And we can't do knockout humans, but we can give them anti-CTLA-4. But in any event, in knockout mice, there are our culprits. Those are the ones that you see with the antibody. when you treat humans or mice with anti-CTLA-4. They appear in fairly large numbers in mice that genetically lack CTLA-4. There are these other cells, FOXP3 cell. This guy here is kind of mixed up. And look at this one. I mean, you heard in the introduction and in Laurie Glimcher's announcement that Laurie was one of the ones who worked out the linkage between transcription factors and phenotypes. Look at this guy. I mean, you're supposed to have an individual type, like GATA3 is supposed to be just in Th2s. ROR gamma T is supposed to define Th17s. BCL6 is supposed to define follicular T helper cells. That cell's got 3 transcription factors in it. And we sorted these cells later and showed that they're making all of those cytokines. And so these are really cells that have gone. We think they're intermediates. And taking away CTLA-4 allows them to come up. So why is this? Well, again, this is the pathway of differentiation of CD4 T cells that Laurie and others did a lot to work out. T cells come out of the thymus. CD4 T cells come out of the thymus with a MHC2 restricted receptor and with CD4 on them, but their function is not determined until they see antigen. And then what happens is, depending on the context in which they detect antigen, in part by TCR signal strength, and then the cytokines that are around, et cetera, duration, et cetera, is determined by the STAT molecules involved in signaling, but more by the transcription factor that they end up with, and that determines the cytokines they make. And so again, what Pam found are these cells that have T-bet, but they have ICOS, which normally these guys, and these guys have, but Th1s don't. And so where did ICOS come from? Sorry, there's another way of looking at it where you use the same data, but you look at archetypes, which you define cells which are the most distant and still the most similar. And again, I'm not going to spend much time on this, but this is where those ICOS positive CD4s are. So they're really distant from in their phenotype from the bulk of cells that we see in those CTLA-4 knockout mice. So what happens? Well, it's not anything in the thymus, because we went through the thymus and looked at everything we could. Phenotypically, everything's exactly the same. The size of the thymus is not affected. The syndrome, the lymphoproliferation, occurs in mature cells after they leave the thymus. It doesn't start until cells after that. It's not due to lack of selection, because we did all the experiments where we put a gene in, et cetera, gene product in, and so negative selection was fine. It's not something that has anything to do with proliferation or activation. So what we're left with is we deregulated phenotypic constraints and just expanded the palette, if you will, of possibilities that developing CD4 has by taking away the constraining effects of CTLA-4. And so how does that work? Well, we think what it is that CTLA-4 is always diminishing the effective TCR signal strength. It's always there. It's never there in anybody who's alive, because it's lethal to take it out. So there's some degree of diminution of the overall TCR plus co-stimulation signal strength by CTLA-4. And when you take the molecule away genetically, or by blocking it with the antibody, we think that those developing cells get a supernatural, and I don't mean spooky or cosmic or anything like that. I mean more than you would see in nature normally levels. And it's been shown, there's a precedence for that. Kim Bottomley showed very early on, as soon as TH1, the whole TH1, TH2 lineage demarcation had occurred, she showed that signal strength of cells with a different hierarchy of peptides, for example, different strengths, stronger signal strength tended to give you more TH1s than TH2s. We think that removing ICOS just gives you more effective signal strength. It makes a super TH1 out of it, because you don't have that attenuation that you always have naturally. And it just so happens that those cells make a ton of gamma interferon and TNF, and can even start making granzyme when they encounter a tumor in the tumor microenvironment and kill tumor cells. So again, I should turn the picture off. So anyway, we think that all this does support that when you take CTLA-4 away, you give the T cells more possibilities, and you allow these mixed phenotypes to emerge also. And so, and one other thing is you could track individual components in a thing called pseudotime mathematically by just looking for distance from the last common precursor. And what this enables us to say is that these cells that have Tbet started out with ROR gamma T, FOXP3, and Tbet, and gradually lost the GATA3, and then lost B cell 6, and ended up just with high Tbet. And so they start out with multiple transcription factors, then finally end up with just the one. And so this, I think informs something. Oh, this is just a control showing you that move CTLA-4, it doesn't change anything about CDH. If you move PD-1, you really don't see anything. It's not there in wild type or hets. But anyway, this tells us a couple of things. One is that you need to realize that when you start messing with these pathways, start manipulating these pathways, you run the possibility of creating a new cell type that could be good or bad. In this case, it turns out to be good. And the other one, it suggests that the variegated, nuanced pathway of CD4 functional development that John O'Shea and the late Bill Paul have put forth, saying that binary thing is too simple, and that there are intermediates, I think has a lot of validity to it based on our results with CTLA-4 knockade. Anyway, so the last thing is just to talk of this part is to talk about memory cells. So this is a typical experimental setup here. We give, I think I missed the first. So we give, in this case, vaccine consisting of irradiated tumor cells to mice with the antibodies, and then wait 70 to 80 days, and then re-challenge them with tumor. And what you can see here is that when you re-challenge them, they got the vaccine plus CTLA-4, most of the mice are almost completely protected. Those that got PD-1 are very poorly protected. Most of the mice come down with tumors, so that memory is less effective. We can also do this with peptides, with a model peptide. In this case, ovalbumin, put it into immunize mice with that, and then come back and look at the number of cells that come up. And again, you can see you get many more cells coming back with that CTLA-4 than PD-1. And we looked at a number of parameters, but what we saw with TCF and TOX is very interesting. For those of you who follow the literature, it's becoming increasingly important that T cells, when they're activated, end up, some of them express TCF, some of them TOX. TOX tends to go with terminally differentiated effector cells, which are very good at killing things, but they don't last very long. And the other cells can develop TCF1, which is shown by Rafi Ahmed and John Wherry initially, and since been by others. These cells are the progenitors which give rise to a durable response. And so this just shows that if we look at these guys where we count the number of cells that we get out using this model peptide vaccine, you can see when we give anti-CTLA-4, there are many more TCF1 cells than you get with PD-1. And that button is too close for my big thumb. And conversely, there's more TOX cells with PD-1 than with CTLA-4. So we think that this is another thing. Perhaps the fact that it's working during initial priming, it just inherently sends cells down this memory pathway when you block CTLA-4 rather than that effector cell pathway. And that's why you end up with greater memory. And that may, although there's a lot of other factors obviously involved in the durability of a clinical response to a tumor, it suggests that the fundamental biology here may explain that difference. And so very quickly, and again, I added this because I want to say it's not all T cells. There are other cells involved, and we're coming to realize, due to some work that Dr. Sharma did in our immunotherapy platform, and others, of course, that myeloid cells are really real big players and when you look at these tumor responses, typically in a negative way. And so people have tried to deplete them or give things that block CSF1, for example. When you do those, it usually just really hurts the immune response too. So it's a double-edged sword, and we've done, I'm not going to talk about it, but we've got experience. We can show that if you can get either an improvement of a response or just completely destroy a response if you get rid of cells in the wrong way. Anyway, we got interested in LILR molecules. These are immunoglobulin-like receptors. There's two types. Those that are called B have TIMs in their ITIMs in their motif and tend to be inhibitory. The ones LILRBA have ITAMs and tend to be inhibitory, activating. We concentrated on LILRB4. Its ligands are thought to be integrins and ApoE. It's the equivalent, the GP49B in the mouse is the equivalent of LILRB4. We chose this because we saw in a number of database examinations that it was highly expressed in mice, which, for example, had chronic exposure to LCMV. Anyway, you can see that this molecule, we looked at a variety of mouse models and human cancers, and what we can find is the expression of LILRB4 in a vast majority of these in the hematopoietic cells, not in the tumor cells. So unlike PD-L1, we don't see this one on tumor cells. And so the first thing we did was single-cell RNA, and that tells us that when you look at expression of it on myeloid cells, it tends to be in those that have a suppressive phenotype rather than the ones that are activating. We knocked it out and gave them tumors, and you can see that tumors grow faster, suggesting that that has an inhibitory. In fact, this is two different mouse models. And when we treat them, it's got out of order. But anyway, when we treat the mice with antibodies to CTLA-4, this is an antibody treatment. What we can treat with an antibody, and we can also see improvement in survival by treatment with antibodies. And when we look at what happens, there are a number of clusters that are decreased in frequency after you treat it, and these tend to be the ones, again, that are suppressive here rather than the ones that are activating. So it tends to suppress the inhibitory ones and elevate the inflammatory myeloid cell population, which is the sort of thing you'd want to happen if you want to get a good response. This just shows in T cells it affects Tregs a little bit, increases effector cells and CD8 cells when you block these. So again, indirect effect of blocking it is increase in the T cells that you want shown there. And in any event, so we think this is one that should be added to the armamentarium with a few other prominent factors such as VISTA and LILRB2. Anyway, just to close, we've got all these things there that have been, have known and been in various levels of development. The only ones, though, which have received extensive positive clinical experience are CTLA-4, PD-1, and LAG-3 now so far. And I think a lot of that has to do with the fact that trials that have been done were not done in the right way. You'll hear about that from Dr. Schwam in a minute. But the point is we have a long way to go of doing combinations of these, combinations with conventional therapies as well as with targeted therapies. And I'd like to close just by sort of pointing out this. This is where cancer therapy was for a long time. You take a large number of patients, you treat them with the drug, you see if there's a statistical significant increase in median survival. It could be weeks to months. There's nothing wrong with that, as long as there's a quality of life. But what we know from ipilimumab, if this is about 20%, you can cure a fraction of patients. You can raise the survival tail, that is, unless biased, I guess, thing. You raise the survival tail. You can move it over 4 months, as Ipi did, but also have a tail of 10 years or more. With Ipi at about 20% of patients. And of course, what we need to do is not this so much anymore, but we need to concentrate on raising that survival tail as high as we can get it, and as many times of cancer as we can get it. And the good news is, 10 years ago we were saying, can we do this at all? And now we know, yes, we can. And so the challenge now is how do we do it? How do we get the rest of the tumors from 35, 40% up to 60? How do we get melanoma up to 100? And how do we get pancreatic cancer and glioblastoma off 0, basically? And so those are the challenges, and you guys that were in this course are the people who are going to meet that challenge and carry that on. So there's lots to do. but we know how to do it. So good luck with your careers, and I wish you the best. And thank you for giving me the time to talk.
Thanks so much, Jim, for this wonderful talk. We have time for 2 or 3 questions before the coffee breaks. So please raise your hands.
Yeah. One of the things that it's really cool is that the dramatic phenotype of CTLA-4 knockout mice can be rescued with CTLA-4 gene that lacks the intracellular domain, suggesting that the main mechanism of action is competing CD28 for the same receptors. So to what extent everything that you are seeing, which is very interesting in the phenotype of the CD4 cells, is the result only of CD28 overstimulation and controlled overstimulation?
What's the...
So the question is
it all just a question of overstimulation? I think so. Yes, I think that the appearance of those ICOS positive cells is exactly that. It's what we consider supernatural, overstimulation.
Yeah, now there are efforts trying to confine to tumors in a controlled fashion CD28 co-stimulation, aside from what you pioneered together with Lipinchen of expressing CD80 in the tumor cells, but using biospecifics, etc., to target co-stimulation, and maybe a good way to have it done in a different manner.
Well, I don't think that's going to get around the problem of CTLA-4 because it's still going to be there. What a number of people are doing now is to try to either put CTLA-4 in a cell, in the cell instead of B7. I would say put anti-CTLA-4 antibodies in it. They work better than this. There's also some efforts to actually make bites, with anti-CD3 or tumor antigen on one end, or with CD28 on the other to just deliver that more forcefully. That's still going to be inhibited by CTLA-4, but would be another way of delivering it. And doing that with anti-CTLA-4 would make that even better, I think.
I have a question for you. So, is the hypothesis of anti-CTLA-4 depleting Treg cells, is it still alive, or do you think that we can't stand on this anymore?
Is it still alive in humans?
In humans.
There are some people who think so. I've actually heard some people use one of Pam's papers and quote it incorrectly, where they looked at numbers versus, look at percent versus absolute numbers. But in any event, I don't, at least the data that we have, Pam and Tony Rivas says there's none. Alan Corman, who worked with me on developing IPI and then went to BMS and worked on that a lot. He's the one that showed that happens, that it does happen with the mouse. And when he put the FC that's on ipilimumab on our antibody, it no longer worked very well. And so it's very much alive, but I think what we're learning is they're trying to, there's an antibody in development, late in clinical development now, about to go into patients after safety trials, that has defucosylated so that it binds to FC receptors better. Mm-hmm. That's one attempt. There are several other attempts to do it out there, including engineering it in other ways to kill Tregs. Or there's another group still trying to develop variations of IL-2 that would be non-alpha IL-2 binders that would work on effector cells but not on Tregs, and could possibly be used to appropriately modified to kill Tregs. So that's a big open space now, I think, in the field is some other way of getting rid of Tregs in humans.
Great.
I mean, just to directly answer the question, ipilimumab does not deplete Tregs in humans.
Another question? We have time for one additional question.
Hi, sir. Nice to meet you. It's not a question, but if you were to give an advice for us, what that would be? An advice. Sorry.
Oh, I would say, as general advice, I would say, my advice in a very general way would be to ask yourself, why you're doing science. And a lot of people come into my lab and say, well, I want to cure cancer, but very rapidly they find out that, you don't just do that because you want to. It's hard work. It's very frustrating. And you have to love science to do it. I think that science chooses you. You don't necessarily choose science. And if you really love doing experiments, however they come out, if you take pleasure in designing an experiment and getting the proper controls done and making it, making something, learning something, whatever it is, that was a bad idea, that was a good idea, doesn't matter. If you love putting a good experiment together and take enjoyment from it, then you should be in the field and love it and work hard and do it. If you find that frustrating, everybody finds it frustrating at times. It can go years, I know. You can be wrong a lot, I like to say one of the differences between a clinician and a scientist is that a clinician needs to be right, 100% of the time, because they got to have all this stuff in their heads, and patient presents, they got to be right, or the worst thing happens, they don't help a patient. But a scientist, we're supposed to be wrong a lot. But definitively wrong, not kind of I think I'm wrong, I think I'm right. Definitively wrong or definitively right. And then, and if you can put up with that about going years without doing anything, that's really going to, you want to tell your grandmother, this is what I do, and it's important, you slog it along, but you enjoy it, I used to ride a motorcycle. I enjoyed working on it as much as I did riding it, So if you have that sort of feeling, I think that, you can stick with it. And if you get that kind of joy, just stick with it. Good things will happen for you down the road.
I think we have another question. Tomás, yes.
Yes, thank you very much for your talk. I cannot believe I'm asking a question to a Nobel laureate. So my question is aimed at the mechanism of action of these ICOS positive Th1 type cells. So do you know if the CD4-CD8 collaboration is different, or perhaps these cells have like a cytotoxic action, like it was shown in bladder cancer, like a granzyme positive CD4 cells?
Yes, I can't give you hard data for whether there's help, but I strongly suspect that there is. We've got some data from mice that suggest that they can provide help to CD8s. But we've also shown that they can, under some circumstances, they're pretty contrived in the mouse, but we can create a circumstance where there's no CD8 T help and kill on their own.
Okay, before going to our coffee breaks, I would like to take the opportunity to thank a very special person who helped a lot to help us to have the possibility to have Jim and Pam here is Laura Bober. Thank you very much, Laura. So we now have 15 minutes, coffee break. Please make sure you come back. And now we will have tenemos un anuncio para el curso. Vamos a hacer una foto con todos los estudiantes del curso aquí afuera.
remain host. No, que siga siendo host. ¿No? No. Diego. No, ellos son. Diego, ¿puede ser? Bueno, ya creo que es host. Ahí está Lori. Ah, y a ver qué me dijo. A ver, para. Chat. A ver si me contestó. Ay, no veo el chat.
¿No te lo habían pedido?
Ay.
Hay que ir a un Switch, por favor, hay que ver si hay lugar en el Switch. Es complicado.
Ya.
Sí, ya acá no existe el chat. Yo de hecho estaba haciendo otra cosa para allá y vine hasta acá. Yo no sé qué.
No como personal del sistema del ministerio, ¿no?
Ese cable anda azul. Hay un cable azul que no andaba en el.
Pero no es ese. No, no es ese.
No estaba con eso. El teléfono, ¿por qué no pasa el teléfono? El teléfono está, tiene una salida de.
Hello.
Hi.
Tiene que tener una salida acá arriba.
Is this the right time?
Hi, Lori, can you listen to me?
Yes, but I now have to go back to where I was.
Yes, I know. Sorry. The honoris causa act.
I misunderstood the timing that we got a while ago. So I'm going to try to come back here in 45 minutes.
Okay. If you can,
so that-
it is really good. We will really appreciate that. But if you can't, we understand. You'll be waiting for too long. We know that.
So have we started the recorded?
We are just starting in 2 minutes, the recording talk. So we have 45, 46 minutes.
Okay, I will try to come back. I can come back for a few minutes.
Okay. That's sorry for the delay. Please apologize us. Best.
No problem. Thank you.
Thank you.
Hello. We are running out of time, so we will be back with our next speaker. Our next speaker, also Doctor Honoris Causa of the University of Buenos Aires, will be speaking alive. We're speaking is Lori Glimcher. She will speak about identification of RIOC2 as a master regulator of human blood cell development. So, is it ready? They told me it's a pre-recorded because she was waiting, but it's a pre-recorded session.
Good afternoon. It's a pleasure to be with you, and I want to thank Gabi Rabinovich and the University of Argentina for the honor of receiving an honorary degree from this wonderful, first-class, superb university. I am just so sorry I can't be with you, but this is truly a huge honor. So today, I want to talk about two stories that are related to each other, and both of them originate from a little studied atypical kinase called RIOC2. In the first story, I'm going to explain how this kinase, which is known to assemble the pre-40S ribosome, led us to a new treatment for anemias, which was IL-22 blockade, including in myelodysplastic syndromes. And in the second story, we discovered that REOC2 acts not only as a kinase, but also turns out to be a master regulator transcription factor, raising the possibility that small molecules, coactivators, or staple peptides might stabilize this protein and hence could be useful in the treatment of anemia in many diseases. So as we all know, anemia and blood disorders are very, very common worldwide. And in fact, according to WHO, the prevalence of anemia is about 23%, and that actually is increased in geriatric populations, the rise in chronic diseases, cancer, inflammatory diseases, poor nutritional habits, and it's exacerbated in developing countries. So what do we have now that is going to be, how do we treat now is what I'm trying to say, the anemias of cancer and inflammation, because our therapeutics are small in number. Obviously, transfusion is one possibility. And we've known and used for years erythropoietin to stimulate red blood cell development, and more recently, luspatercept, which is which targets the human activin receptor type 2B, and through TGF-beta superfamily ligands, it can reduce SMAD signaling, which results in increased erythropoiesis. Most recently, a novel hypoxia-inducible factor, proly hydroxylase inhibitors, have been developed in more than one company, and one of those has been approved now by the FDA. And that's very exciting and is effective in increasing erythropoiesis. But the truth is that despite these possible treatments, patients become resistant to erythropoietin. The current treatments are only efficacious now in about half of myelodysplastic syndrome patients, and so resistance emerges. So we need to continue to figure out new therapeutics to reverse anemia, whether it is in cancer or in other settings, as you'll see as we continue this seminar. So, for example, if you look at myelofibrosis, which is another form of cancer, the biggest issue in myelofibrosis is anemia. And myelofibrosis is really transfusion dependent at this point. And if you look at this graph, patients who are transfusion independent have decreased mortality as compared to patients who are transfusion independent. And the treatments that they get actually increase cytopenias, and that's including thrombocytopenia as well. So we need to continue to look for other avenues. And so what I want to talk about is the work of Mahesh Roundal, a former postdoctoral fellow in the lab, who has now moved on, and his discovery that there is a cytokine that, when inhibited, can reverse erythroid dysfunction in stress-induced anemia. So what are we talking about? We're talking about this kinase called REOC2. And REOC2, its official name is right open reading frame kinase 2. We actually discovered REOC2 REOC2 because there are only 2 or 3 publications of REOC2, and that was work done in yeast. So it's a very little studied atypical serine threonine protein kinase. And it functions, it's known to be functional and actually indispensable as a component of the pre-40S ribosome subunit. And we picked it up when we were looking for interesting proteins downstream of the ER stress response, which we've worked on for many years. And so we did transcriptomics to identify genes that were either increased or decreased in the absence of the IRE1 XBP1 stress pathway. And we identified REOC2 as decreased levels downstream of XBP1 deletion. And we thought we would look into that because it was such a little studied kinase at the time. Interestingly enough, when you look at patients who have myelodysplastic syndromes, and you look at in the most common mutation in MDS patients sits on human chromosome 5, and the arrows here show you the deleted part. Interestingly enough, REOC2 is just within what we call the commonly deleted region that about 15% of MDS patients have. So that was interesting to us. And you can see here where that is situated. So let's just do a brief discussion of what is myelodysplastic syndrome. MDS are groups of cancers that are characterized by failure of blood cells in the bone marrow to mature. They occur in about 7 out of 100,000 people, usually around age 70. And while a sizable percentage of MDS cases do progress to acute myelogenous leukemia, most of the morbidity and mortality associated with MDS results not from transformation to AML, but rather from hematologic cytopenias. 2 to 3 year average survival time following diagnosis of MDS is owing to the fact that there are very few treatment options, and hence, particularly in elderly people, there are substantial risks of allogeneic bone marrow transplants. And hence, we really have a dearth of effective FDA-approved drugs. Makes it imperative to revisit the origins of blood cell differentiation defects, as we showed earlier, that underlie anemia and myeloproliferation to be able to identify new druggable targets. So anemia is the most common hematologic manifestation of MDS, particularly in the subset of patients with del(5q) MDS, which I showed you on the previous slide. It is the most commonly detected chromosomal abnormality in MDS, as I said, in 10 to 15% of patients. So let's talk a little bit about what REOC2 does in MDS. We wanted to explore it, so we knocked it out, and knocking out REOC2 leads to embryonic lethal homozygote germline. So we then turned to VAV1-CREs, and homozygous VAV1-CRE REOC2 also is embryonic lethal, but luckily haploinsufficiency of REOC2 in hematopoiesis is viable, yields viable animals. So similar to other ribosomal protein haplo mouse models. So when we looked at these animals, you can see that they had anemia. They had reduced peripheral blood, red blood cells, hemoglobin, and hematocrit, and that's what we're going to look at in the following slides. And we discovered that REOC2 haplo-mediated anemia was secondary to a defect in erythroid development in the bone marrow, which is a major site of erythropoiesis, as you know. We characterized the stages of R1, R2, R3, R4 of erythropoiesis using TUR119 and CD71, and identified that it occurs early in erythropoiesis, as you can see here. Now, it's known that ribosomal protein deficiency mediated reduced protein synthesis predominantly affects erythropoiesis over myelopoiesis. And we also discovered that these haploinsufficient animals showed a decrease in cell quiescence with a cell cycle block at the G1 phase. I'm not showing you that, but that is indeed what we found. So cell cycles are altered as well. The expression of P21 was increased in erythroid precursors from haploinsufficient VAV1-CRE. Go to the next slide here. Okay, so we then asked, what is the effect of REOC2 haploinsufficiency on stress-induced erythropoiesis? We were using young mice who had been given non-lethal phenylhydrazine treatment that produces acute hemolytic stress. So REOC2 haplosufficient mice, VAV1-CRE mice, developed more severe anemia and had a delayed recovery of red blood cells, as you can see here, compared to the control animals. And they succumbed faster to a lethal dose of phenylhydrazine as well. The anemia in these young mice was preceded by a reduction in bone marrow erythroid precursor frequency, which indicates an erythroid differentiation defect, which is consistent with the role for REOC2 in driving erythroid differentiation. There were fewer CFU-E colonies observed in erythropoietin-containing methocult cultures. And I'm not showing you all this data. We don't have time for that. But as you know, in myelofibrosis, you not only get anemia, you also get myeloid proliferation that leads to, in about 20% of cases, to acute myelogenous leukemia. And so we looked at myeloproliferation and found out, like MDS patients who have both anemia and monocytosis, we saw this in this animal model as well. Haploinsufficiency of REOC2 increased the number of monocytes, decreased the percentage of neutrophils, and increased the numbers of granulocyte macrophage progenitors in the bone marrow as well. So that is typical of what we see in humans with MDS. And I'm going to tell you later about recent studies by another postdoctoral fellow, Shrestha Ghosh, who revealed an additional and unexpected mechanism by which REOC2 regulates GMPs and megakaryocytes. Next slide here. So why is this, why does REOC2 deficiency, haploinsufficiency, lead to erythroid differentiation defect? And so we did a volcano experiment, as you can see here. And what we found was that REOC2 haploinsufficiency led the upregulation of about over 550 distinct proteins in erythroid precursors. Notably, it also resulted in downregulation of other ribosomal proteins, the loss of some of which, like RPS5 and RPL11, has been implicated in driving anemias. But what we saw here was the alarmins, including S100A8, A9, CAMP, NGP, and others, were the most highly upregulated proteins in the data set. Elevated alarmin concentrations were previously reported in bone marrow of patients with MDS. The upregulated proteins with the highest fold change, which I've mentioned now, are proteins with known immune functions, such as antimicrobial defense. We did GSEA analysis, and that again indicated possible role for the immune system in driving the changes that we're seeing in these REOC2 haploerythroid precursors. An independent analysis of the REOC2 proteomics dataset using the Metacore pathway showed immune response as the top differentially regulated pathway in these animals. So we decided to look at T cells first. We polarized naive T cells from haplomice and looked at the presence and function of CD4 helper T cell lineages first. We looked at Th1, Th2, Th17, Th22, and also Treg cells. And we saw that the secretion of multiple cytokines was completely normal, but we observed an exclusive increase in IL-22 secretion from naive T cells that were polarized towards the Th22 cell lineage. The frequency of these IL-22 positive, CD4 positive T cells was also higher, and the concentration of IL-22 in the serum and the bone marrow fluid of older REOC2 haplomice was also significantly increased. So we used, using known IL-22 target genes, that we curated an IL-22 signature gene set that showed significant enrichment in the REOC2 haploproteomics data set using GCEA. And I think I've got that on, is not actually shown in this slide. So suggesting that IL-22 induced inflammation somehow is the contributing factor that is induced by REOC2 haplo loss and makes erythropoiesis inhibited. Now, the other thing that it does that we saw was an upregulation of P53 in REOC2 haplo-insufficient T cells, and you can see that here. So we're getting an increase in P53, which drives IL-22 production in T cells, and it's very selective in Th22 cells. So what's going on here? You'll see in the next slide, the alarmins that are produced from these stressed REOC2 haploinsufficient red blood cells are increased in erythropoiesis and also increased IL-22 in Th22 cells. And you can see here that reduced REOC2 and increased alarmins are expressed in patients with MDS. And I'm showing you that here. So IL-22 here is increased, especially in del(5q) MDS patients. So how does IL-22 and its receptor do this? And I think we all know what IL-22 receptor is. It's produced by CD4 T helper 22 cells, also by ILC3s, and it binds to a dimeric receptor, IL-22 receptor alpha and IL-10 receptor 2 signaling, and it does that via the TYK2, JAK1, STAT3 pathway. And I'm not going to go through all the other information about IL-22 and its receptor, but it's known to activate many, many processes that involve antimicrobial immunity, inflammation, tissue repair, and so forth. So we started looking at the function of recombinant IL-22. And when we took wild-type mice or that treated with phenylhydrazine or REOC2 deficient mice stressed by phenylhydrazine administration, and we treated them IP with recombinant IL-22, it led to decreased numbers of red blood cells, hemoglobin, and hematocrit. And that was because of decreased bone marrow erythroid precursor cell frequency and increased apoptosis of erythroid precursors. So this treatment also led to an increase in peripheral blood reticulocytes, which indicates that there's trying to increase erythropoiesis under stress. Recombinant IL-2 also dose-dependently decreased terminal erythropoiesis in vitro assays. Notably, IL-22 mediated inhibition of in vitro erythropoiesis led to the induction of P53, suggesting a feedback loop between IL-22 and P53 in driving dyserythropoiesis. So exogenous recombinant IL-22 exacerbates stress-induced anemia in wild-type mice and also in REOC2 deficient animals. So what's the molecular mechanism here by which this occurs? And it turns out, to our surprise, that IL-22 receptor, which is thought to be expressed only on epithelial cells of non-hematopoietic origin, for example, epithelial cells, mesenchymal cells, but we discovered that erythroid precursors in the bone marrow also express IL-22 receptor. And it's detected exclusively in erythroid progenitors among all the lineage negative cells in the bone marrow. So that is probably the mechanism through which IL-22 is working to induce anemia at early stages. If we make a genetic deletion of IL-22 receptor using an erythroid-specific CRE, we again show this, we now show an increase in erythroid cells, RBCs, hematocrit, and hemoglobin. So if you delete the receptor in erythroid progenitors, you now get an increase in erythropoiesis. So we then looked at compound deletion of IL-22 in the REOC2 haplo mice. And indeed, if you reduce IL-22 even by 50% haploinsufficiency, you get increases and restoration in peripheral blood and also in the bone marrow of red blood cells. So the next step was to actually ask whether an antibody that would neutralize IL-22, what effect would that have on anemia? And when we treated animals with an antibody that neutralizes IL-22, again, we're seeing here, both in wild-type mice and in REOC2 haplo mice, an increase in red blood cell development, again, looking at peripheral blood and in bone marrow, which I'm not going to show you. So clearly, in genetically wild-type mice, it seems as if the levels of IL-22 probably actually monitor whether an animal is becoming somewhat anemic and you then get because you have an increase in IL-22. So we wanted to look at more than just myelodysplastic syndromes. We've looked at this now in a couple of different models of MDS, but we thought, what about the anemia of inflammation, which is also very present? And so we looked at the APC-Min model, which is a model of inflammatory intestinal neoplasia leads to colorectal cancer. And they develop anemia early on at about 9 weeks of age. And again, if we inhibit IL-22 with an antibody, we can reverse the anemia and the red blood cell number, the hemoglobin, and hematocrit in this different model. And so now it's time to also look at a clinical grade human antibody, because these are mouse antibodies. And we were fortunate to be able to obtain a clinical grade human antibody that also works in mice, and it cross-reacts in mice. And we see the same thing. If you inhibit with this particular monoclonal antibody, again, it really improves red blood cell development, hemoglobin levels, and hematocrit. And this is actually in wild-type mice, and also in the presence of stress-induced anemia. So now we want to turn to MDS patients themselves, because what I've shown you mostly here is mouse models. And so we looked at the bone marrow fluid in MDS patients. And what you can see here is that IL-22 is elevated, particularly in the del(5q) MDS patients. If you look at levels of ReOcT2 mRNA, you can see that there is an inverse relationship with ReOcT2 mRNA levels and levels of IL-22. The lower the level of ReOcT2 mRNA results in both bone marrow fluid and, as you can see here. Next slide. Okay. So that's not just true of patients with MDS, but if you look at other diseases where patients experience anemia, chronic kidney disease is absolutely one of them. It's frequently observed in patients with CKD, and it's associated with very poor outcomes. Well, the anemia of CKD is resistant to erythropoiesis stimulating agents in quite a substantial percentage of patients, suggesting that, a lot of things are happening, a lot of pathogenic mechanisms other than erythropoietin deficiency. So we looked, with the help of Joe Bonventuri, we looked at levels of IL-22 in patients with CKD versus healthy patients. And in particular, we separated out CKD people who have anemia and those who do not have anemia, have chronic kidney disease, but have a normal hemoglobin. And you can see that this middle draft, you can see an increase in IL-22 in those CKD patients who are anemic. And again, if you look at hemoglobin and its correlation with IL-22, again, you see correlation, very strong correlation. We were then fortunate enough to get some data from a phase 2 clinical trial of IL-22 inhibition. This was not in the setting of MDS or anemia. It was a phase 2 clinical trial that involved an inflammatory disease And the effect of IL-22 inhibition was fairly modest, but in terms of the inflammation of that disease. So we wanted to know whether IL-22 inhibition in humans with a human antibody also happened to also increase hemoglobin, which hadn't been studied, but thanks to the cooperation of this company, we were able to look at those data. And what you can see here is that IL-22 inhibition in human patients increases hemoglobin. So that's very encouraging as we think of a new therapeutic to increase red blood cell formation in patients who have multiple diseases that lead to anemia. So in summary, and I'll let you look at this yourselves, I think the bottom line is that we need REOC2, which we thought at this point was an atypical kinase, because in its loss, even 50% loss, this generates anemia for a number of reasons. When we think about phase 1 and 2 clinical trials in stress-induced anemias, we can select those patients who have high levels of IL-22 to test out whether we will get significant increase using either IL-22 monoclonal antibodies as a therapeutic in those individuals. And this is a schematic of that. Basically, to summarize, secretion of alarmins from red blood cells activate Th22 cells. REOC2 haploinsufficiency induces p53 upregulation, which leads to increased apoptosis of erythroblasts. And that's by one mechanism by which REOC2 controls IL-22 production at the molecular level. So we're excited about this and hoping we can move into humans. Now I have a few minutes left, and I want to talk a little bit about the second story. And that is, we were a little bit puzzled as to how REOC2 as a kinase was affecting all of these changes. And another postdoctoral fellow in the lab, Shrestha Ghosh, asked the question, does REOC2 control erythropoiesis and myelopoiesis and thrombopoiesis only because of its function as a kinase? And so she postulated that maybe, here she is, whether REOC2 has another function entirely. And what she has discovered is that REOC2 is actually a transcription factor that sits at the apex of lineage commitment in hematopoiesis. She looked at REOC2 and realized that REOC2 had a winged helix turn helix motif, and that's very common in transcription factors. They have the Reo kinase domain, and they have, and she identified the predicted nucleic acid binding motif, and this is what it looks like. She used, as you can see here, that human REOC2 does activate human erythropoiesis, and these are peripheral blood from stem cells from human blood samples. And I think you can see here, if you knock down REOC2 or knock it out in stem cells, what you see is a reduction in erythropoiesis and an increase in myelopoiesis and megakaryocyte thrombopoiesis. If you do that in humans. So it activates human erythropoiesis, but it represses myelopoiesis and megakaryopoiesis. And here's what REOC2 looks like. It has a winged helix turn helix motif. And I'm going to move ahead fairly quickly, but here's where we've identified the DNA binding motif. This is the kinase domain, and this is the less conserved C terminal, and this is DNA, and this shows how REOC2 binds to DNA. We did a series when we did this using a number of mutations that Shrestha did. She looked at the helix turn helix domain. She predicted 7 evolutionarily conserved residues, and she mutated those, and that's how she identified the DNA binding. She also identified an overall loss of REOC2 actually reduces chromatin accessibility at the promoter's intronic and intergenic regions of many different genes. So it's a global change in chromatin accessibility that's consistent with REOC2 being a transcription factor. She then wanted to see how REOC2 regulated the expression of several important hematopoietic lineage-specific transcription factors. And I say that we think it was positioned at the apex of that. And indeed, when she did transcriptomics, she showed that, and here she's using human hematic stem cells, progenitor cells, and knocking down or knocking out REOC2. So we have a dose response here. And she discovered that there was a block in lineage commitment and a concomitant elevation in myelopoiesis and megakaryopoiesis signatures with dose-dependent loss of REOC2. And remarkably, she saw that loss of REOC2 altered the expression profiles of the key transcriptional factors that determine hematopoietic lineage commitment. For example, GATA1 and KLF1 were downregulated. They are critical transcription factors for erythropoiesis, while upregulated GATA2, SPI1, and RUNX3 were upregulated, amongst others. So there's a correlation between REOC2 levels and levels of lineage-specific transcription factors. And I'm going to go quickly through this because I know I'm almost in time. We'll just show that REOC2 binds to the DNA motif that she identified and to the 5' UTR and to its target genes, GATA1 and RUNX3, and she's looked at all of the other ones as well. So schematically, if you take human stem progenitor cells and you reduce or knock out REOC2, this is what happens. You get a decrease in these factors that govern erythropoiesis, and decreased erythropoiesin is the outcome. And in megakaryocytes, decreased RUNX2 decrease leads to increased megakaryocytes and increased, and this is reminiscent of patients with MDS. She showed that this was the de novo nucleotide sequence in the promoters of the targets. We don't have to stress on that. And that the N-terminal helix turn helix domain has a DNA binding domain. We did structural experiments with Dr. Greg Petsko, and we're able, after doing a lot of mutations, we were able to show where the transactivation and transrepressor domains sit. And that's, you can see that here's the Rio domain where the kinase bends. this is the two transactivation domains. Let me go back a second. These are the two transactivation domains in yellow. In green, you'll see what we think is the trans repressor subdomain that leads to increases in myelopoiesis. Here, of course, is the DNA, and the purple, these two are the transactivation domains. So we've dissected it. And then we asked, what about MDS patients? Again, we need to bring this back always to the patient. And so Schrechter looked at the mRNA levels of REOC2 to see if they correlated with these transcriptional targets that we'd shown in MDS patients. And again, you have a very strong correlation, positive correlation of REOC2 mRNA and GATA1 and KLF1, and just the opposite in the repressed genes, which are GATA2 and RUNX3. And this is what's happening in patients with MDS. We also looked at the mRNA levels of REOC2 and whether they correlated in MDS. And again, here we go, beta hemoglobin, alpha globin, and are increased and correlated with levels of REOC2, while two other genes that are negatively correlated with myeloid genes, such as CD11B and ITGB3, you see a negative correlation, as one would expect. This is true in AML patients as well. You see an increase in GATA1, KLF1 with REOC2, and a decrease, a negative correlation with myelopoiesis. So, okay. Sorry, I forgot to advance that. And finally, coming back to the huge number of patients who have anemia in chronic kidney disease, we were able, again, to look at the mRNA expression of REOC2 in chronic kidney disease patients as compared to healthy controls. And again, you can see here, if you look at the mRNA levels of REOC2, they are reduced in anemic patients with chronic kidney disease compared to healthy controls. And that's also positively correlated with the expression of other early erythroid genes like transferrin receptor, hemoglobin beta, and hemoglobin alpha subunits. So to summarize, we believe that these data establish REOC2 as a master regulator of hematopoietic differentiation and human blood cell development. And this is positioned very early in lineage commitment in hematopoiesis. And I think this shows that quite well. If you look at REOC2, I'm showing here, it's the domains of REOC2. It drives GATA1 and KLP1. You get normal hepatopoiesis and you get normal myelopoiesis. But if you delete or even reduce by 50% REOC2, you see a loss of red blood cells and a corresponding increase in myelopoietic cells. So for the last here, just showing you a nice picture of this structure, again, thanks to Dr. Petsko, where we see how REOC2 acts as a transcription factor. And you can see where it binds DNA and the transactivation domains and the transrepressor domains. So let me end by thanking especially two of the postdoctoral fellows in my laboratory who were responsible for these two stories, Mahesh Roundal and Shrestha Ghosh. We've had many other individuals who have helped us with various things, but I want to also point out Dr. Greg Petsko, who did the structural work for us, and Joe Bonventry. So thank you very much. And again, I wish I could have been there in person, and I hope that it's a wonderful, wonderful event. So thank you again. Hi, everybody. I get to apologize again for not being able to be there with all of you, but I'm happy to answer questions. So I gather there aren't any questions. No, go ahead.
Great. So again, congratulations, Laurie, for the Doctor Honoris Causa. It's great to see you now in person, and we have time for 2 questions. Yes.
Hello, Laurie. To drag this pathway, the mutation analysis that you perform rules out that the kinase activity is involved in the function. So once the REACT2 binds DNA, is the kinase activity dispensable?
I did not hear all of that. Maybe.
No, let me try to do it louder. Is the kinase domain required for the activity according to your mutation analysis or not? And if so,
I'm sorry, this looks like this might not work. Because I still can't understand the question. It had to do with the DNA binding domain, which is a helix, wing helix.
Alguien, can you hear us now or not? I think she can hear us.
I can hear you a little bit.
A little bit.
Can you hear me?
Okay. You want to repeat? But
So, Laurie, the question was about the kinase domain. Is it required for activity? And if you were going to drag this factor, how would you do it?
Well, I can't hear the question, but let me just say we've been thinking about ways to translate this to individuals who have anemia, whether it is anemia secondary to cancer or to chronic kidney disease. And we have an opening in terms of reversing. I'm sorry?
No problem, no problem. Yes, go ahead.
So let me just thank you again, and I wish I could have been there, but one of my very closest friends is having a symposium to celebrate his 70th birthday. So
thanks so much, Laurie, and I'm sure next time you will be here. And
yes,
we will bring the prize and the diploma to you to Boston.
Thank you.
Thanks so much. Thanks so much. Thanks so much for your time. Okay, so we will move to our next speaker, and our next speaker is Argentinian, but will speak in English. Yes, and he is Sebastián Amigorena from Institut Curie in Paris, France, who is now the Doctor Honoris Causa from the University of Buenos Aires.
So just one word in Spanish before I start my talk. Es difícil explicar qué se siente, ¿no? Después de en este tipo de situaciones. Primero porque la uva para mí fue como una especie de imagen extraña y misteriosa durante mucho tiempo cuando era chico, que mis padres eran profesores y se fueron. No se vayan a imaginar que era ningún héroe de la militancia política. Yo era más o menos así, mucho más cortito que los palos de esa noche. Pero bueno, ¿viste? Se vive en la imaginación de los niños cosas que después duran y no cambian. Y bueno, es importante para mí este título y quería realmente agradecerles a Gaby y a Jorge de haberme incluido en esta lista. Me siento un poco extraño estar con este grupo de gente y de inmunólogos que son todos mis ídolos desde siempre y me da mucha alegría y mucha proud. Muchas gracias a todos. Ahora, okay, so that was just a thank for Argentinian University. I'm going to be discussing something very unusual for me. So I don't really know how science took us to work on CAR T cells, but I will try to explain that to you, starting from something that was totally unrelated. So the start of this story is something around a little bit less than 15 years ago, when my lab moved from a cell biology building when I have been working for 10 or 15 years, to a building where there was a lab working on epigenetics. So the understanding of epigenetics by this colleague is Geneviève Almousny, who became a friend. She always says epigenetics, so it's a very bizarre word because it's very broad and you can call anything epigenetics. She uses it for chromatin dynamics, which I think is a nice way to think of it. If you want to regulate cell functions, and you need proteins in the cell, transcription factors and repressors and modulators to get access to specific genes, and that requires chromatin to open and to close at specific locations. So let's understand epigenetics as chromatin dynamics in this context. I am the one who should change the slides, I guess. These are some of the disclosures. Immunotherapeutics is a company that we just founded to try to bring this to the clinics, and I can tell you more about that a bit later. So this is the pathway that she was working on and she participated to describe, and that we started with for all this. Is a pathway of this guy here, H3K9 trimethylated. So this is an epigenetic mark. It's a trimethylation of the tail of histone 3 on position 9 for this lysine 9. And when this gene gets trimethylated, that promotes the binding of a complex of proteins that are actually at the same time writers of epigenetic codes and readers that close chromatin. So when you put H3K9 trimethylated on the chromatin, on these histones, chromatin closes, and that represses gene expressions. The proteins involved are some of these. So ZDB1 is the one who puts the first 1 and 2 methyl groups, and then SUB39H1 is the one that puts the third group, which is really the one that locks the binding of these guys here, HP1, heterochromatin protein 1, and there are 3 isoforms, and they bind to this histone tail here and promote closing of the chromatin. The alternative state is this acetylated form of this same position, and that is associated with chromatin relaxation and opening and gene expression. And actually, what happens when you knock out SUB39H1, and that is what I will tell you a lot about, is that the chromatin opens at certain sites. We don't really know the link between the specificity of certain genes that get opened and the actual complex that binds to the chromatin. That is still unclear, but it has broad effects in the whole genome at different specific places. And so it started. Actually, Geneviève was telling me what she was doing at lunch, and I was impressed by the fact that it was these cells, so they are all work on MEFs and fibroblasts, which are big flat cells that sit there and divide from time to time when they want. And they're super boring, I thought. I didn't tell her that. But it was like not a lot of dynamic biology. And I told her, we have these cells that are great. You activate them, you can synchronize them, they start proliferating, differentiating different types. And so we can look at choices and fates and lineages in a very accurate way with relevant models in vivo to do that. And the first model that we looked at that time, so this was published in 2012, was the differentiation of naive cells into Th1 and Th2. And what we saw was pretty interesting. So, so it's a SUV39H1 knockout. So you relax chromatin, what happens? What happened is that differentiation of Th1 and Th2 was just normal. It was no big phenotype. But if you then take Th1, differentiate the cells, and we did this in vitro or in vivo, and you put them in the opposing condition, so you take a Th1 and put it under Th2 conditions, or a Th2 and put it on Th1, you see that in the case of Th2 cells, they become more plastic. So you can repolarize them to Th1, which you cannot do in the wild type. So the conclusion was that Th1 lineage expression, so the genes Tibet, interferon gamma, which are Th1 genes, are suppressed by this type of mechanism in Th2. And we don't have this system of heterochromatin closing. Th2s remain more plastic. They don't commit as strongly. And then we did another work. A second postdoc came to the lab, and she started looking at CD8 positive T cell memory and trying to understand if this closing of chromatin was going to affect the choices between effector and memory. And so let me tell you a little bit more about that. So we used a model that is infection by Listeria monocytogenesis, which is a very strong infection. You have a very strong T cell response, CD8, the epitopes are known. Actually, we followed the effector and memory responses. And this was the conclusion from that study. So basically what we found, we actually used single cell transcriptomics for the first time. This was the very beginning in the 2016 or so. And so that allowed us to analyze in detail the choices. So this is a model that we proposed based on this paper. Many people would not agree, but just let me tell you what was our conclusion. So when cells go from naive to activation, they start cycling. And what we saw in the single cell analysis is that when T cells, CD8 positive T cells start cycling, they turn on both effector and stem memory programs. And the cells individually have up these two programs. And then what happens is that during this proliferation, you can make a choice between going to a memory state or an effector state. And what happens is that when you go to effectors, you change the phenotype, so you lose all the stem memory programs. The cells become short-lived, they don't persist, but they get the effector programs on, and they can secrete high amounts of cytokines and become cytotoxic. And when they are memory, actually, they keep the programs on, and they stay plastic, and they go to this proliferation back again if they meet antigen again and can go to effector path again. So this was the model we proposed. It has some advantages and some disadvantages, and I'm not sure it is correct. But what this paper really did show is that when cells go from, during proliferation, from an early stage to the effector phase, they use silencing through SUV39H1 to silence their memory and stem programs. And so in the knockout for this protein here, the genes associated with persistence and memory remained high, and the cells persisted longer, and the effectors become plastic. Normally, effectors cannot go back to these memory-like cells. They are going to die. And the cells that lacked SUV39H1 remained plastic, and you could take effectors and then put them in another mouse, and they will regenerate memory cells, which doesn't happen when you do this in the wild type. And so, and then we were following, we were not working a lot on tumor, but following all the great works from the people that are here today, Jim and many others. And we thought, okay, memory seemed to be positive in anti-tumor immune responses, so this seemed to be beneficial. And so we tried to see what happened when we knocked out SUV39H1 in T cells in tumor models. This is maybe not very useful. It's a complicated slide, but it tries to explain what Jim explained in much simpler and more clear ways. It's a model for what happens under chronic stimulation in either viral chronic infections or tumors. When the cells go into this exhaustion pathway, it's still, I think, very not well understood, and don't think that all this is just true because it's written down there. It's just a way to understand this pathway. So the cells early on become what people call progenitors, so exhausted progenitors that still are able to self-renewal. They have low or intermediate levels of PD-1 and other checkpoint inhibitors. They express TCF1 and TBET, and they are low for another set of markers. And as chronicity establishes, under the influence in part of these negative regulators, the cells become what many people call terminally differentiated exhausted T cells. And one of the things that is proposed by people working on these is that the cells progressively become resistant to checkpoint inhibition, and you cannot drive them back to this self-renewing state. One of the things that happens here is that there is an epigenetic barrier where cells go from a progenitor to a terminal state and then cannot come back and be reprogrammed with anti-PD-1 blockade. So that is a little bit the overall picture, and the relation between the normal differentiation pathway from naive to effectors and this chronic stimulation pathway is not very clear, but it seems that it can go from different stages into this exhaustion pathway. Now, one of the things you have to keep in mind is that in patients and in mice, there is a correlation between these things here that we call exhausted and effective response to tumors, which is not very easy to understand and keeps puzzling me. So I don't think that we know that this phenotype is associated with something bad in all models, so it's a complicated one. But let me tell you what happens in these SUV39H1 knockout mice, and we did this in both complete knockouts and now we have conditional flox CD4 Cree mice. The phenotype at the steady state is mild, so that is why people did not pay attention to this molecule for a long time. But basically what you see when you measure CD4 and CD8, so in CD4s, these are effector memory and central memory, there is not a lot of phenotype. And in CD8s, and these are percentages, a log scale here, what you see is an increase in central memory cells. So these are CD62L, CD44 high cells. The numbers are increased and the proportions are increased in central memory cells. And this is true in spleen and lymph nodes. Actually, there is an interesting phenotype for tissue resident cells that are also increased in some of these tissues, in some of these mice. So this is just the first experiment we did. We took B16, a lowly immunogenic model, expressing of albumin that makes it a bit more immunogenic. We put the tumor in either littermates or treated or not with anti-PD-1, or knockouts treated or not with anti-PD-1. And this is what happens. This mouse grows very aggressively. This is 20 days. And when you treat with anti-PD-1, you have a partial delay in the response. It's a model that is not very strongly responding. And in other tumors, you completely cure the tumor with anti-PD-1, not with B16F10. When you put the tumor in the knockout mouse, you have an intermediate situation, so the growth is a little bit lower than here, but not a big phenotype. When we treat these mice that are knockout for SUV39H1 with anti-PD-1, now you get a very strong response, and when you measure the size of the tumors at the end of the experiment, you have a number of mice that really are completely protected. So better anti-tumor response by anti-PD-1. This is what happens in the tumor. Actually, in the tumor draining lymph nodes, you see again this increase. These are central memory CD8, so you have more central memory CD8 independently of the treatment in the tumor draining lymph node. And the phenotype in the tumor is increased effector cells. So you have strong infiltration. These are the percentages. These are the numbers. When you treat with anti-PD-1, in this particular model, not strong infiltration. The SUV have a little effect to start with, but when you treat the SUV knockout with anti-PD-1, you get better infiltration. This is due to an endogenous phenotype. I didn't tell you that, but this is really, we did all the bone marrow chimeras and adoptive transfer, and this memory phenotype that you see here is T-cell intrinsic. The cells that are in the tumor look exhausted by many criteria. This is an experiment where we use FACS to look for different markers expressed 1 by 1 or 2 by 2. And what you see is that in both CD8 memory and effectors, you have increased levels when you treat the knockout with anti-PD-1. They express a lot of checkpoints. These are the effectors, of course, have higher expression by their own. But when you treat the knockout with anti-PD-1, the cells express high levels of all these negative checkpoint molecules. They look exhausted. Now, this is EOMS expression and like 3 EOMS, which are also markers of exhaustion. They look exhausted, but I told you they reject the tumor better. And they also have some other characteristics. One of them is that they remain Ki-67 positive, so they are cycling, and you see here the anti-PD-1 knockout as compared to the others. And this is true in the memory and a little bit in the effectors. They are SCA1 positive. SCA1 is a marker of stemness in CD8 positive cells, and it's also an interferon responsive gene. And if you take them out and re-stimulate them right away, they produce interferon gamma, which the other cells in other conditions do not. So it really looks like the knockout plus anti-PD-1 gives a mixed phenotype. The cells look exhausted, express high levels of checkpoint inhibitors, but they produce interferon and they are still capable of self-renewing and cycling. So what we concluded from this, and this is a way to interpret this data, there might be others, is that, oops, sorry, is that SUV39H1 is involved somewhere in the transition between progenitor exhausted cells and terminally exhausted cells. It is an epigenetic regulator that blocks and suppresses gene expression, and we showed before, and we showed in this model, I didn't show you, it acts on different genes that are involved in loss of stemness and loss of memory, so like TCF1, for example, is 1KLFL2. I will show you a little bit of the molecular analysis later on. So our understanding is that this molecule, this epigenetic regulator, is a methyltransferase of histones, participates to establishing an epigenetic barrier that in the wild-type cells makes these terminally cells unable to be reprogrammed and to come back. They are not plastic enough, they are locked in this exhausted state. When you knock out the gene, the cells are more plastic, and then you can use anti-PD-1 to reprogram the cells and bring them down into effectors. This is a working hypothesis in a model. Because stemness and memory was associated in many different studies by other people to be associated with good response to CAR T cells in adoptive transfer models, we thought that maybe we should go and try to knock out SUV39H1 in CAR T cells and see what would happen in vivo. And that's what we did. Just a brief introduction to CAR T cells. So no, maybe I should show this before. CAR T cells is a therapy, an immunotherapy mode that takes blood of patients, modifies genetically the T cells from the patient with a chimeric antigen receptor that allows the T cells to recognize tumors, and then reinfuses tumors into patients. And it has had very, very strong success and complete responses in different types of B-cell malignancies. The most widely used CAR, which is approved by the FDA and has very, very strong 80 to 90% complete responses in different types of B-cell leukemia, is a CD19 CAR, which kills both leukemic cells and normal B cells, which is not a big problem in many patients, but can be in some cases. But 40 to 50% of patients relapse in the next 2 years, so persistence of the response is an issue. And the responses are not good in most solid tumors for the moment. And so there has been several studies, I mentioned that before, showing that stem memory CAR T cells, so when you have a high proportion of stem and memory cells among the CAR T cells that you infuse into the patients, their clinical responses are better. The there are different types of CAR T cells. They all have an extracellular domain that is composed of a single chain FV or an antigen binding device, an antigen that is expressed in the tumors, and then a transmembrane from many times from CD8, but can be from other molecules, and then a cytoplasmic tail that associates a CD3 zeta chain activation domain with items, 3 items, to either nothing or co-stimulation domains. And the two most used ones are CD28 and 4-1BB, and the approved CAR T cells are of this structure here. There were later on CAR T cells associating 2 co-stimulation domains, but they turn out not to be more efficient. So what is in the clinics right now is a CD19 binder and a 4-1BB or CD28 co-stimulation domain together with the CD3 activating domain. And this is what works very well. And so we thought, okay, if we are going to try to improve CAR T cells, we should use something that is already very efficient. Let's use the best. And so we took the 4-1BB CD19 CAR T cell, and the idea was to use the same pathway I just told you about, SUV39H1 knockout, and see what happens there. Actually, what we did at the beginning of this project on CAR T cells was to knock out either ZDB1, SUV39H1, TRIM28, which is an adapter in the pathway, or HP1, and just ask, okay, which one of these is going to increase better the memory phenotype? And this is one of the experiments we did for that. So these are primary human CD8 positive T cells, and we are using CRISPR-Cas9 to knock out either SUV or ZDB1 or CBX5, which is HP1. And you can see that the knockouts by Western blot are very clean. It's not as strong for HP1 alpha. And then we just used to screen this an in vitro assay where we re-stimulate human cells with anti-CD3 and look for the phenotype. And you see that when you look to the relative percent of cells expressing CCR7, CD27, these are memory cells. We see that the SUV knockout, like in mice, increases the proportion of these cells. These are other markers that we followed. Actually, ZDB1 is a big problem. It reduces, in most donors, the proliferation of the cells, and HP1 didn't show much. So this is a mild phenotype in vitro, but based on this, we went on to use, to generate CAR T cells with the CD19 for 1BB, CD3 zeta, so we call it 19BBZ CAR T. We use a lentivirus to express the CAR T. The expression is very high, between 50, 60 to 80% of the T cells, sorry. This is the knockout for SUV. We chose different guides. The knockout works pretty well. It is around 50 to 60% by RT-PCR, but by Western blot, it is much more efficient, and we really have a very nice knockout of the SUV. We also can follow the levels of H3K9 trimethylation by FACS, intracellular FACS, and we can show that the levels of so this is like the substrate of the SUV, are reducing the CAR T cells in vitro to some extent. So, first thing, very few results characterizing the re-stimulation of the cells in vitro. So you re-stimulate. So day 1 is already the CAR T cells. So 1 week, you need 1 week to generate the cells and express the CAR T and do the knockout. And then we re-stimulate several times along the weeks. What happens if you do that is that when you follow the numbers of cells, the SUV seem to remain, to contract. So when you start re-stimulating for 5, 6, 7 weeks, the wild-type cells lose the capacity to continue to expand, or even they start dying, and the SUV seem to do that a little bit better. And you have, again, what I showed you before. These are different markers for memory. The number of memory cells is increased. This is by FACS. The PD-1 positive, TIM3 negative actually also increased. The double positive are a little bit decreased. Tibet expression is also decreased. And then you can look by RNA to different memory-related transcription factors and markers like TCF7, which is increased in the first 2 weeks. LEF1 is increased a little bit later. IL-7 receptor is also increased. And KLRG1, which is an effector marker, is decreased at the later time point. So you basically have a mild phenotype of increased in memory markers in vitro. The strong phenotype when came when we looked what happened in vivo. So in this model here, we are using NSG mice, immunodeficient, a cell line of a leukemia, B-cell leukemia cell line called NALM6, which is extremely aggressive. So as soon as you put it, you can see here the growth of this thing is completely crazy. And we are using CAR T cells. We are injecting here less than 1 million CAR T cells because the CAR T cells are already very, very efficient. And what you see is that when you do that, these are injected at day 3. So you really control the tumor both with the wild type, the mock CAR T cells, and the knockout CAR T cells. And in this particular model, what happens is that as time goes by, mice start relapsing, and the tumor starts growing again. And this is what happened in all the mice treated with the mock CAR T cells. And what you see here already is that in the case of SUV knockout, the control is much more stable, and the mice don't relapse as much. Many of these mice are still free of tumor even after 40 days, and you have here the survival of the mice. We also used a different model. Here is a lung tumor model that is implanted in the lung. We expressed, to try to keep these great CAR T cells, we ectopically expressed CD19 on the lung tumors. We also now did it with another marker, with mesothelin, which is an endogenous tumor antigen, but here is CD19, and again we do the same thing. We wait for 21 days before we treat the mice with the CAR Ts. This is a less aggressive tumor, so it grows slower. At day 21 we treat, and what you see is that in both the wild type, with both the wild type CAR Ts and the SUV knockout CAR Ts, we have a very nice therapeutic response. But again, very rapidly after 1 to 2 weeks, all the wild type mice start relapsing while the SUV knockout CARs control the tumor in the long term. When we analyze the cells in this, so the first thing was to try to see if this prolonged control of the tumor was due to prolonged persistence of the CAR T cells. And this is what you see here. This is the spleen of the mice at day 49. The results are much more dramatic in the spleen than in the lung themselves. The cells persist very long time. You have a 42-time fold change between the mock CAR and the SUV knockout CAR. Both CD4s and CD8 persist much better when they don't express the SUV39H1 molecule. And when you do RNA-seq and do a differential analysis, you see that the molecules that are overexpressed in the SUV knockout CARs include, well, CEL, CD62L, KFL2 and 3, LEF1, and several others which are not indicated here, including IL-7 receptor and CD27. So you have an increase in memory markers in the cells. On the other hand, you have some effector markers that are down-modulated or up-regulated in the wild type. So because it looked like a persistence was increased, and that is why we were controlling the tumor better, we did this experiment, which is really a re-challenge experiment. So again, we put the A549, this lung model, at day 0. We treated at day 10 with a higher dose of CARs to have long-lasting control. And we waited for 2 to 3 months. At the time where the wild type were starting to relapse, but control was partial, we re-injected 2 million, 3 times 2 million cells in the lung of these mice. And that caused all the wild type mice to relapse, while the SUV knockout were still controlling the tumors even after 3 months in the mice. And here you have the numbers of cells, again, increased persistence of the mice, of the T cells, the CAR T cells at day 73 here. So the phenotype of the cells that live long and persist is this one here, CD27 KLRG1 negative. The proportion of the cells and the numbers of the cells are strongly increased as compared to the wild type. And the proportion of cells that express KLRG1, both CD27 positive and negative, are decreased in both cases. So basically, we are increasing the memory phenotype of, and this is true for both CD4 and CD8s. We did single cell analysis. This is single-cell transcriptomics, both in CD4s and CD8. We did that at day 8, at the peak of the response, and at day 28, so once the tumor were rejected and the T cells were persisting. These are all the, we had, in this particular donor, these are the CD4s and these are the CD8. There is a mistake here. So there were very few CD8 positive T cells and many more CD4s. We have 4 conditions, so day 8, day 28, wild type and knockout, and these are all the conditions together. This is what happens when you compare day 8 and day 28, mock and knockout. The cluster 2, which is shown here, are the memory cells, which are also shown here. What you can see, and the cycling cells are here. So what you can see is that at day 8, cells are cycling. They are at the peak of the response. You have a lot of proliferation, and you start having some memory precursors that turn out here. What you can see is that actually cycling is not very much affected between wild type and knockout, but it does go down when you look at day 28, where the cells are proliferating much slower. But both at day 8 and at day 28, the C2 cluster, which is the memory cluster, is increased in the knockout. We have here duplicates, biological replicates for each condition. And you can see here the statistics of that. So big increase, especially at day 28, in this C2 stem memory KFL2 cluster in the knockout. Also a little bit in the cycling cells. These are the proportions of the cluster. So this was to point out that when you look at the number of differentially expressed genes at both days, you see that the more dramatic effect is seen at day 28, and the bigger number of genes that change expression is in this cluster 2, which is the KFL2 at day 28. And again, when you look at differentially expressed genes, you see all the usual suspects, the same that you saw before, TCF7, LEF1, KFL2, which are these memory markers for T cells. So we are increasing T cells, we are increasing memory, we're increasing persistence, and actually what happens at these days, so this is a GSEA analysis with a stemness memory signature. It's just to point out that the increase in the C2 with these memory cells doesn't mean that the other cells are not biased towards memory. So the phenotype, the increase in the memory signature occurs in all the clusters CD4 and CD8. So the knockout of SUV is increasing the stemness and memory characteristics in all the different clusters of T cells that are knockout for SUV39H1. We used a TAC-seq, which is a transposase accessibility chromatin assay to try to see the opening of the chromatin. And we did that at the early time point, at day 8, to see if the programming of the cells was changed by the knockout of SUV. And maybe I won't go into the details of this because it's getting a bit late and you're probably as hungry as I am. But we could show that actually at day 8, you do have an opening of the chromatin in the precursors, and this is also true so these are the memory genes again, this is chromatin opening, this is single cell at TAC-seq, and so you see the opening of the chromatin at this memory loci early on during programming of the response at day 8, which causes so this is before you have a strong phenotype at the RNA level. So it's really the epigenetic programming of the memory response. And again, what we see with the stemness memory signature in the TAC-seq experiment is that the opening of the chromatin at the stem memory loci occurs in all the clusters. We also see that the effector signature is reduced, so it's stronger in the mock. So you have a dichotomy. In the wild type, you're biased for strong effector response, and in the knockout for memory. So let me just finish here with 1 or 2 slides trying to relate the result to the clinics. So what we did here is, first of all, to use a number of complete response signatures that are derived from patient studies. We have 3 here. One is Frayeta et al., Krishna et al., and Kosh et al. There's different models or different clinical trials, both in CLL and melanoma and one in lung cancer. But all these studies defined immune signatures, molecular signatures, in the product that are associated with complete response. And the question is, okay, are these signatures overexpressing the SUV knockout CAR T cells? And the response is yes. So this is a feature map that takes the same clustering that I just showed you before, which was done in our CAR T cells, either mock or knockout. And we are now plotting one of the signatures here, which is the Fraieta signature. And what you can see is that the signature is increased in the knockout and is especially strong in this C2 cluster, which is the one strongly affected by SUV knockout. You have here the quantification of this at day 8 and day 28. So you see that at day 28 in the knockout, the Fraieta signature is strongly expressed. And this is also true for this other complete response signature from other clinical studies shown here. The last slide is this one, where we did the reverse experiment. We looked at the SUV39H1 signature, and we tried to correlate this SUV39 signature with the different signatures that came from all these studies that were associated with complete response or with no response to CAR T cells and to immune checkpoints. And in all cases, but one, this study here by Chen et al., in all other cases, the signature in the SUV knockout is associated with complete response signatures in all these studies. So this pushed us to try to bring this to the clinics, and we are in the middle of that. It is an extremely heavy thing. Only doing one knockout in a CAR T cell requires a lot of technological developments and manufacturing and so on. But we're in the middle of that, doing that with this company I told you about, Memotherapeutics. So this is the conclusion, but let me not spend more time here. I think that I will end up with this slide here, which is to point out the people that did all this. So the mouse work that I showed you at the beginning was done by Letizia Niborski, an Argentinian postdoc, in collaboration with the team of Eliane Piaggio. And the part on the CAR T cells is mainly by 3 postdocs, Miguel Saitakis, Sheila Lopez, and Jaime Fuentalma. They got help and support by others. These are the 2 postdocs that started all this work in the lab over 10 years ago. And these are collaborating groups, and I really thank you very much for your attention.
Thanks so much. Thanks so much, Sebastián, for this wonderful talk. Excellent. So we have time for questions before lunch. 2-3 questions.
Incredible talk, thank you. The H3K9 trimethylation is also involved in the FOXP3 transcription factor and regulation of that. So do you see changes in CD4 for FOXP3?
No, we looked in for FOXP3 levels and activity. We saw absolutely no changes. With another member of this family, TRIM28, the knockout causes an increase in FOXP3 differentiation. I don't know why, because epigenetically there was no molecular basis to understand that, but yeah, it's not happening in this knockout. Yeah.
So this is going to be very interesting. A couple of questions. One of them goes to the transgenes of the CAR constructs themselves. So is this epigenetic mechanism controlling the expression of the CAR molecules at the integration sites in the genome? And the other question is about the potential toxicity. So are you expecting similar, less, or more cytokine release syndromes and potentially neurotoxicity?
Yeah, excellent questions again. So for the first one, we did the same with the CD28 CAR. And Michel Sadlin, actually, we are collaborating with him in a lot of this. He did all parallel work and found the same results in a CD28 model. We never saw any difference in the levels of CAR expression. I didn't show you here, but there are not changes between the wild type and the knockout. For the toxicity, this is a big worry. We are wondering what the authorities will say about bringing this to the clinics. Our arguments are the following. SUV39H1 is not associated with mutations in any leukemia or any other malignancy that is known. So it's not a mutated gene, unlike DNMT3A or TET2. which have similar phenotypes but are mutated in different hematopoietic malignancies. We do see prolonged survival, but we also see reduction in the numbers, and we never saw any mouse that was developing any type of CAR T-derived leukemic phenotype or clones that persist. And we don't see any increase either in cytotoxicity in vitro or cytokine release in vitro. So it's not a gene that is activating cells. Other effectors are rather reduced, both in listeria and a little bit here. We see less effector characteristics and more central memory. So we hope that that will convince them to let us go with this.
Hi, I'm here. Thank you. It was really interesting work. Do these mice, knockout soup mice, have any signs of autoimmunity or any?
Yeah, no, not overall. There are no tissue infiltrations and any kind of autoimmune syndrome or antibodies to DNA or. So it is really a central memory phenotype more than an effector memory phenotype. When mice become very old, both in wild type and in SUV, you see some signs of tissue infiltration and so on, but it doesn't look like we see any strong autoimmunity.
Sebas, I have a question. Okay, my question is whether the memory phenotype conferred by SUV39 knockout is also associated with changes in the glycolytic and oxidative metabolism.
So it can seem bizarre and strange, but we didn't look at that. We should. No, it's. I'm ashamed to say this here, but let me try to think if there is any. We should look at that. So there are changes in metabolism overall. The cells have a higher reserve, glycolytic reserve, and better fitness metabolically, but let's look at that together.
Yes. Why not?
Sounds great.
Gaby.
Okay, thank you very much, Sebastián. Now, you have a question, Thomas? Yes, Tomás has another question.
One more.
Yes.
Actually, I had two. The first question was Gaby's question, so I was wondering about the metabolism of these cells. So the second question is, you showed that in patients responding to the CD19 CAR T cells, the gene expression was like comparable with your knockout CAR T cells. I was wondering because CD19, like acute leukemia relapse is usually characterized by CD19 loss on leukemia cells. So I was wondering if the patients that do relapse, have you looked into the transcriptome of those CAR T cells to see if like those cells lack a memory like T cells phenotype or that can sustain your observations?
Yeah. I don't think we have a clear picture of that. From what I understand, roughly half of the patients that relapse are CD19 negative or low, and the other half are still expressing CD19. Now, is relapse in those due to lack of persistence? It's like the easiest way to understand that, and there was one patient case that was reported a few years ago with TET2, very long-lasting case of tumor control for years and years, and a clone of T cells became sustained proliferation, was TET2 knockout, so the CAR has inserted in the TET2 locus, suggesting that when you have a strong persistence, you control the tumor, but it's anecdotal. I don't think besides the correlation between the good clinical response. So one aspect of it is persistence. The other aspect I didn't discuss here as much is that actually SUV knockout cells seem to be less subject to exhaustion and to this terminal differentiation. So they remain, it's not only that you have more memory cells, but also the effectors seem to be more plastic and seem to be capable of sustaining a response in the environment of the tumor. And actually, you didn't say that, but we have a chemical inhibitor of SUV, and actually it phenocopies. We can combine it with anti-PD-1, and you have a better control of the tumor similar to what we saw in the knockout. So it's like, I think it's a good candidate to try to do something more, but I'm super curious to hear Jim's and Pat after the talk. So thank you very much to everyone for your attention.
Thanks. This was wonderful. So, I think we are all starving and we want to go for lunch. So we have 45 minutes and we wait for you at 3 p.m. again to start again the afternoon session. And we will go for lunch upstairs. Yes. First of all, toda la gente, todos los estudiantes del curso, ahora mismo nos vamos a sacar una foto afuera en la puerta. Sí, sí, sí. Y con las chicas. Vamos.
And one of those. Se viene Bafil, la Feria Internacional de Música.
Good afternoon. Can we start? ¿Podemos empezar? Vamos a empezar, así no nos vamos. Del curso nos íbamos a las 9 p.m. todos los días, pero acá no creo que nos podamos ir a las 9 p.m. Especialmente porque están muy cansados ellos. So we will run to our next talk. Our next talk will be delivered by Dr. Padmani Sharma from the clinic to the lab, investigating mechanisms of response and resistance to immune checkpoint therapy. And we will be given by the Doctor Honoris Causa from the University of Buenos Aires, Padmani Sharma.
Is it on? Yes. Muchas gracias. I'm very excited and grateful today to be honored in this ceremony, so thank you all so much. And I hope that I can share some of our experience with immune checkpoint therapy in the clinic. So just to say that I have both an MD and a PhD, and so I see patients in clinic 1 day a week to take care of cancer patients, and then the rest of the time I'm in the laboratory trying to understand how to develop the new treatments for patients. So this is bridge between the clinic and the lab is very important to me. And I think a lot of the work you're going to see is how do we bridge that in a way so that we can develop the next treatments to be even better. So as Jim pointed out, immune checkpoint therapy has clearly changed the way that we treat cancer patients. So I've been treating cancer patients for 20 plus years as an oncologist, and prior to immune checkpoint therapy, most of our patients who had stage 4 cancer had about a median survival of 6 months. So 6 months is a very short time, and despite our treatments, especially for example, in my specialty of bladder cancer patients, most of our patients passed away. Immune checkpoint therapy, as you can see, targets the immune system and targets T cells. And because it's targeting T cells, not tumor cells, the approvals that we were able to get are listed here. So you can see many different tumor types respond to immune checkpoint therapy. But as Jim pointed out, there are many research questions that we still have to address. So why do only some patients respond and others do not? How can we identify biomarkers that predict a response or the toxicities associated with the treatment? Are there biomarkers to enable patient selection? And can we increase the number of patients who respond to therapy? And are there other pathways outside of CTLA-4 and PD-1, PD-L1 that we can target? So to do that, we really had to rely on the system that we now like to call reverse translation. I think in the laboratory, you're used to the mice that are inbred and the disease is homogeneous. So in the laboratory, you can knock out a gene, knock in a gene, and really understand mechanism in the mouse. And this is very important for looking at mechanistic pathways, and it helps you to go from the basic laboratory to the clinic. However, in the clinic is where immune checkpoint therapy is being used right now. And we currently have more than 2,000, if not up to 3,000 clinical trials that we're doing with immune checkpoint therapy. And because of the number of clinical trials and the number of patients who are being treated, thousands of patients receiving the treatment, we now have to think about the reverse translation. How do we go from the clinic back to the laboratory? And so this is the way in which we think about it. The patients are polymorphic, the disease is heterogeneous, and now we have to take the patient data and guide what we do in terms of research. And patient data is really hypothesis generating. It's correlative data. It is not testing a mechanism. Patient data will always be correlative. And therefore, you have to use the patient data to generate a hypothesis and then test that hypothesis in an appropriate model in the laboratory. And so this is the process of reverse translation back and forth between the lab and the clinic that we implemented. In order to do that, to really get access to the patient samples, we had to rethink how we designed our clinical trials. So clinical trials, for the most part, are phase 1 safety dose escalation studies, phase 2 efficacy studies, and phase 3 for a comparison to standard of care. We decided that we needed another set of studies, which we called phase 1A or phase 2A studies. And these studies are what we call pre-surgical, tissue-based, or neoadjuvant approaches, meaning we give the treatment prior to surgery, Then we get access to all of the surgical samples that we can then use in the laboratory to understand what's happening within the tumor microenvironment. So this allows us to have access to samples to understand clinical signals, as well as biomarker analyses, and to gain mechanistic insights. We established these in 2004, and I wrote the first clinical trial, which initiated in 2006. So I wanted to highlight for all of the students in the audience that I did all of this before there were any FDA approvals. So these were at the time considered experimental drugs. And as you can imagine, there are many experimental drugs being tested right now. Everything from anti-LAG3, anti-TIGIT, anti-ICOS, these are all experimental drugs. So there is a way to really understand from the human perspective, what are these experimental drugs doing to the human immune response? And although the mouse and the humans, we have to go back and forth between the two, I just want to remind you that there are a lot of things happening within a human immune response, which is very dynamic, that you won't be able to detect in a mouse system very easily. So it's important to bring these two things together. So the first clinical trial is highlighted here that we did in this neoadjuvant approach. And this was the first neoadjuvant clinical trial ever with immune checkpoint therapy. It was also the first clinical trial in patients with bladder cancer ever. And so we had a small trial, and again, for those of you in the audience who are treating patients, we didn't need hundreds of patients. This was a 12-patient study. And it doesn't matter which hospital you work at, 12 patients are pretty doable for any hospital to accrue the small trials. So I encourage you to think about these as ways to really understand what's happening within patients. So we asked 12 patients who were already scheduled for surgery because they had localized bladder cancer to then get treated with the anti-CTLA-4 prior to their surgery. And then we had access to all of their tumor samples. We had another 12 patients who did not get the drug and went directly to surgery. So that was our control group. And so we could then ask, what does anti-CTLA-4 do to the tumor microenvironment in the setting of bladder cancer patients? So this trial gave us the clinical signal for safety because it was the first study to show that it's safe to give anti-CTLA-4 prior to surgery, and so it set up the entire paradigm for neoadjuvant immunotherapy. And second, it gave us a signal that bladder cancer could respond to immune checkpoint therapy because it was the first time we showed that 3 patients developed pathologic complete responses, meaning all of their tumors went away, and that led us to write the clinical trials then with anti-PD-1 in patients with bladder cancer. But importantly, it also gave us access to the tumor samples. And then we can compare the pre and the post-treatment samples. And in the pre-treatment compared to post-treatment, you can see this really significant increase in the infiltrating immune cells that were associated with treatment. And you could see CD3, CD4, CD8, granzyme, and these are all T-cell markers, but you could also see B-cell markers such as CD20 was increasing as a result of treatment. Importantly, what we saw in the differentially expressed genes was that the top 10 genes were related to the immune response. And this was to be expected because we increased the number of immune cells within the tumor microenvironment. What wasn't expected was that we had the ICOS pathway as a top pathway. And ICOS had not previously been studied in the setting of anti-tumor responses. In another neoadjuvant clinical trial, this is the first combination, tremelimumab plus durvalumab, which is an anti-CTLA-4 plus anti-PD-L1. This was the first combination trial, again, in localized bladder cancer patients. And again, you can see after this clinical trial, we had patients with pathologic complete responses. But importantly, we also had access to the tumor tissues. And you can see now what we were finding was that patients who responded to therapy had the significant increase, or actually I should say higher density of the tertiary lymphoid structures. And tertiary lymphoid structures shown here are comprised of the T cells, B cells as indicated by CD8 and CD20, as well as dendritic cells, as you can see by CD21. Importantly, when we looked at the CD4 T cells, the T cells in the responder patients, which are R, had these ICOS positive expression, and the non-responders did not have ICOS associated with them. So what is ICOS? I think Jim briefly introduced this already. It's inducible co-stimulator. It really belongs to the same family as CD28 and CTLA-4, but again, a role in anti-tumor responses had not been established. So having access to the samples meant we could go back to the actual human tumors and sort the T cells. So here I'm just showing you that in human tumors, the non-malignant tissues did not have much ICOS, it's about 13%. The untreated tumor tissues are about 16% of the CD4 T cells, but the anti-CTLA-4 treated tumor tissues, this was now significantly increased at about 40%. We sorted the T cells from the patient's tumors, and because the patient tumors also had antigens, so here is NY-ESO-1 as a tumor antigen that's expressed on the tumors, we could ask whether or not these T cells recognized antigens such as NY-ESO-1. And in the assays, you can see now these T cells were producing interferon gamma, as well as TNF alpha and MIP1 beta as a result of seeing antigen. So these were antigen-specific T cells. So ICOS is definitely playing a role in the anti-tumor response. We also asked the question whether or not patients who had the ICOS-positive CD4 T cells had better outcome, and they had much better survival, as shown here, compared to patients who did not. So that was all the clinical data. So I had clinical data, and now I had to generate the hypotheses from the clinical data. And so the clinical data suggested to us that the ICOS ligand pathway was necessary for effective anti-tumor responses. And to test this hypothesis, we went back to the laboratory and we used the mouse models, whereby we could then ask in wild-type mice, compared to the ICOS knockout or the ICOS ligand knockout mice, what happens after treatment with anti-CTLA-4. And you can see again, in wild-type mice, you have 80 to 90% of tumor rejection, but in the ICOS knockout and ICOS ligand knockout mice, you can see impaired anti-tumor responses. So then the second hypothesis is why? Why do you need ICOS to generate the anti-tumor response? And so our second hypothesis was that ICOS can induce signaling via the PI3 kinase pathway to promote T-bet expression with subsequent Th1 responses and tumor rejection. So as you know, T-bet is the transcription factor that drives interferon gamma production and Th1 responses. And just to show you here that ICOS, similar to CTLA-4 and CD28, it has a PI3 kinase binding domain in the intracytoplasmic tail. So now we wanted to try and understand whether or not T-bet expression was changing in wild-type mice compared to the ICOS knockout mice. And what you can see, if we took T cells from the spleen of untreated mice, these T cells, either wild-type or ICOS knockout, do not have T-bet expression, or minimal T-bet expression. After treatment with anti-CTLA-4, we took T cells from the spleen, tumor-draining lymph node, or tumor-infiltrating lymphocytes, and compared wild-type and ICOS knockout mice. And after treatment, the T cells in wild-type mice clearly increased T-bet expression, but the ICOS knockout mice failed to do the same thing, clearly indicating a lack of Th1 responses in these mice. But that really told us that ICOS was important. It didn't tell us if the PI3 kinase domain was important. To understand if the PI3 kinase domain was important, we used another mouse model, and this is the ICOS-YF mouse. And in these mice, ICOS exists. However, there's a single amino acid change. So we changed Y to F so that the PI3 kinase domain is no longer capable of binding PI3 kinase. And because of this single amino acid change now, when you looked at the wild type compared to ICOS-YF mice, again, these mice all express ICOS, but the ICOS-YF mice cannot signal through PI3 kinase. So now we can sort the ICOS positive and ICOS negative cells from both wild type and the ICOS-YF mice. And we can ask after treatment what happens to T-bet expression. And only when we have the ICOS positive cells in wild type mice can you see T-bet expression significantly there. However, even in the ICOS knockout, ICOS negative cells in wild type mice, you lose that. And definitely in the ICOS-YF mice, you never see the T-bet expression. And it's clearly indicated the importance of the PI3 kinase pathway, and may be the reason that when we use PI3 kinase inhibitors, they can inhibit actually immune responses that help with tumor rejection. The third hypothesis we had was that the ICOS ligand pathway can be targeted to improve treatment. And I'll quickly show this because Jim already showed it. When we targeted both ICOS and anti-CTLA-4, you can see the significant improvement in tumor rejection and survival of the mice in wild-type mice as compared to the ICOS knockout mice. So clearly there are multiple companies now that are developing ICOS targeting agents. What I have to say is that they have to be sure that they're developing agonistic anti-ICOS antibodies, because too many companies develop an antibody and don't test it for true agonistic function, and an antagonistic antibody will not do the same thing as we showed here. The other thing is that clearly we showed that this is important in the setting of anti-CTLA-4. So if they want to combine with immune checkpoint therapy, they need to use anti-CTLA-4, not anti-PD-1. So these are the two things that, I think is important as the field moves forward. So I just want to point out that we really led the field in terms of neoadjuvant clinical trials. So where are we now with neoadjuvant clinical trials? We conducted the first study, and I'm so thrilled to say that the field continues. So at MD Anderson, I'm thrilled to work with some of the best people, and Tina Cascone is one of the thoracic medical oncologists and a fabulous physician scientist who recently published her data on neoadjuvant immune checkpoint therapy in lung cancer. So here you can see this was the design of the clinical trial where she compared anti-PD-1 to anti-PD-1 plus anti-CTLA-4. And here is her data showing that, again, the waterfall plot. And what she really found was that even on radiologic studies, if you look at RECIST response, you can see even when the RECIST response says stable disease, when you get the actual pathologic specimen in the pathologist's hands, there actually are no tumor cells. So the pathologic responses are even better than what you're seeing on the radiology and the CT scans. So again, the importance of neoadjuvant studies for being able to make these two comparisons, telling us that RECIST criteria may not actually be the best way sometimes to study what's happening in the patients. Here's another study that was conducted by Tina's group, and here they're combining anti-CTLA-4 with an anti-CD73, an anti-NKG2A, and an anti-STAT3, as compared to just anti-PD-L1 durvalumab alone. So it's anti-PD-L1 plus anti-CD73 or an anti-NKG2A or anti-STAT3. And when you look at this now, you're trying to understand, when do PD-L1 negative patients respond to treatment? Because a lot of times the anti-PD-L1 works with PD-L1 positive tumors. They were trying to understand, when are you getting responses in PD-L1 negative tumors? And when they looked at the waterfall plots again, you can see that the majority of the responses were happening here, where they were able to find now the PD-L1 negative patients, which are in these blue, the PD-L1 negative patients who are having these kinds of responses with shrinkage of the tumors. And I'm sorry for those of you who may not know the waterfall plots, this is just a decrease in size of the tumors for each individual patient. And you can see the majority of the PD-L1 negative patients were responding when they gave a combination with anti-CD73. So the importance, again, of a neoadjuvant trial to look at multiple combination strategies to pick the winner. And then I just want to highlight here, this is again a different tumor type, cutaneous squamous cell carcinoma of the head and neck, just to show you the broad way in which immune checkpoint therapy is affecting patients. And when we looked at this study, it's a fabulous thing when you as an oncologist can see this many patients who are in the side of the waterfall plot where their tumors are shrinking. So you can see in this neoadjuvant clinical trial, we had the ability now to look at how many pathologic complete responses were occurring, and therefore these patients will continue to do well. Finally, I want to highlight that in melanoma, of course, neoadjuvant immune checkpoint therapy has been going on for a while. This is studies from Dr. Amari and Dr. Wargo. And they looked at nivolumab or ipilimumab plus nivolumab. Again, small numbers of patients, but you can see that the response rate for pathologic complete responses, 45% in the combination group versus the monotherapy. These are, of course, not statistically comparable due to small numbers, but gives you an understanding of how you can move forward with the combination therapy. And then what about neoadjuvant versus adjuvant treatment? This study was done by Dr. Sapna Patel at MD Anderson to look at neoadjuvant compared to adjuvant anti-PD-1. And what you can see is the waterfall plot, again, the numbers of patients who are responding as a result of treatment. But when they looked at the event-free survival for patients, does neoadjuvant or adjuvant treatment do better? You can see it's the neoadjuvant treatment that had much better outcome compared to adjuvant treatment, clearly indicating the importance of neoadjuvant therapy. So now that I've, hopefully gotten you very excited about the neoadjuvant approach, let's talk about how we can use that neoadjuvant approach to really understand tumor types that are not responding to immune checkpoint therapy, right? So if you think about it, these are the tumors that we talked about here: melanoma, lung cancer, bladder cancer, all up here, head and neck cancer. These are all up here. And they're considered to have lots of tumor mutations. And because they have lots of tumor mutations, they have lots of antigens. And because they have lots of antigens, they have lots of T cells. And because they have lots of T cells, they respond to immune checkpoint therapy. However, tumors such as prostate or pancreatic or glioblastoma are down here. And that's because they have much fewer mutations. And because they have fewer mutations, fewer T cells. And because of fewer T cells, it's thought that they're not going to respond to immune checkpoint therapy. But as an immunologist, we know that actually you only need one T cell and one antigen to start the response. You don't need hundreds of antigens. And all of these are tumors, so clearly they must have mutations somewhere that present as an antigen. So the question we wanted to ask, is it true that immune checkpoint therapy is not generating T cell responses against antigens in these tumor types that have few mutations and in these tumor types that have not been showing great responses to immune checkpoint therapy? So to look at that, we conducted a clinical trial, and this is in 30 patients. And these patients had metastatic castration-resistant prostate cancer. And so we were able to resect a single metastatic lesion, perform exome and RNA sequencing, identify the tumor mutations, treat the patients with anti-CTLA-4, and ask whether or not the T cells can recognize these mutations. And so I'm just going to show you one patient here. This is patient number 7. This patient had 2 mutations, one in rogue guanine nucleotide exchange factor 37 and one in dihydropyrimidase. These were single amino acid mutations. And even though there were single amino acid mutations, you can see prior to treatment with ipilimumab, pre-ipi time point, the T cells were not recognizing this mutated antigen. But after treatment with anti-CTLA-4, now you can see the T cell response against the mutated antigen and not against the wild type antigen. And so these assays, these are ELISpot assays that were done with positive and negative controls. But again, here are the pictures to show the response to wild type antigen versus the T cell response to mutated antigen. Clearly, the T cell can detect the mutated antigen in patients with prostate cancer. So it's not that prostate cancer is not responding to immune checkpoint therapy due to a lack of T cells that can recognize the mutations. So maybe the issue is that the T cells are not getting into the tumor microenvironment. So is it possible that T cells are there and they're recognizing antigen, but they're unable to infiltrate into prostate tumors? So we had another neoadjuvant study, and this again is a neoadjuvant study. You can see the year was 2009, and I'm highlighting it again to show how important it is to do these studies even when you don't have the FDA-approved drugs. So here we gave ipilimumab to patients who were scheduled for prostatectomy. These were 20 patients with localized prostate cancer. And we can compare the pre- and post-treatment samples. And clearly, prior to treatment, prostate cancer is a cold tumor. And it's considered a cold tumor because it has very few infiltrating immune cells. However, after treatment with anti-CTLA-4, now you can see it becomes a hot tumor because now you see all of these infiltrating immune cells that go into the tumor. So again, the issue is not that T cells don't infiltrate. They do infiltrate into the prostate tumors. But as immunologists, we should have been prepared for the yin and yang. Because when you drive immune responses in one direction, it tries to control itself. So if you give T cell receptor signaling and CD28 signaling, what happens? You upregulate CTLA-4 to control that immune response. If you block CTLA-4, you still have to deal with PD-1 because that also tries to control the immune response. Here, we were driving an immune response in prostate cancer, and what we found was that there was definitely pathways that were being upregulated to control the immune response. And for the first time now, we can find PD-L1 and VISTA, which are 2 inhibitory pathways that were being upregulated in prostate cancer. And just to convince you that the gene expression study is being reflected in protein and IHC, you can see prior to treatment, there really is no PD-1, no PD-L1, and no VISTA. We never could find that within the prostate tumor microenvironment. After treatment, though, now you can see lots of PD-1, PD-L1 on immune cells and on tumor cells, and VISTA now on myeloid cells, and I'll show you that. So the thing is that we have to be cognizant when we drive the immune response in one direction, you will have compensatory inhibitory pathways. Which pathways and in which setting, we don't know, and we have to study that in patients longitudinally. So if I only had one time point in this study, I wouldn't have seen this. But because we have both the pre and the post-treatment, now you can see how the immune response changes over time. So both VISTA and PD-L1 are truly inhibitory pathways. When we put them in an in vitro assay, you can see we take the T cells from patient and stimulate with anti-CD3 in vitro. The T cells make interferon gamma and TNF alpha. But if you have PD-L1 or VISTA or the two together, you see this significant decrease now in the interferon gamma and TNF alpha. So we worked with Bristol Myers Squibb to then propose a new clinical trial for patients with prostate cancer, and this was a combination of anti-CTLA-4 and anti-PD-1. So I have to point out, do I think anti-CTLA-4 and anti-PD-1 by itself may be enough? Maybe not. We may need to block VISTA as well, but at the time we didn't have an anti-VISTA antibody. But just to say that we were finding now that patients had better responses to immune checkpoint therapy. And for metastatic prostate cancer, that was very important. But the thing was that the combination therapy also led to increased toxicities and adverse events. And so the dosing and the schedule of these two agents will need to be figured out. What we did see at the time was that the patients who responded to the combination therapy were patients who had soft tissue metastases, meaning they had metastases to lungs or to lymph nodes. Patients who had metastases to the bones did not respond as well. And so prostate cancer, when it metastasizes, predominantly goes to the bones. So why do bone metastases not respond as well as soft tissue metastases? And so again, we went back to the patient samples because we collected samples from the patients. And you can see the soft tissue metastases compared to the bone metastases, pre-treatment samples, post-treatment samples. What you find is that this Th1 signature significantly increases in the soft tissue metastases after treatment with anti-CTLA-4 and anti-PD-1. However, the Th1 in the bone never increases, and each one of these graphs is a different patient. You can see between pre and post responses for Th1, it never increases in the bones. And the bones actually increase this Th17 response. So we wanted to understand why is it that the bone microenvironment would increase Th17 and not Th1? And so we went back to the laboratory and we designed an immunocompetent mouse model whereby we can take castration-resistant prostate cancer, inject it into the bones of mice or into the subcutaneous areas of mice to mimic the soft tissue or the bone metastases. And then we treat the mice with anti-CTLA-4 and anti-PD-1, similar to what we did in the clinical trial, compared to the IgG control. And you can see again, in the bone lesions, you never increase the Th1 response. You do increase Th17, completely reproducing what we found in the clinical scenario. Again, in the soft tissue lesions, you increase the Th1, and actually you don't increase or Th17 decreases, and that's because it's decreasing because you're skewing the T cells towards Th1. So I'm not going to go through all of the details of what we looked at, but for you to get Th17 development, you need cytokine signals, IL-6 and TGF-beta. And what we found is that in the bone microenvironment, the IL-6 levels were always high. BM is for bone microenvironment, and BMT is bone microenvironment with a tumor. So it doesn't matter if there's a tumor or not, IL-6 is always high. But TGF-beta is only high when there's a tumor. And we showed in the manuscript that this is due to osteoclast activity. And so when you have these two together, now you will skew the T cells towards Th17. So we proposed blockade of TGF-beta with an antibody, and the green boxes showed that when we block TGF-beta in combination with immune checkpoint therapy, now we could decrease the tumor volume in the bone lesions. And we can, for the first time within the bone lesions, we had expansion now of the Th1 CD4 T cells and expansion of CD8 T cell clones. And this change in the immune response then led to us having now more improved anti-tumor responses and survival in these mice with the bone lesions. So we have a new clinical trial now for patients with bone metastases where we're using anti-TGF-beta plus immune checkpoint therapy that we're developing. Actually, we're waiting for the right anti-TGF-beta because there are multiple being tested in phase 1 studies. But the bone microenvironment data really told us that different microenvironments in the same patient will have different immune responses. So a patient who has lung metastases will have different kind of immune response compared to somebody with bone metastases. And this is important because it tells you that the specific organ niche drives the kind of immune microenvironment that exists. And so we took that lesson to glioblastoma, because glioblastoma is in the brain. And we wanted to ask, what is the specific immune microenvironment within glioblastoma in the brain compared to other tumor types? So we took human tumors and we compared glioblastoma to lung cancer, renal cell carcinoma, colorectal cancer, and prostate cancer. And we did a significant number of analyses. Here, this is just showing the CyTOF analyses to look at specific immune cell subsets. And you can see in glioblastoma, we found this L8 subset that only exists in glioblastoma. We couldn't find it in any of the other tumor microenvironments. And even after treatment, so this is untreated and treated, anti-PD-1 treated GBM, this L8 subset in each of these patients persisted. So the L8 subset we knew now was a significant proportion in GBM. And when we did single-cell RNA sequencing, we found that this L8 subset had a macrophage signature, an immunosuppressive signature, and a hypoxia signature that was associated with it. We then proposed that if we used a CD73 knockout subset, because again, the L8 subset was CD68 positive, CD73 positive, if we used a CD73 knockout mouse compared to wild-type mice, we would see a difference with immune checkpoint therapy in the treatment of GBM. So in this mouse model, you see we have the wild-type mice and the CD73 knockout mice. And when you use the CD73 knockout mice and inject into the brain the glioblastoma cells, and then treat with anti-CTLA-4 and anti-PD-1, this is the red line, now for the first time you're seeing really significant improvement in treatment of these mice. And so we think that CD73 now is a new target for glioblastoma treatment because it's blocking the myeloid cell component that seems to be the most immunosuppressive within GBM. So we wanted to also ask, are there upstream elements? And I think this was very nicely introduced in the by the previous talk about epigenetic elements, so that we're really looking at what are the epigenetic elements that can control immune cell differentiation. So here I just want to point out that when T cells use CD28 and T cell receptor signaling, one of the things that happens is that they upregulate this EZH2. And EZH2 is critical because it maintains stabilization of FOXP3 through the trimethylation of H3K27. And so because we're driving the on signals, CD28 and T cell receptor, the on signals, these on signals drive the T cell to function, but they also drive inhibitory pathways to control the T cell response. And this is one of the ways in which CD4 T cells can do that, by increasing FOXP3. In patients who did not respond to anti-CTLA-4, we found that those patients had an increase of this EZH2 expression, indicating that EZH2 could be controlling the immune response in patients. And so we proposed looking at what would happen to these FOXP3 positive cells if you used an EZH2 inhibitor. And so we used the FOXP3 GFP mice, whereby we can sort FOXP3 cells based on GFP expression, and we could incubate them then with or without an EZH2 inhibitor, perform RNA sequencing, and ask whether or not we change the function and phenotype of these cells. And so when we did that, we found that with EZH2 inhibitor decreased the FOXP3 expression, which is important again because FOXP3 is a transcription factor that allows for the T cells to become immunosuppressive. And it also increased these interferon gamma related genes, CXCL10, granzyme B, and interferon gamma. So even though that there were these FOXP3 cells, they were now behaving more like effector cells and less like suppressive cells. In a functional assay, again, without the EZH2 inhibitor, you can see in the CFSE dilution analysis, you can see how these regulatory T cells can suppress the effector T cells from proliferation. But once you have an EZH2 inhibitor now, you can see that the effector cells can clearly proliferate, and these regulatory T cells are no longer able to suppress the immune response. So we did a preclinical mouse study whereby we used an EZH2 inhibitor in combination with anti-CTLA-4. And so in this study, this is showing MB49. We did another model too, but in both models, you can see that the combination led to decreased tumor volume and increased survival as a result of blocking the EZH2 pathway. So this led us, as I mentioned, we're always doing these iterative studies. We learned from the patients that EZH2 increased when they didn't respond to therapy. We took that back to the laboratory and learned that in EZH2 mice, if we blocked EZH2 in mice, we can improve the response. And now we take that back to the clinic, and we wrote a new clinical trial whereby we're giving an EZH2 inhibitor, DS3201, in combination with anti-CTLA-4. So this clinical trial is currently accruing patients, and so hopefully I'll have an update on what happens, but it's just a way to show you how the iterative process works as you learn from the patients, test in the animal models, and come back to the patients again. And hopefully, as you can see, we're collecting the tumor samples again through this trial, so we'll learn from this as well to understand what other potential resistance mechanisms exist. I do want to touch a bit on biomarkers of selection for patients because I think it's very important that we think about combinatorial biomarkers if we're going to start enrolling more and more patients for immune checkpoint therapy. And the biomarkers should reflect both the tumor cell status and the immune microenvironment. I think we're used to PD-L1, which reflects only the immune microenvironment, or we're used to things such as DNA damage repair or MSI high, which only reflects the tumor cells. But if we could use the two together, we may have a better way of selecting patients for immune checkpoint therapy. And so we did a small study, again, this is in patients with bladder cancer, where we had 12 responders and 13 non-responders. And we asked, were there specific mutations within the tumor cells that predicted for patient response? And here we found that patients with an ARID1A mutation were the patients who tended to respond. So if you had ARID1A mutation, you had a better response to immune checkpoint therapy. And when we went to a larger clinical trial and looked at the retrospective analysis, in this trial with Checkmate 275, we had 139 patients, and this is the Genentech and Vigor 210. We had 275 patients. Retrospectively, if you looked at it, the patients who had ARID1A mutation would have had a much better outcome compared to patients with ARID1A wild type. And when we went back to the laboratory, we knocked down ARID1A in the bladder tumor cell lines. And when you knock down ARID1A, you see now you enrich for interferon gamma. And if you treat it with anti-PD-1, you can now see that the knockdown had a much better response to anti-PD-1 compared to the wild type. So ARID1A seemed to be a real good indicator of if you have loss of ARID1A, that tumor cell is now producing more interferon gamma and more likely to respond to immune checkpoint therapy. So we went back now to look for an immune biomarker. So that our ARID1A is a tumor cell biomarker. What's the immune biomarker? Using the same kind of study in the 25 patients, we identified CXCL13, and then we went back to the retrospective analysis of Checkmate 275 and Vigor 210. And in this large cohort, if you stratified patient according to CXCL13 high expression, medium expression, or low expression, it's the patients with CXCL13 high expression that had much better outcome. So now we have an immune biomarker, CXCL13, and for those of you who follow the chemokine field, you know that CXCL13 is super important for T cell recruitment and B cell recruitment within the immune microenvironment. And as I showed in the beginning, T and B cells are an important part of tertiary lymphoid structures. So being able to recruit both of those to the immune microenvironment would be important for responses to immune checkpoint therapy. And if you looked, we then went back to the lab to test the idea that CXCL13 was important. And in CXCL13 knockout mice, where we implanted tumors, you can see that if you lost CXCL13, you knocked it out in the mice, they no longer responded to anti-PD-1 in the same way as the wild-type mice treated with anti-PD-1, indicating, again, the importance of having CXCL13 expression in the immune microenvironment. When we retrospectively looked at the two data sets we had, Checkmate 275 and Vigor 210, if you combined ARID1A mutation and CXCL13 expression, the overall survival significantly improves in terms of these patients if they had both of these biomarkers within the microenvironment. So we wrote we wrote 2 new clinical trials. One is a protocol that's enrolling patients only with bladder cancer, and they must have an ARID1A mutation. And then we stratify their response based on CXCL13 expression. And the other cohort is any patient, any tumor type. It doesn't matter the type of tumor. As long as they have an ARID1A mutation, they can now receive treatment with anti-PD-1. And so that's how the biomarkers, I think, can inform us and really guide us to make sure that we're taking the right patients into clinical trials. So on my final slide, I'm going to pitch a new concept. I think back in 2004, I had a similar talk where I pitched the concept of neoadjuvant treatment for immune checkpoint therapy, and at that time a lot of people didn't believe that immune checkpoint therapy could be helpful in a neoadjuvant setting, so let's pretend it's 2004 and I'm pitching a whole new concept again. And in this concept that I'm pitching, it's about stage 4 cancer. So in stage 4 solid tumors, patients have disease throughout the body. And in that setting, we never do surgery to just remove one lesion. No surgeon will take you to just remove a single lesion for stage 4 disease in the solid tumor setting, because we've done it. And when we've done it, it's failed to show any improvement in survival or clinical benefit. So based on the data that removing a single lesion in the stage 4 setting is not helpful, it's never been done again. However, that was all done in the era of chemotherapy and the era of targeted therapy. We've never done it in the era of immunotherapy. And so the thinking from my standpoint as an immunologist is that removing a single lesion does a few things. One, it allows for antigen release to prime an immune response that can then take advantage of immune checkpoint therapy. And 2, it balances the playing field. Because if you have a large, bulky one tumor and 300 small tumors, if you remove the large, bulky one tumor that T cells can't get to very well and leave the 300 small tumors that T cells can get to, then you can balance the playing field for the immune response to then allow for clinical benefit. So we conducted a clinical trial, keeping in mind that the clinical trial was for safety, so I'm not going to be able to show you statistical data for efficacy. But we did look at survival benefit as an ad hoc analysis. So that's a caveat, because we had to write the trial more for safety. Based on safety, and this is unpublished data right now, but based on the safety, we were able to complete the trial. 105 patients with stage 4 clear cell renal cell carcinoma that we then gave immune checkpoint therapy, either as anti-PD-1 alone, anti-PD-1 plus bevacizumab, or anti-PD-1 plus anti-CTLA-4. So we're able to give 2 to 3 doses of immune checkpoint therapy, take the patient to remove a single lesion, and then continue just anti-PD-1 alone. And when we compare the patients who went to surgery versus who didn't, because again, after this treatment, they were evaluated by surgeons to say, yes, they can remove a single lesion, or no, they can't. And so in this ad hoc analysis, and I cannot compare this statistically, so take that with a grain of salt, what we found is the patients who went to surgery had a 2-year overall survival estimate of 86%, and the patients who did not make it to surgery had a 2-year overall survival estimate of 48%. So it's very hypothesis-generating data that we will need to test in another prospective study. But I clearly think that this concept of stage 4 solid tumors need to be reevaluated in the era of immunotherapy, whereby removing a single lesion may actually be beneficial in the setting of immunotherapy, and this will need to be tested in larger studies. And again, because we have access to the tumor samples, because we're removing a single lesion, I just want to show you that we're able to do correlative studies on metastatic lesions now. And so, as you know, there's very different immune microenvironment in primary tumors versus metastatic tumors. Now we can start to address these issues. So putting both the clinician hat and the researcher hat, you can combine these things to start to see ways to really bridge the questions that we need to ask. And hopefully we're providing benefit to patients as well. So I'm going to stop there and just show you this cartoon that says that the field has a lot of work to do, because all of these agents are being developed by multiple pharma companies, but they don't know how they work. They don't know where they work, in which patients they work, and at what time point in the immune response they work. So we all have a lot of work to still do. But clearly, immune checkpoint therapy has joined the whole rank of surgery, radiation, and chemotherapy, and is now a new pillar of cancer treatment. But we have to move forward with combination, because that is the future, combination strategies. But in order to do that, in order to understand which combinations to give to which patient, we have to look at both pre and on treatment human tumor samples, because the human immune response is so dynamic over time, so we need to look at that carefully. We also need to look at organ-specific microenvironment, because each organ has its own immune microenvironment that may allow for a good response or maybe not such a good response. And then I hope I've convinced you that these small clinical trials of 10 to 20 patients in the pre-surgical or tissue-based setting, neoadjuvant approaches, really do provide a feasible platform to study the biologic effects in patients and provide insight into mechanisms that can be targeted for rational combination therapies. So I'm going to stop there. I have many, many people to thank. Their names from my lab are on the slide. I just want to highlight JJ Gao and Sangeeta Goswami. They both spent 6 years in my lab and did a lot of the work that I showed, but now they have their own labs and they're doing phenomenal work. And I also want to thank the patients, because these patients participate in our clinical trials so that we can have access to all of their samples. So thank you so much.
So thanks so much, Pam, for this wonderful talk. Thanks so much. This was great. So are there any questions in the audience? Yes.
Thank you for the talk. It was incredible. Do you think that a breast cancer patient would benefit also with the neoadjuvant scheme?
Yeah, I think so. And I mean, breast cancer is actually the tumor type where neoadjuvant approaches have already shown so much benefit, right? I mean, even chemotherapy for breast cancer gives you large pathologic complete responses. So combination with immune checkpoint therapy would make sense. But I think we have to try and select the patients based on appropriate biomarkers. That if we do a large trial with lots of different patients and we don't really understand what the appropriate biomarker is, we may have a negative trial. Because it's too diluted with other, issues that's happening in the study.
I have actually a question.
Yes.
Could you associate the expression of CXCL13 with the formation of tertiary lymphoid structures?
Absolutely, yes, we do. See, I didn't have time to show. I could give an entire talk on tertiary lymphoid structures because we've been so fascinated by them.
Yes.
But yes, CXCL13 is definitely part of the tertiary lymphoid structure formation.
Great. And do you see a room for targeting rank ligand in addition to TGF-beta in bone?
Yeah, we tried to get samples from patients who are on rank ligand to see if we see a change in the TGF-beta and the immune microenvironment. It's been a little bit difficult to get those samples, but in the few that we saw, we do see a change in TGF-beta, but not a change in the immune microenvironment per se. Okay. But that may be because they're not getting immune checkpoint therapy with the rank ligand. So I have to, try and figure that out.
Both of them, there are no trials with both of them.
Right, there are no trials with both of them.
Es importante que para hacer las preguntas se pongan de pie, por favor, para el registro.
Hi, great talk.
Thank you.
Going to the last concept, the solid tumor concept.
Yes.
What about like the trials or the efforts to combine DNA damage response, like PARP inhibitors and anti-immune checkpoint therapy? I mean, would that be a bad idea from your perspective? Because you would be like not using the immune perspective to dissect the big tumor, but the DNA damage.
No, I think it's fine as long as you're doing the correlative science and testing the hypotheses appropriately. I'm not saying that this is just for immunotherapy, but I think, again, I don't know that PARP inhibitors by themselves or DNA damage repair agents by themselves may have such an impact, but I do think that if you give the DNA damage repair agents or DNA damaging agents or PARP inhibitors, you can then generate antigen release due to cell death, and then come in with immunotherapy to really take advantage of that. So the timing and the schedule of when to give it becomes important.
This is kind of the second time I see ARID1A mutations being correlative. So what do you think is the biology behind?
What do I think?
Is the biology behind it?
So yeah, again, I showed briefly, but ARID1A mutations actually, because it's part of it's actually allowing for more interferon gamma. You're seeing an interferon gamma response, and you're allowing for upregulation of all of those interferon gamma related genes. It is allowing for that gene to now be expressed more.
But the mutation is in the tumor.
Yes, yes, by the tumors. So MHC goes up, class 1 MHC goes up, all of these things happen as a result of the interferon. TAP goes up, a lot of things change because you've now changed the interferon gamma gene expression within the tumor cell.
Any other question? Okay, great. So thank you very much.
Thank you.
Thanks. This was wonderful. So let's go to our next speaker. Our next speaker is Nacho Melero from CIMA, Universidad de Navarra, Pamplona, España, Spain.
So
the students of the course can't go. Can't go away. So Ignacio is now Doctor Honoris Causa from the University of Buenos Aires.
So it's a real pleasure to be here. I'm very grateful and honored for this honorary degree in the University of Buenos Aires. Every time we think from Spain about the University of Buenos Aires, very important names come to our minds. Obviously, Cesar Milstein was trained here in this university, and without Cesar Milstein and the monoclonal antibodies, probably none of us would be here. Very important people who were given Nobel Prizes for excellent work around biochemistry and physiology. Jorge Luis Borges, probably the best writer in Spanish after Miguel de Cervantes. And it was a real honor to have something like this. Gabriel Rabinovich and his team obviously are very good friends apart from colleagues. So my talk is to be around a molecule that happens to be expressed on activated but not resting T cells that goes by the name of CD137 and also by the name of 41BB. When Bjorn Kwon discovered this molecule, it happened to be a cDNA clone expressed by activated cells, and he gave to that cDNA clone that name. So in the previous talks, we have heard a lot about inhibitory mechanisms that can be blocked. So it's a matter of removing self-inflicted inhibitions. And it's amazing because if you look at the human genome and the immune receptors, very often you have more inhibitors than activators. So there must be evolutionary pressures for that. In the case of CTLA-4, seeing the knockout that was presented to us, it's obvious that it's related to autoimmune prevention. And polymorphisms in these genes are very much associated to autoimmunity. In the case of PD-1, probably things are different, and the evolutionary pressure comes from placentation. So what the tumors are doing is to use a placenta strategy to permit fetal maternal tolerance of alloantigens. But what I'm going to talk to you about is a different story. Rather than releasing the brakes, we are going to talk about acting agonistically on receptors, so kind of pressing the gas pedal. And the best gas pedal that you can have is the one that is expressed in the cells that you want to accelerate. So this can be achieved by the so-called co-stimulatory molecules. And costimulatory molecules come in many flavors. We have heard about CD28 and ICOS, for example, but many of them come in the TNF receptor family, and they are members of the TNF receptor family but are devoid of the death domain that recruits the caspase machinery that trigger apoptosis. Several members that belong to this family are CD27, OX40, 4-1BB, CD30, and HBM, and all of them can co-stimulate to some extent functions of T cells. To convey intracellular signals, they lack any known enzymatic activity, so to do so, they need to interact with adapters that send the signal downstream. As I mentioned, you don't have the dead domain. You don't have the dead domain, but you have these recruitment of traps that eventually lead to the activation of MAP kinases, and especially both the canonical and non-canonical pathways of NF-kappa B. And we think that that's very important. So this molecule I'm talking to you about is a member of the TNF receptor family without a dead domain, and that happens to be expressed on activated but not resting T cells. And from its discovery, it became obvious that if you mimic the action of this molecule with agonist antibody, or the only known receptor, that contact receptor that was discovered, which is 4-1BB ligand or CD107 ligand, you can stimulate proliferation, inhibit apoptosis, and enhance effector functions. However, in mice that are deficient of this molecule, there is no much of a phenotype apart from somehow higher susceptibility to die from influenza virus and other viral infections. And in humans, three families with mutations in this pathway have been described, and the patients have lymphoproliferative disease and problems to control herpes virus infections, including EBV. So the experiment that triggered everything happened a long time ago when Liping Chen and me were working together with Karel Hellstrom, and what we simply did is that we took antibodies that were agonistic for 4-1BB and injected them to mice bearing established tumors, and a good number of these mice developed CTL responses and rejected their tumors. So over the years, we have tried to figure out the mechanism. Very early on, it was possible to see that CD8 cells were involved, because if you deplete them with antibodies, you spoil the anti-tumor effect, and the tumors were heavily infiltrated by CD8 cells upon rejection. NK cells happen to express 4-1BB when they get activated, but we have not yet solved whether 4-1BB expression there is needed, although there were reports that you can enhance NK activity and persistence through that pathway. What's very clear is that you need dendritic cells to have anti-tumor effects. Dendritic cells could also express 4-1BB, but the important part here is that they are needed to cross-present tumor antigens to T cells. And what we were able to show is that if mice are deficient in conventional type 1 dendritic cells, the ones that are able to mediate cross-priming, and a matter that Sebastiana Migorena has studied in very much detail, then you completely lose the anti-tumor effects through 4-1BB co-stimulation. Another funny finding is that the tumor microvasculature expresses 4-1BB as a result of hypoxia. And we were able to publish that if ligated at that level, what you get is more expression of chemokines and of BCAM and ICAM1, so tumors get better colonized by T cells that now undergo diapedesis and enter into the tumor. And then there was something kind of embarrassing, and that's that the very same antibodies that we had made, when given to people who were working in autoimmune models in mice in which the cells that were mediating those responses were CD4, and then the autoimmune conditions ameliorated. And we don't know the exact reason for that. We know that might be some level of activation-induced cell death involved in these CD4 cells. And then Tregs express 4-1BB, and in the tumors they are expressing it at very high levels. But ligation at this level, we have not clarified what's happening. So there are still a number of mysteries behind this molecule. Regarding signal transduction, as I mentioned, it ligates to TRAF1, TRAF2. Now we have immunoprecipitated that and studied it with proteomics, and we know other members in the pathway. Actually, TRAF2 is supposed to work as an E3 ubiquitin ligase that creates K63 polyubiquitins that are docking sites for downstream signaling. We were able to see that the deubiquitinases, such as A20 and SIL, go to the complex and regulate signaling. And more recently, we have found, and we are sending this manuscript, that CAPs associated there are absolutely required for the anti-tumor effects and mediate the enzymatic functions going to through ubiquitination to NF-kappa B and MAP kinase. One of the things that we observed is that when we were treating these mice with antibodies, there were some level of periportal infiltrates that were absolutely mediated by CD8 cells, also some CD4 cells that were polyclonal in terms of TCR repertoire, but that were able to kill some hepatocytes due to apoptosis, as seen here in this tunnel assay. And two companies developed agonist antibodies for 4-1BB in order to develop them in the clinic. Bristol Myers Squibb developed utomilumab, which is an IgG4 antibody, and Pfizer developed utomilumab, which is an IgG2 antibody binding to a different site. The 4-1BB antibody by Bristol Myers binding to the cysteine-rich domain 1. It does not block ligand. The other one does. The other thing is that urelumab is probably a superagonist. It's extremely potent with intrinsic activity of the receptor. It's not very cross-reactive with cynomolgus, and that probably created a problem. And it had a very clearly single-agent activity in stage 4 melanoma patients and in B-cell lymphoma. Utomilumab is a very weak agonist. That's something that several groups agree about. It's very well cross-reactive with cynomolgus, with the monkeys, and Siegel-Gellinger reactivity has been only found in a few cases of Merkel cell carcinoma of the skin, but not in other tumors. So what happened is that after a pretty uneventful dose-finding, dose-escalation clinical trial, when exposing larger number of patients to this drug, a good number of them at doses above 1 milligram per kilogram started to develop high levels of transaminases indicating liver damage. And actually, at lower levels, these could be reduced, but at the risk of underdosing the target. And even at these doses, we are still having a small fraction of patients who develop elevations of transaminases. And it must be said that there were a couple of fatalities in those phase 2 trials, and these led to halting development and reassessing the maximal tolerated dose that eventually went to a flat dose of 8 milligrams per dose every 3 weeks. One of the features that became very prominent is that if you ligate 4-1BB, you dramatically increase the level of interferon gamma production by CD8 cells. And as you have heard here, interferon gamma is the main inducer of PD-L1 in tumor and immune cells, and therefore it made a lot of sense to combine 4-1BB agonists together with an anti-PD-1 or anti-PD-L1, and in many mouse tumor models, this happened to be synergistic. And this was driven into the clinic, and this is a clinical trial in which these tolerable doses of urelumab were combined with the anti-PD-1 nivolumab in a single-arm clinical trial in melanoma patient with kind of a promising overall response rate, but without a randomized clinical trial, we cannot know. A similar trial was undertaken by the Pfizer group, and this involved the combination of pembrolizumab with utomilumab, in this case at the high dose level. There were it was a single-arm clinical trial, no clear evidence for synergy, but at the same time, there were kind of unexplainable responses in small cell lung cancer, anaplastic thyroid cancer, etc. One of the features we believe 4-1BB is so active is that it has a tremendous effect on mitochondria in the T cells it co-stimulates. So you don't need to be an expert in confocal microscopy or in electron microscopy to realize that there is hyperplasia and hypertrophy of mitochondria if you co-stimulate T cells with agonist antibodies or with the natural ligand coated on two beads. This was not only an increase in mitochondrial mass, but also, and importantly, an increase in the transmembrane potential of the mitochondria, indicating more active mitochondria. And in seahorse assays, it was possible to see that basal respiration from the T cells was increased, as well as the maximal respiratory capacity of the mitochondria, indicating better fitness to survive, for example, in a hypoxic environment. And indeed, using a CD137 knockout mouse, it was possible to see that in tumor-draining lymph nodes, the T cells were having a lower mitochondrial capacity as a baseline phenotype of not being able to signal through 4-1BB, in this case, in the tumor-draining lymph nodes. Again, if you give to these mice an agonist antibody for 4-1BB, in vivo, you can recapitulate the mitochondrial hypertrophy and the gaining of function in the tumor-draining lymph nodes, indicating that you could be reinvigorating these cells in this sense. So there were several genes that could have accounted for that, and we went after a transcription factor which promotes mitochondrial biogenesis, but very importantly, there is a protein that is involved in fusion of mitochondria and in the generation of crista in the mitochondria, which is OPA1. And what we were able to see is that if we silence OPA1, as opposed to when we are using control siRNA, we were spoiling part of the tumor effect, indicating the involvement of OPA1 in this mitochondrial effect. Another thing that probably goes to some of the epigenetic changes that have been reported here is that with the help of Manel Esteller in Barcelona, we analyzed genome-wide DNA methylation in human T cells with or without co-stimulation with 4-1BB. So I'm going to use just one slide to show you that we observe a clear change in the methylation status of the genome of these lymphocytes, leading to higher hypomethylation of the cytosines in there. And we had a list of genes. Some of them have clear and important effects in that we confirmed were upregulated, and these include a microRNA, CCL5, that attracts CDC1s, and also of the TCF7 locus that encodes to TCF1. So this is very interesting because by having been co-stimulated once in the presence of 4-1BB, T cells remember that that happened and become more prone to respond better in a subsequent antigen engagement. But a key word around 4-1BB over the years have been synergy, meaning that with 4-1BB agonist therapies, it was easy to have combinations that rather than additive were synergistic at inducing tumor control. And you can even have a decently successful career taking you to a honoris causa degree at the University of Buenos Aires by proving that you can combine synergistically 4-1BB agonists with a number of therapies, including conventional therapies such as chemotherapy, radiotherapy, surgery, other monoclonal antibodies you have heard in previous talks, CTLA-4, PD-L1, vaccines. It's very difficult to find a therapy that has some effect that it happens not to be potentiated by anti-4-1BB. But if I were to choose a really potent synergy, that would be adoptive T-cell therapy. So I'm going to use a few slides. So this is the kind of synergy that you don't need a mathematician to show you that it's happening because single treatments do not do too much. And if you put adoptive T-cell therapy, in this case with T cells that recognize ovoalbumin and that you are familiar with the OT1 TCR transgenic cells, together with anti-4-1BB, you eradicate all the tumors. But with a limit, because if you postpone treatment of B-system over melanomas later, the synergy doesn't go as well. There are many interesting things. You see more T-cell infiltration. The other thing is that if you are using OT1 cells that do not respond to 4-1BB, you lose the effect, but you also need CD137 expression in the endogenous repertoire of the receiving and treated mouse. And if you don't have these endogenous cells, you get an effect, but eventually all the tumors relapse. And having these conditions in place, we could perform intravital microscopy. And this is a lot of fun, but basically what we did is to place under a glass chamber tumors that happen to be red because one of the histones is labeled with a cherry protein. Then the T cells are green because they are EGFP and they recognize ovoalbumin, as I told you. And we can monitor this over time, and we can also see the vessels because there is an intravascular fluorescent dye there. And we recapitulated the anti-tumor effects and the increase in tumor T-cell infiltration. So I'm not telling you that it's not related to that. But what was more striking to see is that if you were watching these cells in the tumors, there was a clear change in the behavior. If you track them, they were staying kind of longer on target, and they were behaving differently. And if we look at that, even in longer footage, we were able to see that they stayed longer on target, and those tumor cells started to show nuclear degeneration suggestive of apoptosis. And that means that it's not only an increased number of T cells, it's that we are changing the mind of the killers, which is an important thing to do. So maybe there is a perfect marriage here, and it has been shown by other groups as well as ours, that if in a tumor lymphocytes infiltrating tumors, you select out those that are expressing 4-1BB at a given moment, indicating recent stimulation through the T-cell receptor, you enrich for fitter T cells that give rise to a better yield of the cultures. And if you keep the agonist antibodies during the culture, the yield of the culture is also better in terms of tumor cell recognition. And in a previous talk, it was told to you by Sebastian Amigorena, much better than me, that the C137 cytoplasmic tail signaling from the CARs is very good at keeping T cells alive and permitting this phenotype. And then we keep seeing these hot spots from where tumor T-cell infiltration is happening that we believe are related to this phenomenon of favoring tumor migration from acting on the tumor musculature. So is there a future? Well, we hope so, and we can try to target co-stimulation to tumors rather than in the whole mouse. We can try to have some kind of conditional molecules that will only work in the tumor. Certainly, it's a good idea to have synergistic combinations. We can even think about intratumoral delivery strategies, encoding genes or whatever. We can have combinations with neoantigen vaccines, for example. So let's see. So one of the ideas that we have been testing is intratumoral injection of the antibodies, but another way to go is to make the antibodies in such a way that would be only operational in the tumor. The other strategies, what can do is to target 4-1BB co-stimulation to tumor-associated antigens or to elements present in the tumor stroma, such as the fibroblast-activated protein. So this idea of the conditional antibodies, we tested it together with Cytoms and Bristol Myers Squibb, and this is the so-called probodies. In this case, the light chain of the anti-4-1BB antibody is linked to a masking peptide that doesn't let it bind to antigen, and this linker here is sensitive to proteases that are enriched in the tumor microenvironment. And by means of doing so, what happens is that when these antibodies get exposed to tumors or to the recombinant proteases, then the antibody unmasks and it's able to bind to the T cells, and this actually happens to levels comparable with the probody and the father or the parent antibody that has not this masking effect. And the nice thing here in a tumor that is very sensitive to anti-4-1BB monotherapy, which is the CD26 transplantable colorectal carcinoma, what we were able to see is that the probody antibody had a very similar effect as compared to 1D8. And very interestingly, that the level of transaminases and the presence of liver CD8 T cells was much attenuated as compared with the parent antibody, although not totally abolished, maybe because the tumor here is a cleaving antibody that will eventually recirculate and, importantly, go through afferent lymph to the tumor-draining lymph nodes. When you have a safer way of using an antibody against 4-1BB, you immediately think, as in the previous talk, in neoadjuvant therapies. And actually, the group of Michelle Teng was the first to report that neoadjuvant approaches in mouse models were a very good idea to treat tumors. And the combination she was using was anti-PD-1 plus anti-4-1BB after some screening. And in this scheme that Michelle Teng invented in Australia, in which the tumors are spontaneously metastatic and give rise to lung metastases, spontaneously treatment with these antibodies, anti-PD-1 and the anti-4-1BB antibody or the probody prior to surgery. So surgery is postponed a couple of days after treatment, clearly reduced tumor metastases. And the probody was able to do it almost as well as the parental anti-4-1BB antibody, leading to much better survival of the mice. The probody also preserved its synergistic activity with adoptive T-cell therapy, and this probably opens the possibility of using 4-1BB co-stimulation in vivo. Another group that developed a conditional antibody was the group in Chugai. This is a Japanese company affiliated to Roche. And what they generated was a very clever idea. This is an antibody that only binds to target in the presence of high concentrations of ATP, as usually observed in solid tumors. And in this case, they were able to see that this was true and that the antibody could bind target. And using CD137 human knock-in mice, the mice now express a human 4-1BB hooked up to the signal transduction machinery of the mouse. And they were using these mice with conventional transplantable tumors and giving their antibody that binds only to 4-1BB in high ATP conditions as those in the tumor. They were able to see very nice anti-tumor effects using this conditional antibody. So it's time to go back to the clinic. So we go back to the future. And the first story I'm going to tell you is something that was developed by PRED, the Roche European franchise. And they were using a FAP, a fibroblast activating protein, targeted 4-1BB ligand that is an agonist antibody. In this setting, the construct is an FAP monovalent binding antibody hooked to a trimeric 4-1BB ligand and to an inert FC part of an antibody, a highly flexible molecule by cryo-electron microscopy. And the concept here is that you won't get cross-linking elsewhere, but around fibroblasts that are activated. And those are only in the tumors and in scars in healing tissue. So it seems to be probably a good idea to avoid liver side effects. And the group of Pablo Umaña in Roche was able to show all that in a paper published in Science Translational Medicine, and the whole concept was active all the way to humanized mouse models. And therefore, we started a dose escalation clinical trial. And this dose escalation clinical trial was a single agent dose escalation in a conventional way, but with some or with a two-cohort overlap with a combination with atezolizumab, which is a PD-L1 blocking antibody, again testing the combination. And it was the usual type of patients that we get in the phase 1 clinical trial units of heavily pretreated patients with no other option to treatment. And the first good news is that the antibody was the construct was pretty safe in monotherapy. There appear some cases of rashes that were not surpassing grade 3 and were controlled by local steroids. There was a few cases of pneumonitis, but did not require hospitalization, only in one of the patients. And there were a few cases of other complications. And in combination with atezolizumab, the pattern of toxicity is very overlapping what we see with checkpoint inhibitors. But most importantly, this treatment was almost entirely liver safe, and no cases of transaminase elevation were observed. As a monotherapy, there were some cases where tumor shrinkage was observed, and 2 responses could be objective in a case of carcinoma of the thymus and in a renal cell carcinoma. In the combination with atezolizumab, more activity was spotted, but with only one exception in which the patients had been previously declared checkpoint inhibitor refractory patients. So the jury is still out whether this is adding something on top of PD-1 blockade. But from a pharmacodynamic point of view, it's very clear that in monotherapy, the construct was doing something very interesting, and that's because we could detect in peripheral blood increases in proliferating CD8 T cells and T cells expressing 4-1BB, as well as in soluble 4-1BB and in other cytokines such as CXCL10. And in the cases where we had pair biopsies, we could see that there was an increase in the level of CD8 infiltration, both in the stromal tracts and in the tumor cells, and those CD8 cells tended to be expressing more Ki67, indicating that they were in the cell cycle in a state of proliferation. In a sub-study that we did at our institution, we labeled this moiety with zirconium 1889, which permits PET imaging. And comparing PET imaging with glucose, with FDG, to these, we could see that we could, in most of cases, see the tumor, as here, this presence in the stoma and some of the malignant lymph nodes there, and also in the lung metastases. Interestingly, you cannot image by PET, FDG PET, the brain, because it's very metabolically active and cause glucose, but here we detected a small metastasis that was actually confirmed in the patient that had been unnoticed previous to this. So the second story in the clinic I'm going to tell you is about a bispecific antibody made by Genmab and BioNTech. BioNTech is the company that by generating mRNA vaccine probably saved a large part of the world, including Spain, during the pandemics. And this is a bispecific antibody that binds to PD-L1 and 4-1BB. So the logics here is that this bispecific is intended to bind PD-L1, preferentially in the tumor tissue, and once attached to a solid phase on a plasma membrane, be able to cross-link 4-1BB in neighboring cells. In order to make this possible in an IgG4 format, there is a technology that permits in an irreversible way the swapping of heavy chains in this IgG4, and then it would create this type of biology. And in this case, the phase 1 dose escalation clinical trial was straightforward. We were testing progressively increasing doses in the usual, with the objective of determining the maximal tolerated dose and figure out the safety and try to get some signals on whether this molecule was active. Remember that here you are blocking PD-L1 at the same time you are intending to produce 4-1BB agonist activity. From the point of view of safety, this was less clear in the sense that we observed that transaminase elevations of grade 3 and over were occurring in about 10% of the patients, and in some of them we were seeing milder elevation levels. I have to say that never with bilirubin increases or with signs of liver failure. Pharmacodynamically, again, and in peripheral blood, it was very easy to see, beyond a certain dose level, that we were seeing increases in circulating interferon gamma, CXCL10, and proliferating CD8 T cells in peripheral blood. And there was something funny, and that's that beyond 45 milligrams per patient, we observed this phenomenon, but this got attenuated at higher doses of the compound, which is very intriguing. From the point of view of waterfall plots that were presented previously by Pam Sharma in a number of clinical trials, there were some cases in which monotherapy happened to induce tumor shrinkage. It was amazing that most of these patients who are progressing very rapidly when they enter clinical trials had a control rate, and in these cases, all of them were had been previously treated with PD-1 or PD-L1 agents. In an expansion that we are running with patients with non-small cell lung cancer, at the time of this cutoff, there were another 3 responses in patients which seemed to be durable. This gentleman is in my institution, is much durable, but actually around here now. But the point is that these patients, again, were non-small cell lung cancer patients, either adenocarcinoma or squamous with previous exposure to a PD-1 or a PD-L1 agent. So we were talking about this curious dose effect, and it's really curious. And one of the problems is that when you are acting agonistically, you can overshoot the target because monoclonal antibodies are bivalent, but only bind to one epitope in the target moiety. And because of that, at a certain level, you can be inducing cross-linking at an optimal level, but if you increase the concentration of antibody, you can stay in a fashion where you are not leaving sites, binding sites for cross-linking. In addition to that, if you are ligating in 2 separate cells, such as you have 2 targets, for example, an Fc receptor here and the 4-1BB molecule here, and you need a binding in both sites. If you happen to saturate both without allowing cross-linking, then you end up with bimodal dose-response curve, which is very interesting. So yeah, the future is here, and it's kind of funny. Everything related to biotechnology tricks, but things look very interesting. So you will see that we have now a very good number of agents targeting 4-1BB in the clinic. Actually now, if I count correctly, 12. And we believe that we could be doing something better. That would be using adoptive T-cell therapy, and you know, TILs and CAR T-cells are around, especially for solid tumors. And combinations, as alluded in the previous talk, are probably the good way to go, and there are several options. So these are the many antibodies that are in the clinic right now, most of them based on the concept of multi-specificity. We can term that bispecific, etc. Many of them are relying on targeting PD-L1 as well as 4-1BB in many different formats of the molecule. I commented to you about one of them, but others are in dose escalation. Then the other strategy that is very popular is targeting molecules that happen to be expressed on the surface of tumor cells with some level of heterogeneity, but permit targeting the tumor tissue in solid tumors with these kind of agents. And we are talking about 5T4, Glypican-3 in hepatocellular carcinoma, CEA in colorectal cancer and other digestive tumors, EGFR, HER2. With HER2, we have already heard about data. It's a very complicated, after trastuzumab deruxtecan, it's a very complicated scenario, but they are reporting on around 60% overall response rate using this in previously treated with anti-HER2 agents. Mesothelin, this could be a very good idea, for example, in ovarian carcinoma and pancreatic cancer. PSMA, this has already started clinical trials with Crescendo, very interesting. There are other targets, including CD47, trying to exploit here a dual biology, also in permitting phagocytosis of tumor cells by blocking the interaction with SIRP1 alpha. In the area of hematological malignancies that I know very little because I treat patients with solid tumors, there is a CD19 4-1BB ligand, very similar to the one I told you, with a trimeric ligand that is also made by Roche. They have an impressive overall response rate when they combine it with a T-cell engager, and that's an area that we are focusing also in solid tumors with other T-cell engagers. There's a molecule that we have started clinical trials, and it's in our institution as well, that is dually target CD40 on APC and 4-1BB on T cells. This is undergoing dose escalation as we speak. They are other bispecifics. And as I mentioned at the beginning of the second part of the talk, FAP is also pursued not now only by Roche, but also by Boehringer Ingelheim. So you probably know these characters. My distinguished colleagues probably don't know that. So I'm translating to English because they speak in Spanish. No? And say, haven't my lord had too much trouble and meager success with these C-137 adventures? And Don Quixote goes and answers, Sancho, my friend, for many years we have ridden together. Immunotherapy is the most difficult of the wars, and 4-1BB perhaps the most singular of its battles. So this is where I work. This is in the University of Navarra, Spain, in northern Spain, close to the Basque Country and the French border. It's an interesting place because I happen to work in a teaching hospital, Clínica Universidad de Navarra, and in a research unit. And translational and reverse translational research is about building bridges. So I cross this street many times a day. The bridge doesn't exist, quite unfortunately. Now I have to build a much larger bridge because I have been appointed professor of oncology in Oxford University, so I will be traveling. But that's a much longer bridge. But I think we will manage. These are the people who did the work, preclinical and clinical. I'm very proud of them. Actually, we should be recruiting, so you'd be able to find me in PubMed, and I would be very happy to interview people from Argentina, because I must say that I had a wonderful student in the early days in Pamplona, Guillermo Mazzolini. He had to go back to the University of Austral because they were graduating students that day, but he promised me that he's watching the recorded talk. The companies are also instrumental. Without them letting us drive their prototypes, probably we would be, it's a thing close to nothing, but it's a very interesting time. So thank you for your attention, and happy to answer questions.
Thanks so much, Nacho. It was a wonderful talk. Great, great. So it is open for questions. Any question from the audience? It can be in Spanish, and we translate. So, buenas tardes.
No, yo hablo español muy bien, ¿eh? No.
Sí, sí.
Peor que Jorge Luis Borges, claro.
Well, I will start with one question. So I didn't get very well the concept of the ATP-dependent antibodies, because in most tumors you have CD39 and 73, and you get adenosine from ATP, so in most immunosuppressive tumors.
Right.
So how do they function, these antibodies, in those immunosuppressive tumors?
Even with this adenosine synthesis pathway going on, still it's surpassed by the amount of ATP that is there. Part of it is because of tumor necrosis, especially in the areas of hypoxia. There is a lot of ATP. It's very well measured. But you are right. If you start metabolizing that out through the ectoenzymes, you end up with less ATP. And that potentially makes sense for a combination with oleclumab, in order to further increase the ATP. At the same time, you are blocking the adenosine synthesis, and therefore the inhibitor, the immunosuppressive effects through A2A receptor and A2B receptor.
Great.
Nacho, I love your talk. I'm going to steal half your slides, I think. I wanted to ask about if the immune system has this yin and yang, and you know the TCR, CD28 gives you CTLA4, interferon gamma gives you PD-L1. What do you think is happening when you do stimulate 4-1BB? Like, what is that pathway that's trying to control it? Is it one? Is it multiple?
Yeah, you're absolutely right. There are mechanisms that would come up that could be immunosuppressive. But what I think here is that we are doing something that doesn't exist in nature. So there is no way you would be ligating like crazy or all the 4-1BB that it's in the body. So we are doing something pharmacological, not physiological. And once you enter in that realm of doing something, it's like the apprentice of wizard, no? So we have to learn, but we believe that we are impacting many aspects of the T-cell biology, and in a way that it has a seal. It's kind of an effect that lasts longer. And I wish we would totally understand that. But you are right. I mean, we face the same potential obstacles of TGF-beta being around, the myeloid cells being around. So we ended up doing something very funny. I have still 2 minutes. So, and that's that we wrote in Nature Cancer a review about combinations. No? So I draw a palette like a painter with the brush and the different colors. And actually thinking, we totally agree, Padmani, that combinations are very important. But then the editors told, take out your palette and write a very boring table. So I did that. But when this came in the July issue of Cancer Cell, of Nature Cancer, then the cover was a palette and say, oh, they stole my idea. No? So I went there and in the counter cover, it said that artistic credit goes to Dr. Ignacio Melero. And I said, oh my goodness, because my wife is not here now because she left with some relatives she has in Argentina. But it's very funny because my wife thinks that I'm unable to have any artistic taste. No? Actually, she chooses all my ties because of the color. But I went home very proud with the issue of cancer. You see, artistic credit given to mine. But what I did all this humor just to tell Padmani that combinations are the way to go, and certainly 4-1BB monotherapy is not going to do it, but could be a very good ingredient to concoct a good cancer treatment. Is that a good response?
Yes. Any other question?
You have 1 minute and 16 seconds.
So, my last question.
Oh.
So, I want to know your opinion about why 4-1BB in CAR T cells enhances persistent and memory T cell phenotype compared to CD28.
Yeah, well, the short answer from the textbook is that you engage the BCL-XL pathway very potently with 4-1BB through the NF-kappa B pathway. Then the mitochondrial phenotype seems to be important according to Carl Jung. He has an immunity paper reporting on that. And there must be mysteries. And I think that this chromatin reprogramming that we started to scratch around is extremely important. These cells, maybe in the line, Dr. Amigorena, Dr. Honoris Causa Amigorena, reported before, might be very important. We think that chromatin reprogramming. Now, instead of talking, you remember from the course, staying late here, studying signal 1, signal 2, signal 3, IL-12, and interferon gamma. You must remember that. It is going to be in the exam. So then the thing is that about these TNF receptor molecules, Tanya Watts and others use the nomenclature of signal 4. And signal 4 is it's a way whose main reason, evolutionary reason probably, is to keep the cells alive, because they are selected because we believe you are an antiviral lymphocyte. You should go on, become memory, persist, and do so. And that's how we interpret that, but we can be totally wrong. I'm typically very, very bad at interpreting evolution because I am an MD.
Okay, so thank you very much, Nacho. So after these wonderful talks, we have 10 or 15 minutes. So let's say 10 and you take 15. So 10 minutes, you have the coffee break there, and then we go back to our closing lecture by Larry Steinman, and we will move from cancer to autoimmune diseases, inflammatory diseases, and all the drugs and the discoveries before the closing session. So maybe we can start with our last session. Everybody wants to go to dinner, so let's start. Pablo, could you please call the people from the coffee break to come? Thanks. Great. So, we are just getting to the end, and it will be not the very end, but the end. So, we will go to our last talk, and we will shift from cancer, from immunotherapy in cancer, to immunotherapy in inflammation and autoimmune diseases. You know, the last part of the course, that we talk about multiple sclerosis and all the targets. So the person who identified all these targets is the person who will give us the talk. So our last speaker is Lawrence Steinman, now Doctor Honoris Causa from the University of Buenos Aires.
Thank you very much. And it's a pleasure to be here. It's a little bit difficult to be the closing lecturer because there are people who want to have the rest of their Friday afternoon. But what I want to talk about today is exactly the subject that Gabi mentioned, multiple sclerosis. But we've heard brilliant lectures all day long about cancer. And we're not shifting actually 180 degrees at all. The whole idea of cancer therapy is really concordant with the idea that we want to get our immune system to attack a tissue in our own body and kill it before it kills us. So that's one area of commonality. Another thing is the brilliant work that we heard in the first lecture in unleashing the immune system. One of the side effects, the most important effect, is you get to live, and as things are developing, we can talk about a cure. But one of the issues that comes up, nothing is for free. Many people get an autoimmune condition. The last thing I'm going to talk about this afternoon is what happens when we know what the actual antigen is that's triggering the autoimmune disease. Is there a way to tolerize to that antigen without perturbing the whole immune system so the tumor escapes? And it looks like there are some possibilities. So the title is, another area that is common is the idea of the niche. And we talked about the tumor niche. There's also the issue of the brain niche. And I'm going to talk about something that again intersects with one of the most famous immunologists from the University of Buenos Aires, Cesar Milstein, and his work on developing effective monoclonal antibodies. But I'm going to show you that within the niche of the central nervous system, the body's own immune system makes monoclonals to many of our common viral infections. And they dwell there for a very long time. And that fact led to understanding what the trigger is for multiple sclerosis. So on this slide, there's the campus where I've spent 42 years on the faculty and where I trained as a neurologist. On the right is an insect called a katydid, and I made that for my granddaughter to try to explain the concept of molecular mimicry. It gets a lot deeper than that. The title of the talk is Trapped in the Brain, Deciphering the Specificity of the Clonal Immunoglobulin to Explain the Pathogenesis of MS. And then I'm going to show you that there's actually a hot zone in one of the transcription factors for Epstein-Barr virus that encodes not one but two chloride channels of interest. So here's my connection to the University of Buenos Aires. I actually have to read this slide because it's too complicated and I don't have a genealogy diagram. So on the right is my father. He was an immigrant from a city called Zhytomyr, which is now under missile attack, probably as I talk, and my mother. On the picture here on the left is my grandmother, and next to her is my cousin Esther. So to read here, the University of Buenos Aires has had its share of remarkable graduates. They include Nobel laureate Cesar Milstein, who made possible monoclonal antibodies. The first woman graduated from the medical school at the University of Buenos Aires in 1889. Pictured is my cousin Esther, who graduated from the University of Buenos Aires around 1950, and her two sisters, Sonia and Leke. Esther's father, Ruben, was my grandfather's cousin. Ruben's mother was the sister of my grandfather's mother. That's why I'm reading it. So Zhytomyr in those days was what we would call a genetic bottleneck. In Jewish folklore, they call it a shtetl, a little village. But that's all I want to say at this point. So Epstein-Barr virus. We've only known about it for about 50 years. Actually, 2014 was the 50th year, so now we're up to 58 years. Epstein and his technician, Barr, who later got a PhD, were the ones who discovered it. Dennis Burkitt, a surgeon working in Uganda, noticed that there were children who had head and neck tumors that were lymphomatous, and it was called Burkitt's lymphoma. So over the years, there's been a high index of suspicion that Epstein-Barr virus has a lot to do with triggering MS. And we're going to immediately hit a conundrum because of all the people in the audience here. About 95% or more have been infected with EBV and have positive EBV serologies. So the first question anyone is going to ask, I'm cutting off a lot of questions by raising them myself. How can you say that a virus that has infected 95% of us is the trigger for any kind of disease at all? And actually, Epstein-Barr virus, I'll talk about it as the trigger for MS, but it's the trigger for a lot of malignancies besides the Burkitt's lymphoma. We've been studying it for a long time, and one of the things we try to do in our work is we use a lot of animal models, and I'll show you, but we also use human tissue. But we're not as fortunate. I'm envious of my colleagues in oncology. They have surgeons who take out the tumor. Many of the tumors anyway are, they call them liquid tumors. They're in the blood. This is a disease in the brain. There are lymphoid-like structures in the meninges, but we don't get to biopsy it very often unless somebody thinks that it's something other than MS. And then if it's biopsied and it turns out to be MS, people are actually skeptical that the surgeon should not have done the procedure in the first place. But we did a study a few years ago looking at many of the Epstein-Barr viral genes, and they're in the brain of MS patients, and we could see short mRNAs called EBER. We saw latency in lytic viruses, LMP1, BZLF. The trouble is we didn't have any good p-values in that paper because we didn't have a lot of brains to study. But what we were able to do is show that in almost all the MS brains we're seeing a lot of latent and lytic viruses. Lytic virus was predominantly in the chronic stages of the disease. And then the second paper down here is time correlation between mononucleosis and initial symptoms of MS. So an electrical engineer at Stanford whose wife developed MS after very late onset mononucleosis decided he would use his engineering mathematical skills to look at data from the Karolinska. And his conclusions were that there's an invariant time constant between MS and mononucleosis. And he also found in the studies we published with him, but he was actually our teacher, that males and people with high EBNA1 were prone to develop MS more rapidly following infectious mononucleosis at all ages. So at the medical school, I headed the immunology department for about 10 years, and I gave some lectures, and one of the lectures that I gave, of course, was on MS. And I would start the lecture by, until January of 2022, by asking the question, what causes MS? And the correct answer was, we don't know, it's multifactorial. And then you can read down the list of the various factors. But something very exciting happened in January 2022. A paper was published from the Harvard School of Public Health by Alberto Escudero, who studied a database of 10 million US military veterans who would get their blood examined upon entry into the military and about every year thereafter. So this was the paper that came out in January 13th in Science. 10 million patients, and I think it's written here a 32-fold risk factor, and this black belt epidemiologist went out and said in this paper that Epstein-Barr virus was the cause of MS. So embedded in the data, they had approximately 805 samples that they had longitudinal measurements of various EBV serology. 804 of them were positive. They also had 35 individuals who entered the military and who were EBV negative and who went on to develop MS, and 34 of the 35 converted to serologically positive. So the editors at Science asked me to write an editorial. I combined forces with Bill Robinson, who I've been collaborating with for over 20 years. We mentioned that story about the 800 out of 801 cases, and then 34 cases were negative, and 35 of them went on. And we wrote this editorial. Inside the paper was some supplementary figure. And this is important to you people who are working scientists or who want to be scientists. We all have different skill sets that we bring to the game. So one of the things that's useful is to cooperate with people with complementary skill sets. So Escudero was an epidemiologist, and he worked with a virologist at Harvard, Stephen Elledge, and they had a viral scan. And the viral scan, this is supplementary figure 1, they didn't comment much on it, is that EBNA1 between peptide 365 and 420 had the highest p-value for associating with MS. That p-value has 4 zeros, then a 7.3. There were some other EBNA1 fragments that were 0034. They did mention a capsid protein that has a high association. I'm going to talk about a little bit about how the field has changed since January 22, and the fact that Moderna is working on an mRNA vaccine to EBV that might indeed include this capsid, so we have to be aware of the pitfalls. So we talked about various mechanisms on how a virus could infect cells, and this virus, EBV, has a particular tropism for the B cell. And we talked about that in the little perspective we wrote, and we mentioned molecular mimicry. Now, while all this was brewing, the other journal called Nature had our paper, and they had it for a long time. I was talking to Gabi about this during the recess. The enemy is us, because us, meaning we scientists, are reviewers. So they'd ask for one thing, they'd ask for another thing, and we pointed out to the editor that this striking paper is coming out on January 13th. Our paper, they sent us a pre-galley. Couldn't you publish it simultaneously with Science? They said, no, what you need to do is put it on a preprint server. So we put it on a preprint server, and then we could talk about our paper, and our paper came out 3 or 4 days later. It doesn't matter now, but again, since I'm talking to students, you're going to run into this problem as you build your careers. Jim Allison said in the first lecture that if you're going to go into science, you better love it, and you're going to have many disappointments, and some of the disappointments are going to be large. When you're young, even the small disappointments seem larger than maybe they need to be. But one of the problems we run into is this problem with peer review. So I'm going to share a lot of data in this talk that if somebody asks me to email the paper because you don't have a subscription to Nature, I can't email it to you because it's about 70 megabytes spread over 30 pages unless I make a PDF and reduce the file size a couple of times. The ordinary reduce the file size won't even cut it. Now let's get to some science. So in the News and Views, Hartmut-Veckerle wrote, to quote his words, understanding factors that lead to the development of MS may aid efforts to develop new therapies. Clinical data now implicate a viral culprit and immune system dysfunction. And then most people who study MS propose the factors underlying initiation of the disease come from outside the brain. I'll show you how that happens. The debate about the nature of these factors has split researchers into two main camps. Most see autoimmunity as the driving factor for the illness, but a minority invoke viral culprits. So there's a compromise solution, was the way Hartmut diplomatically wrote it. And yes, there is, and both sides of this argument were correct. I'm going to show you what this molecular mimic is, but there's some interesting backstories as we go along. So in almost every paper that I write that I think may have impact, I often write something like this in the last sentence. Our results provide a mechanistic link for the association between MS and EBV and could guide the development of new MS therapies. We didn't list them, but they would include antivirals, an EBV vaccine, or what I'm going to talk about at the end of the talk, how we now know what the antigen is, how we might be able to do a tolerogenic strategy to get rid of it. So how did we find the antigen? So another digression. You're going to hear a commonly stated aphorism that we, in science, stand on the shoulders of giants. But this is an interesting picture because these are street entertainers in Madrid. And they're not scientific giants, though they're standing on each other's shoulders so they'll look bigger. But the photograph was taken by a giant, Ramon Cajal, who won the Nobel for showing how the brain is composed of cells with cell bodies and axons, and also his students defined the glial cells. So this was a picture of people standing on each other's shoulders taken by a giant. And then another thing to remember as students is that we use a lot of technologies now that would have taken a long time if it were 10 years ago, 20 years ago, 50 years ago. So one of the technologies that enabled this, in the field where I work, multiple sclerosis, we've known that there are clonal antibodies in the spinal fluid. In fact, it's one of the ways that we decide to make the diagnosis. There's a few other diseases. We see it in syphilis, we see it in Lyme disease, Borrelia, but generally we don't see clonal antibodies very easily unless somebody has, for instance, a monoclonal gammopathy in their lymphoid populations. So this was a clue. But the work came from Tiselius, who won a Nobel Prize for electrophoresis, and then people who developed the ability to electrophorese spinal fluid samples. Denise Karcher in 1959 showed that this works better on paper, doing paper electrophoresis, and now it's a certified diagnostic test. Do you have oligoclonal bands or do you not have oligoclonal bands? So these are the three key individuals who were involved in the work. Tobias Lanz came 14 years ago to Stanford from Heidelberg, and he learned immunology in my lab from Peggy and Bill Robinson. Bill Robinson is now the chair of rheumatology at Stanford. So we started out looking at the spinal fluid, and we see that in the CSF there are IgG plasmablasts, which really don't appear in any appreciable amount in the peripheral blood. So we also were able to see, let me just go back here, in the peripheral blood. Bill had developed for cancer screening a technique for being able to barcode and pair heavy and light chains and then do single cell analysis of plasmablasts. So this is how we got started at looking at some of the clonal expansions that were of interest. And there were certain immunoglobulin heavy chain V gene families that were overrepresented in the CSF. V3 through 7 were preferred. But another thing was very interesting. And by the way, here's Cajal. And about 1992, we published in Nature that the main integrin molecule for homing to the central nervous system was alpha-4 integrin. And on the 20th anniversary, the drug had been approved. The artists at the Journal of Cell Biology drew this picture of lymphocytes with alpha-4 binding to VCAM and doing something called diapedesis, moving through the blood vessel. Well, 100 years earlier in 1918, Cajal had studied the same thing. We would have cited him for doing this 100 years ahead of us, but I didn't know about it. I saw this drawing at an exhibit of his sketches. So what happens is that, as I said, alpha-4 is important for the cells getting into the central nervous system. But when they get into the central nervous system, they lose alpha-4. The underlying mechanisms behind that are being studied. But it's a situation where they can get in, but they can't get out. And these plasmablasts can dwell in the central nervous system for a very long time because the oligoclonal bands in MS are quite persistent over a long time. So the situation is set up to begin to ask, so what are they binding? And believe me, many people had that idea, and many people saw that they were binding to many molecules of interest along the way. So in this slide, here you can see in the blood, integrin alpha-4 is high. The plasmablasts in the CSF, it goes down considerably. So in these analyses of the clonal populations, you can see that in the blood, this looks like the sea creature called sea anemone. It's very spiking. You don't see these, in a clonal family, these members of the family that show somatic hypermutation. as if there's some antigen driving a very specific adaptive immune response at the B cell or plasmablast level. And this is a very important part of the work. So along the way, Tobias made about over 100 monoclonal antibodies from this collection in CSF from patients. And then we had done a lot of work with antigen microarrays, and we made a microarray that was both customized, and there were many other components we could buy off the shelf. So we had 23 recombinant latent and lytic EBV proteins. We had 2 entire EBV viral lysates. We had 240 peptides spanning 4 prominent EBV proteins, including EBNA1. But we also had lysates from 7 other viruses that over the course of understanding what these oligoclonal bands were, people had identified. And there's a major group of clonal bands from they call it an MRZ reaction, from measles, rubella, and varicella zoster. So we had those lysates. And we put them on planar arrays. And we asked, so what are the various monoclonals that we had shown somatic hypermutation and that we had made in sort of mini industrial quantities. And the monoclonals, some of them bind measles, rubella, and zoster. They didn't have much affinity for CMV, although there's some CMV binding as well. But in the extensive EBNA1 binding, there was a particular region that many of the monoclonals were binding to, and I'll get to that in a second. There were an ability to do proteomics on some of the binding, and we saw this region between amino acids 394 and 399 that was proline rich, 2 prolines, then 2 arginines, then 3 more prolines. And it's a very disordered region of the protein. It doesn't have a lot of structure per se. And binding studies were done that had respectable nanomolar binding. But we also learned, and I'll show you in a coming slide, that some of the neighboring serine residues were very important for increasing binding. So I have here KD measurements for the germline and KD measurements for one of the monoclonals. In the germline, before all the somatic hypermutation, actually there's a lot of binding to EBV from the germline antibody, but it increases after some phosphorylation events. I'll show you those events, but Chris Garcia, who's in the same building with us at Stanford, a Howard Hughes investigator and a brilliant X-ray crystallographer, collaborated on this part. And again, one of the messages is epidemiologists were giving us input. We're doing sort of classical immunology, molecular immunology, but having a crystallographer close at hand. We heard about it in Lori's lecture about her collaboration with Greg Pesco, which is some beautiful pictures. Anyway, the heavy and light chains interact with these prolines, and the binding is very strong because there's a proline cage that comes around this component of EBNA1 and those antibodies that are in the CNS. So another piece of technology that you could just buy, a collection of the entire human protein. So we got human protein microarrays representing 16,000 proteins spanning 80% of the human proteome. And we asked, what are the antibodies binding? Again, we're exemplifying it with a couple from the collection. But at the top of the let's see. Oops. Yeah, okay. I won't attempt to use the laser at the top of that gel. You can see on the heat map, there's intense binding to a molecule called GlialCam. I had never heard of GlialCam, but somebody pointed out to me that in some paper we had written about 10 years earlier, it's in one of our supplementary tables, but we had no idea what to do with it. So GlialCam, it's a glial cell adhesion molecule. It's expressed in oligodendrocytes. Those are the cells that make white matter, and astrocytes. It's involved in chloride and water homeostasis. And there's an ultra-rare disease where there's a mutation and it causes something called megalencephalic leukoencephalopathy. And I can say that very fast because I do pediatric neurology some of the time. It's called MLC, which has a different meaning to many of us immunologists. And it's a chaperone of a water channel called aquaporin 4. Now, about the same time, Thomas Olsen at the Karolinska had sent a paper to PNAS where he discovered a molecule called anoctamin 2, which is also a chloride channel, and its molecular mimic is to EBNA1, and it's just a few residues away. So independently, we had the epidemiology from Alberto Escarrillo. We had input from top-rate virologist Stephen Elledge. They put their data in a supplementary table. We had a separate paper from Thomas Olsen and colleagues about a chloride channel anoctamin 2. And here we're finding that there's another chloride channel related protein called GlialCam. And by the way, the other proteins that are reddish, orange, etc., they need to be investigated. So I don't know if the whole story is solved. We were talking about the phosphorylation of the serine residues. I think it comes up on the next slide. But they've been doing genome-wide association studies in MS forever. And of course, HLA is the most prominent association. But there are a number of kinases that are, they call them minor hits with a relative risk factor that's much lower than what you see for HLA DR1501. And I'll show you what phosphorylation does. So when the serines at 376 and 377 get phosphorylated, binding gets much stronger. So I brought up this conundrum. Almost everyone in this audience has EBV positive serology, and hopefully no one in this audience has MS. And how does that come about? So you may need some secondary pathological events to happen, such as getting those serines phosphorylated. So that part of the story has to get worked out. Then one of the things we wanted to know is how prevalent are these antibodies to the GlialCam and anoctamin 2. So the short story is we don't know yet. We're undertaking, in collaboration with the Karolinska, longitudinal studies on CSF. But I can tell you from the peripheral blood, and you can see it a little bit here in panel N. This is what I'm telling you. This particular figure goes to panel Q, and each one of those figures is an enormous amount of work. It's about, there's a high background, but about 25% of patients in their peripheral blood at one point in time have this GlialCam antibody. We don't know in bigger studies of CSF how much more common it is. And all of the appropriate controls with a scrambled peptide are involved. And Chris Garcia made X-ray pictures of both the phosphorylated and non-phosphorylated molecules. And let's see if I can go back and show the binding. Didn't project very well here. This is just more that the, again, it's certainly not 100% at any one point in time in the peripheral blood. It's about 30%, 25% people will have positivity. So we have to put that part of the story together, and we're, as I said, collaborating with the Karolinska group, and we also have a collaboration going on that U.S. military database. Alberto Escorillo has been very generous. We have also studied the T cells in individuals with MS. These are peripheral blood T cells, and they're stimulated by GliaCam. We have examples that they're making interferon gamma and IL-10. There should be another slide around here where they're, I guess that panel, the flow cytometry panels didn't show up in some kind of translation from the group that I sent, but they make gamma interferon and granzyme are characteristic of the T cells that we can find in the peripheral blood. So we did another thing. About a century ago, there's an idea from Robert Koch about called Koch's postulates, that if you want to prove that something caused a disease, if it's an infectious agent, you transfer the agent to an appropriate subject. It better be an experimental animal, and see if the transfer of the agent causes the disease in the animal. Well, you can't put Epstein-Barr virus into a rodent. The tropism doesn't extend to the rodent world. So we took the peptide, and there's this 90-year-old model of MS or neuroinflammation called experimental autoimmune encephalomyelitis. So when we give the GliaCam peptide in the EAE model in the red, you can see that the amount of paralysis is worsened. 1 is a weak tail, 2 would be a hind limb, mild paralysis, and we were getting up to more severe paralysis. So that's some version of Koch's postulates. So we go back to this supplementary figure 1, and between 365 and 420, there's the Anoctamin 2, the Karolinska contribution. There's what we saw with the work on GliaCam. There's some other groups that have reported other interesting molecular mimics to other myelin proteins in that region. How could one region have so many hot spots of similarity between molecules in the nervous system and a virus? So here they are laid out. For interest, it's a highly disordered area of the EBNA1. And then there's a lot of diseases where there are immune responses to transcription factors and small ribonucleoproteins. We see this in lupus and other diseases where there are intracellular proteins that are used as routine serology to help make a diagnosis of the disease. So I mentioned at the end of the talk, and we're nearing the end, I was going to talk about what could we do if we had a technology to simply shut off the immune response to GliaCam or Anoctamin 2 or any antigen of interest. And I think that this kind of work could apply to checkpoint inhibition therapy if we knew the antigen that's involved in individuals who get inflammatory bowel disease or other autoimmune diseases. What could be done just to silence that so you get your cancer cured and you don't have to have that other burden of an uncomfortable but not life-threatening autoimmune disease? So in about a year ago, it was 2021, January, the group from BioNTech, and you can see Ugur Sahin, Özlem Türeci, Caitlin Carrico, Ari Weissman is in Mainz at the university. they showed that if you modify the nanoparticle with 1-methyl pseudouridine, they were able to really impressively reduce experimental autoimmune encephalomyelitis caused by a myelin protein. So about 20 years earlier, and we have taken this all the way through a phase 2, we used now, which seems like the underdog strategy, we made DNA plasmids. We think DNA isn't as good as RNA for viral vaccines, but who knows in tolerance. But we published in 2003 using protein microarrays. We made 4 DNA plasmids to 4 of the major myelin proteins, and we engineer the backbone. There's non-coding DNA called the CpG motif, which activates the innate immune system through toll-like receptor 9. So we took many of them out. And in our work, we were able to, with that cocktail of DNA, reduce the relapse rate in a mouse that we followed for 90 days. The relapse rate went from about 1 relapse a month down to 1 relapse in, or 1.5 relapses over 90 days. And we had these microarrays with all the various myelin proteins, and you can see with the cocktail we're able to turn that yellow hot set of antibodies that are made to myelin proteins in the model and really turn them icy blue. We also added IL-4 in one of the groups, and that improved things a bit. We then took that into a phase 2 trial. The FDA only wanted us to tolerize to 1 myelin protein, so we chose the most abundant one, myelin basic protein, and we ran the trial for a year. The primary endpoint was what happens in early in the trial versus how many MRI lesions you would get between weeks 28 to 48. And we reduced them by about 50%, but the p-value was 0.07. There was a secondary endpoint where the p-value was 0.05. At that time, 2 things were happening. The monoclonal antibody to alpha-4 was really knocking down the disease. It reduced relapses over a 2-year period by about 75% and reduced MRI lesions by 90%. And Novartis came up with a drug not quite as good as the monoclonal, but you could take orally. So we sort of put this on the shelf, and I would call this a pretty big disappointment after all the work. And the investors who put money into this company lost a lot of money, but it was a valiant attempt. This just shows graphically what I was talking about. The 0.07, there was a 50% reduction. The 0.05, that secondary pre-specified endpoint was a 61%. But we couldn't compete with what I call the big hammers. And we also saw in this work that the more active the disease was, the more efficacious was the tolerizing strategy. So more recently, we have gone back to these experiments to tolerize to GliaCam. And we took out not just some of the CpGs, but we took all of them out. And there were 232 CpGs in the DNA backbone, and we took them all out. And this is just some of the early data. We're able to reduce the severity of EAE. In the middle panel, 50% of the animals were disease-free. The DNA plasmid encoding a scrambled peptide rather than the GliaCam peptide. There were no disease-free animals, and relapse-free, you can see we reduced from about 80% to 30%. So I don't know what BioNTech is doing, but I do know that they have more resources than what we're doing. But we'll see what happens. This is the kind of fun challenge. I think we're the underdog. But getting back to the subject of off the shelf for some of the revolutionary therapies of students, and thank you for the honor bestowed on me today.
Thanks so much, Larry, for this inspiring talk. So, there are questions now? I hope there are from the students.
A really large, really big stretch, but derived antigens are usually tolerogenic in the gut. And then also there's this work from Jennifer Gomerman that shows that anti-inflammatory IgA plasma cells can migrate to the meninges. And I was thinking if you considered orally delivering the antigens so that you can induce tolerance and maybe these plasma cells can differentiate there and migrate. Maybe it's really crazy, so you can tell me.
I'm not sure I understand your question.
Oh, sorry. I was wondering if you thought about orally delivering the antigen.
Oh, sure. Yeah, I think oral tolerance has been another, people in the world of intellectual property patents talk about it as a long sought after goal. In an experimental animal, it's been known for a long time, you can give an antigen orally that can shut down a disease like EAE. Getting it to work in humans has, it's been tried but not successfly. There are now some brilliant formulation chemists. I'm thinking of people like Bob Langer at MIT. Maybe Bob could come up with some nanoparticle to deliver an antigen with one pseudo-uridine or a construct. So there, I wouldn't rule out oral. My attitude about oral medicine, I know some people are needle phobic, but if I have to take a therapy that works through a needle and it's a disease that could kill me or disable me, just put up with the needle. But I know that the pharmaceutical industry covets having oral therapies.
Thank you.
So, Antoniela Sabeki and others kind of trace back the evolution of the antibodies and see what was the kind of the original antibodies binding to. And are these the result of affinity maturation and that by accident you end up with the cross-reactivity, or even the primary response to EBNA1 was doing it?
There's pretty good binding from the germline. Once they undergo somatic hypermutation, it gets a little bit better. And there's a lot of hand-waving at this point that maybe it's the phosphorylation that increases the binding a little better, but I'm really struck at how strong germline binding was, and even why there's germline binding to this ubiquitous antigen. I think that's very interesting unto itself.
And the plasmablasts in the central nervous system, in the cerebrospinal fluid, are they immortalized by EBNA1 and EBNA2, or is that possible?
Yeah, it's a very interesting question. It's possible. There's no evidence. You know, there's another connection when you said immortalized. So the saga of Cesar Milstein, graduate from University of Buenos Aires, working out what it actually takes to make industrial quantities of useful monoclonals. A scientific neighbor of mine and a good friend who passed away, his wife is still active, Len Herzenberg, when he was doing a sabbatical in 1976, he coins the term hybridoma. So your question triggered this thought in my mind: is the EBV infection of the B cell parent leading to the plasmablast, in a sense, making them hybridoma-like? Something to think about. Again, these are kind of literary thoughts. We need some science.
Thank you for a really interesting talk here in the back. I was wondering, you mentioned that, of course, not everyone that gets infected get MS, and I was thinking about the cancers that are induced by the same virus, and if there's any...
I think we're good.
Somebody switch off the light. No problem. But it's not that we need to go.
Okay, see if there's any evidence of a role of EBNA1 in cancer induced by the same virus.
Well, EBV is responsible for some gastric carcinomas. I feel a little bit awkward talking about oncology in front of all these world-class, but there's a good association between gastric cancer and EBV, and nasopharyngeal carcinoma too. So EBV's a bad player, except that all of us are infected and all of us look wonderful for a Friday after 6-7 hours of lecturing. So. It's a good question. Where's our fearless leader?
Thank you very much, Dr. Steinman.
So we will now move a little bit to the closing ceremony and to close and to conclude. So before going to the conclusion, it's impossible to finish this immunotherapy meeting without doing a tribute to my friend Enrique Merri, who passed on August 27 this year. Was a great friend and a great colleague who supported us a lot, an Argentinian scientist, and I would like to read some words about him. So Enrique Merri was a professor in the Department of Microbiology and Immunology, director of the AIDS Malignancies Scientific Interest Groups, and co-director of the mentoring program at the Miami Center for AIDS Research. He was director of the CFR Sylvester Argentina Consortium for Research and Training in AIDS Malignancies. He had the idea to put together scientists from Argentina. We were a group from Argentina, then Pedro Can from Fundación Huésped, then Omar Coso from IFIBIME and Facultad de Ciencias Exactas, and the University of Miami, and talk with the NIH and the NCI to do a consortium together. He passed on Saturday, August 27, after a battle with lymphoma. He was one of the first that characterized the role of Kaposi sarcoma herpes virus in lymphomagenesis in 1996, and did a lot of work related to these viruses and Kaposi sarcoma. So I just wanted to do a tribute to him. He really did a lot of basic and translational research work that was extensively published and was widely cited, and he developed a lot of antiviral and anti-tumor approaches to treat these Kaposi sarcoma patients at the time that there was no cure for these patients. So, in memorial of Enrique Merri, who was twice honored by the government of Argentina, his home country, for his scientific contributions. Most recently, earlier this year, he received from the Argentinian Ministry of Science and Technology. He bestowed him on the highest honor of Raíces Award. So, thank you very much. And we will miss him a lot. There are a lot of Argentinian sciences all around the world, but really Enrique always wanted to help us a lot Diego is there. Diego is one of the part of this consortium, and we want to continue with the consortium, but he was really essential for us. So I just want to say some words to conclude and thank for your inspiring and outstanding talks. We really enjoyed a lot, and what we learned, I don't want to say many things because we are all starving, we want to go for dinner, and we want to celebrate this, but what we learned is that it's at least 2 or 3 important things that are the common denominator between our outstanding speakers. We need to learn from the clinics. We need to learn from the patients, as Dr. Sharma said, as Dr. Allison said, as Larry Steinman said, as Nacho Melero said, and Sebastián Amigorena said. We need to go from the patient to the bench, and then again, back again, from the bench to the patient, and then from the patient to the bench. And this is the real world, not only from clinical trials, but also from the real world. Also, we have the future. There is another revolution. We spoke about the revolution of immunotherapy, but there is another revolution of immunotherapy, and the revolution is the combination. It was clear from Dr. Sharma, it was clear from Dr. Allison, and it was clear from all the speakers from Nacho and Larry. We need to find combinations of different immunotherapeutic strategies and to find another landscapes, to go out of the box and to find another type of landscape, epigenetic landscape, metabolic landscape, the microbiota, and to look at other components of the machinery. So thank you very much for all the wonderful talks, and I hope you enjoy staying in Argentina. And also want to thank to all the students of the course for staying 10 days, 11 days, from very early in the morning until the night, until the evening, in the Faculty of Exact and Natural Sciences. And now we would like to do some special thanks, probably in Spanish. Maybe we move a little bit to Spanish. So Ada and Vicky, we can come here. Sí, there. Empezamos a dar en orden a los speakers.
Sí, ¿te parece que se lo bajamos?
Sí, se lo bajamos, sí. So, the diploma that you received was from the university, but we want to give you something from the Symposium of Immunotherapy, from us. And this is just to remind you forever and to put in your office or to put anywhere. So, we can go to Dr. Jim Allison, Dr. Pam Sharma, Dr. Nacho Melero, Dr. Larry Steinman. Sebastián. We will give to Sebastián in the dinner. You can show if you want. Ahora quisiéramos, we shift a little bit to Spanish, quisiéramos agradecerles a todos. La verdad es que ha sido un esfuerzo grande, pero un placer enorme poder organizar esto. Desde el momento que se nos ocurrió una idea en el 2018, que parecía muy loco traer al premio Nobel a estos gigantes enormes, y le planteamos a muchísima gente que esto iba a ser posible. Y bueno, y llegamos a este momento donde realmente fue tan emocionante hacer terminar con el curso con todos y no querer irnos porque ya estábamos tan encariñados. Así que bueno, hay mucha gente que tenemos que agradecer y queremos agradecer porque en esta vida no nos podemos ir sin agradecer. Así que, bueno, un agradecimiento enorme Primero a la primera persona a quien yo le conté que quería hacer este curso y que aceptó, y un aplauso grande para Jorge Geffner. Jorge me sigue en todas las locuras y en este no fue la excepción y lo volví medio loco, así que, bueno, el segundo agradecimiento, vamos a empezar con lo institucional y después vamos a otros. Son sorpresivos. Después queremos agradecer a la Facultad de Ciencias Exactas y Naturales, porque es el lugar donde que nos permite hacer docencia en forma libre, y que este curso era un curso no tradicional, era un curso diferente, un curso que traía clínica, que traía pacientes, y desde la gestión anterior de la facultad a la gestión nueva aceptaron y bueno, y nos apoyaron para poder llevarlo adelante, y lo pudimos hacer en el edificio más hermoso que hay, en el cero más infinito y en el aula más hermosa de ese edificio. Así que queremos invitarlos a pasar a Juan Carlos Reboreda, el ex decano de la Facultad de Ciencias Exactas y Naturales, y a Vale Levi, la vicedecana actual de la facultad. Sí. Gracias. ¿Una foto? Bueno, vamos. Foto. ¿Sale? Está difícil, ¿no? Es que se ató mucho.
Al medio.
Al medio, bueno. Gracias, muchas gracias. ¿CONICET? ¿O el ministerio? ¿Al CONICET? Sí. Bueno, obviamente nosotros pertenecemos al CONICET y tenemos la camiseta muy puesta del CONICET, y esto lo hacemos en representación al CONICET. Y bueno, no siempre comulgo con los presidentes que hay en CONICET, pero esta vez sí. Así que bueno, mi amiga Ana Franchi. El BIM. Sí. Bueno, y nosotros trabajamos, nuestro laboratorio está en algún lado, que ustedes no vieron, los estudiantes, y nuestro laboratorio está en un lugar que nos cobija desde el año 2007 y donde estuvieron los premios Nobel. So we work, I translate a little bit, we work in the laboratory that Bernardo Husay, the Nobel Prize, the first Nobel Prize of Argentina, work. And so we want to invitamos a la directora actual del IBIME y a la representante de la Fundación IBIME, a la directora Victoria Lux y a la representante de la Fundación Damasia Becu.
Gracias.
¿Viste? A ver. Sí. ¿Qué le ponemos?
Nómbralo si querés.
¿Algún representante del Ministerio de Ciencia, Tecnología e Innovación? Matías, te toca a vos. Sí. Un representante del ministerio. El ministro no. Lo esperamos un poquito más. Ah, bueno, entonces, en representación. Gracias. El ministerio nos dio este lugar y nos ayudó muchísimo con toda la parte creativa y artística para que todo pueda salir adelante. Y bueno, obviamente que esto no fue totalmente gratis hacerlo, así que hay mucha gente que nos ayudó, y si hay algo que hice un nuevo doctorado en este tiempo es en pedir dinero. Así que, bueno, toda esa gente merece un reconocimiento, porque la verdad es que confían en nosotros con los ojos cerrados porque esto podía ser bueno o no. Así que queremos reconocer a las fundaciones sin fines de lucro que realmente apoyan la ciencia argentina y que sin ellos no podría ser posible hacer este tipo de eventos y tampoco podría ser posible hacer ciencia. Así que en representación a la Fundación Sales, llamamos a José María Candiotti, el presidente de la Fundación Sales. Gracias.
¿Sabés la foto? ¿Dónde la tendríamos que sacar? En el medio.
En el medio, bien, bien. Bien.
Porque va a ser más representativo. Ahí está. Muchas gracias.
Bien, gracias a vos. Bueno, como representante de la Fundación Williams, que también nos ha ayudado muchísimo, Martín Oliver. Martín. Gracias, Martín. Gracias. A ver, una fotito. Así vamos al maneo, así para hacernos sirve. Sí. Gracias. Martín.
Esto va a la vitrina.
Bien. Como representante de la Fundación Bungeborn, a Asunción Zumárraga. Bien.
Gracias. Gracias, Miguel.
No, por favor. Bien.
Gracias. Gracias, Che. Gracias a Dios.
¿En qué edad?
Bristol. No, la Fundación. Sí,
Estamos con el representante de la Fundación, Carlos Barón, René Barón. ¿O lo guardamos? ¿Paola? Se nos tuvo que ir.
Bien, guardamos.
Guardamos, igual que la uva. Bueno, y hubo dos laboratorios, que uno muy relacionado con algunos de los drug targets, con ipilimumab, que es BMS, Bristol Myers Squibb. Así que bueno, queremos invitar, ahí lo veo, a Mariano, no sé si está Tamara. ¿No? Gracias. Muchas gracias, Mariano. Gracias. Y el segundo laboratorio que nos salvó la vida, y pudimos dar muchas becas, es el laboratorio Vago Bioprofarma. Bueno, y hubo dos sociedades científicas que también sponsorearon y nos ayudaron mucho. Una es la Sociedad Argentina de Inmunología, a la cual queremos mucho y a la cual pertenezco hace 20, 30 años casi. Así que en representación a la secretaria de la sociedad, Mariana Salatino, que también es investigadora de nuestro equipo. Dios. Bien, ¿y? ¿Hay alguien de la Asociación Argentina de Oncología Clínica que haya quedado? Martín, ¿algún socio? Bien, no, ya es retarde, la guardamos estas. Y bueno, y hay personas que ayudaron mucho, pero mucho. Realmente, there are people that were instrumental for organizing this meeting, que realmente se pusieron al hombro todo, que pusieron el corazón, el alma, y que cuando yo les propuse la posibilidad de hacer esto y les dije no, pero un curso y además un simposio, y además tenemos que invitar gente de afuera y tiene que ser gente grosa, gente muy buena, y además de todo tenemos que tener en un curso oncólogos clínicos y había que hablar con las fundaciones y había que hablar con todos y había que conseguir becas y había que hacer coffee breaks y había que hacer todo. Y no queríamos contratar una coordinación y hubo 2 personas en mi laboratorio que dijeron, nosotros queremos, y no solo queremos, sino que hace 4 años que vienen trabajando con un motor impresionante, con un compromiso, con mucho amor, con mucha pasión y jamás perder esa sonrisa que las caracteriza. Yo las respeto, las admiro profundamente y entonces, bueno, quiero reconocer a Ada Blindner y a Victoria Zumblad. Un aplauso enorme.
Si sabía que había todo esto, lo hacía seguro.
¿Quieren hablar?
Estoy muy emocionada. Bueno, gracias. Fue un placer hacer este curso con Gaby, coordinar este curso. Fue mucho trabajo, pero el esfuerzo nunca está mal, siempre para algo es bueno. Así que estamos súper contentas. Gracias y me alegra mucho que lo hayan disfrutado. Tuvimos muchas buenas repercusiones. Todos nos dijeron que les encantó, que quedaron agotados pero extenuados y de alegría. Así que encantadas y it's a pleasure to coordinate this symposium with you. It's amazing for me. So thank you. Thank you for coming.
Es medio repetir lo mismo, pero bueno, fue mucho trabajo, pero valió la pena. Estuvo muy bueno y bueno, qué suerte que... Muchas gracias, Gaby, por la oportunidad de hacer estos trabajos y generar estos encuentros, estas actividades. And it was a pleasure to have you here to visit us and to listen to your conferences. So, thank you very much.
So they are the heroes, they are the people that really support it. Y imposible tampoco terminar sin agradecer a una persona que también que se engancha cada vez que le propongo algo y además de eso, cuando le dijimos, we said, we want to have here Pam Sharma and Jim Madison. And said, well, but they will say probably no, they travel a lot, what we can do. And Laura Bober said, I will convince them.
Thank you. Thank you so much. I didn't expect this. My contribution was great, but minimal because I was all the time talking with Pam and Jim and the secretary and the girls, you know, Vicky, and it's amazing all what you did, and of course you, because you are the man of all this, and really you deserve the successful that you get with this amazing meeting.
Y finalmente, hace aproximadamente 2 meses se sumó a nuestro laboratorio una persona que iba a trabajar en la parte administrativa nada más y que se la robamos a otra parte del instituto. Entonces hablé con Ana y le dije, no hay otra persona para reemplazar porque yo quiero a esta persona. En 2 meses hizo cosas como si fuesen 1 año y nos ayudó un montón. Así que, ¿está Amalia? Siempre es invisible, siempre está ayudando un montón. Ay, Amalia. Vení, sacamos la foto. Gracias. Bueno, buenísimo. So with this, we will close the session. We wish you the best. We will go for dinner with our speakers. And we wish to all the students, les deseamos lo mejor en todo. Espero que hayan disfrutado mucho el curso. Para nosotros fue un placer tenerlos. Aprendimos mucho de ustedes y nos hicieron mucho bien en un momento también que necesitábamos organizar esto. Así que esperemos repetirlo, no sé, cada cuánto. El año que viene no, eso seguro que no. Next year not. Probably in 2 years. Pero realmente fue un éxito y hay algo que tengo que decir, es que toda la gente que invitamos a dar clases, y fueron muchos por día, muchos por día, ninguno nos dijeron que no. Y un aplauso también muy grande quisiera para todo mi equipo. El equipo, mi equipo de trabajo, todo mi laboratorio. Entonces, an applause to all my lab, the young students of my lab, the investigators of my lab that are there, over there. And yes, it's great to have students like you that not only join in papers and experiments and every day, but also in this kind of events. It's very important for Argentina, and it's very important to have you in the lab. So thanks so much. Muchas gracias a todos. Buen viaje a los que tienen que volver al interior del país. Muchas gracias por todo y bueno, cuenten con nosotros para lo que necesiten.